and the recording is in progress. So I am gonna call. Can everyone hear me? I want to say first, welcome to those in the room. Good morning, and to those online. And we now have several hundred online, depending on your time zone. Good morning, good afternoon, or good evening. I'm Drew Bremer. I'm the director of the Office of Nutrition Research here at the NIH, and it's an extreme honor and privilege to welcome you to this joint NIH FDA Nutrition Regulatory Science Workshop. The foundations for this workshop began many months ago, and it's so cool that we're all here today to see the product. But the topic of this workshop really could not be more timely. I'd like to first start with gratitude. Gratitude for those who are speaking and presenting and who will be actively participating in the workshop sessions over the next 2 days. And those of you who are online, joining us remotely, all across the country and all across the globe. Second, I would like to thank our organizing committee and the moderators who will be working the workshop over the next 2 days. And just to make sure I don't forget anyone, I did bring my cheat sheet. And so those of you who don't have a program, they're out in the back. But I do encourage you to have one so you can follow along and appreciate and see who the people are who made this happen. So starting with the organizing committee, comprised of individuals from the NIH as well as the Human Foods Program at the FDA. I want to say thanks to Sam Addis, Allison Brown, Rebecca Buckner, Kelly Casabal, Steve Hermansky, Kirsten Herrick, Claudine Cavanaugh, Helena Castro, Sherry Shelley, Jill Reedy, Ashley Vargas, and Krista Zanetti. I also like to take pause for a moment and a special thanks to my scientific co-lead and dear friend from the Human Foods Program at the FDA, Dr. Robin McKinnon. Also, in the spirit of thanks, I want to recognize Mark Dennis and the team from the Scientific Consulting Group Incorporated who've helped with all the logistics. And finally, last but certainly not least, Kim Barge from our office. Now, I promised Kim I wouldn't embarrass her with hundreds of people watching online, but without Kim, this wouldn't be happening today. Kim not only is the glue that holds our office together, but she has this amazing ability to pull things together, and she is the logistical mastermind of today's event. So Kim, thank you. Over the next 2 days, you'll be hearing a lot from investigators in this space. And the intent of this workshop is to really highlight how science can generate and provide the evidence base to inform food-related policy and regulatory decision-making. That's super important. The science is driving the show. And as important, how the NIH and FDA, by working together, can support the requisite science to address priority areas and priority gaps in nutrition research. And with that, I would like to pass the baton to my colleague and dear friend, Robin McKinnon.
Thanks so much, Drew, and welcome. Thanks and welcome to all here in the room and those who are joining us online. And again, adding my sincere thanks to all who are joining today for this considerable effort, this investment of time and energy to be here today. So in the newly established FDA Human Foods Program, where the vision is for the food supply to be a vehicle for wellness for all, focusing on the areas with the greatest potential impact to benefit, to protect and promote public health, nutrition, microbial food safety, and food chemical safety. And in the nutrition space, we're focusing squarely on what we can be doing, what more we can be doing to protect and promote public health and to reduce the burden of diet-related chronic disease. Now, as Drew mentioned, the goal of the workshop is to focus on how nutrition science can generate evidence to inform food-related policymaking and regulatory decision-making. And we're looking to do this over the next day and a half. What we want to do is identify the critical research gaps, the research resources, and the priorities in this area, and to highlight how nutrition science can be conducted in a way that can inform food-related regulatory decision-making. So we'll also provide some specific examples of how the agencies have worked together in a couple of key areas. So this morning's session, that will help lay the groundwork for the workshop. We'll be looking at the roles, the responsibilities of the different agencies, show how we've worked together in a couple of these key areas. And then this afternoon, we'll dive into some key topics for nutrition regulatory science, including ultra-processed foods. And then tomorrow, in the final session, we'll focus on the research resources that are available, some that are existing, some that could be augmented, and some that are on the horizon. There is a lot more that we could be covering in this day and a half, but time is not unlimited, unfortunately, or maybe fortunately. But the plan here is at least to stimulate the thinking in this broad area of nutrition regulatory science and to help chart a new path forward so we can all work together to be doing what we can to advance and protect and promote public health. And now for some introductory remarks, we're going to hear first from the NIH Director, Dr. Monica Bertagnolli, and then we'll hear from Dr. Rob Califf, the Commissioner of Food and Drugs. So Dr. Bertagnolli will be hearing some remarks.
Hello, I'm Dr. Monica Bertagnolli, Director of NIH, and I'd like to welcome you to this very important workshop on nutrition regulatory science. I'm sorry I cannot be with you in person, but I want to thank you all for attending. And I'd like to thank the FDA for co-sponsoring this event. I especially want to acknowledge Dr. Califf for his support. As we hear frequently from Dr. Drew Bremer, the Director of the Office of Nutrition Research, or ONR, here at NIH, nutrition is everyone's business. We all eat, and the food we eat directly impacts our health. This is ancient knowledge, familiar to people for millennia, and yet we still have so much to learn. NIH currently invests $2.2 billion in nutrition-related research annually, supporting investigators around the world who are focused on understanding optimum nutrition and exactly how nutrition relates to health and the risk of disease. Nutrition is bound to all aspects of health and disease, and the large majority of NIH institutes and centers, 24 of the 27 total, engage in research related to nutrition. This research is coordinated at NIH by ONR within the Office of the Director. We currently support the single largest national investment in nutrition research, the NIH Common Fund's Nutrition for Precision Health Project, powered by the All of Us research program. This workshop is an important opportunity to think about how we can be more coordinated and effective in our research on nutrition science so that we produce evidence and data that better inform policies relating to food and regulatory decision-making. At NIH, we feel very fortunate to have a close working relationship with our sister agency, the FDA. Although this is the first joint workshop that focuses specifically on the topic of nutrition regulatory science, we've held a number of joint workshops before, and our two agencies are in constant contact on issues that affect public health. Since 2021, an NIH-FDA Joint Leadership Council has helped us make sure that regulatory considerations are integrated into our planning for biomedical research, and that the latest science is fully incorporated into the regulatory review process. One important component of this council has been an active joint agency nutrition working group. A good model for how research can better inform regulatory decision-making is the NIH FDA Tobacco Regulatory Science Program, which you'll hear more about today. As for regulating nutrition, NIH has made many contributions that have informed FDA decisions. Among them, NIH supported the research and evidence base that helped lead to the FDA's decision first to require labeling and then to remove partially hydrogenated oils or artificial trans fats from the U.S. food supply. NIH also supported the science linking excess sodium intake to an increased risk for hypertension and cardiovascular disease, which led to dietary recommendations to limit sodium, and more recently, to the FDA providing voluntary targets for the food industry to lower sodium content in their products. NIH has also led research efforts that supported FDA's requirement to declare added sugars on nutrition labels. Research is clearly crucial to generating the evidence we need to inform regulatory decision-making. This kind of work is a core part of the NIH mission, which is not only to advance science, but to use scientific advances to improve public health. A suboptimal diet is now the leading risk factor for morbidity and mortality worldwide, surpassing even the hazards of tobacco smoking and of hypertension. This workshop could not be more timely. I appreciate the time and energy involved in planning an event like this. So thank you to those who made this workshop possible and to our Office of Nutrition Research for supporting it. I also want to thank all the speakers and participants for your presence, whether on campus at NIH or attending virtually, and for your contributions to these very important discussions.
Yeah, it's great to be with y'all today. I have a lot of fond memories of Natcher, but I haven't been here in a while. I used to spend a lot of time here in Natcher. I was upset initially there was no coffee when I got here. I find, as far as I can tell, the evidence on caffeine is getting better and better as time goes on. Thank goodness. And I do want to, of course, thank the NIH and Dr. Bertagnolli, and want to join her and welcome you here today. She's been a tremendous partner as we're preparing to transition back to the private sector, where we'll join many of you who are in this conference. I think all of you, or most of you, have probably heard that I do feel that urgent action is needed in this arena. Chronic diseases, including heart disease, cancer, and diabetes, are the leading causes of disability and death in America. And they're also the leading drivers of $4.5 trillion in annual healthcare costs in this country. I'm not used to being in last place. I was a competitive basketball player. I like being in first place. But we're now clearly at the bottom of the pack of large high-income countries in our life expectancy. And then an article that just came out last week in JAMA, we also earned the distinction of having the most number of years spent with disability during those years of living that we have. I was astounded. I helped start a medical school in Singapore, and in a recent visit there with our FDA colleagues, they asked for an urgent meeting about their major public health problem. We were excited to spend a half a day. The problem was half of their teenagers are expected to live to be age 100. So they're trying to figure out what to do with those years between age 70 and 100. Not a problem that we're experiencing quite to the same extent. The health and life expectancy numbers in our country have been going the wrong direction for far too long. And we have to put tackling diet-related disease at the top of our collective priority list. And this, of course, means conducting high-quality nutrition research. Improving the American diet to help food become a vehicle for wellness requires a whole of government and whole of society effort. And we, the NIH and FDA together, are at the center of this. FDA with our accountability for food safety and labeling, and NIH is our national research engine. But let's not leave out CDC, USDA, and others in this regard. I think particularly right now, the USDA will be critical. And we're at the FDA. We at the FDA are committed to do our part. We just went through the largest reorganization in our history to be organized to take this on. I had the chance to do a fireside chat with Francis Collins yesterday for FDA employees. Both in his leadership times and with Dr. Bertagnolli, collaboration between FDA and NIH has been a priority with issues like tobacco, rare diseases, and of course, nutrition. I'm especially glad that tobacco will be highlighted here because I think that program has really hardwired in a number of important elements. As Dr. Bertagnolli mentioned, NIH efforts have been critical, and the PHO issue has already been discussed, and I'll just emphasize it again. And of course, the role of sodium in hypertension and stroke, and the role of sugars in obesity, diabetes, and ultimately cardiovascular disease and cancer. While there are myriad factors influencing health, the science is well settled to support the concept that some basics exist to guide diet recommendations: less sugar, less sodium, and less saturated fat. With robust science, we can then apply our regulatory tools, like those related to labeling and in food safety, to take action that can inform consumers and move the food industry to offer healthier choices. In the case of sodium reduction, strong scientific evidence, much of it supported by NIH, demonstrated the increased risk of developing hypertension and heart disease from excess sodium intake. The average sodium intake in the US has been about 3,400 milligrams per day or above for many years, far above the upper limit recommended to reduce risk of chronic disease of 2,300 milligrams per day. Using the evidence generated by NIH and others and applying our authorities and expertise, we issued draft and final guidance for 2.5-year voluntary targets for the food industry to reduce sodium across a range of food categories to support reductions down to 3,000 milligrams per day. And just recently issued a new draft target to further support reductions down to 2,750 milligrams per day. This is another milestone in our iterative stepwise approach to sodium reduction. Here, I'll just interject, since I'm a free person now, to say what I think. One important thing for people to know is in the process of doing this, I had numbers of people, and I was here in 2016 as commissioner, numbers of people screaming at me to make this mandatory. And I had some members of Congress saying that if we made it mandatory, FDA budget would be cut to zero. And so the voluntary target was a matter of trying to find the middle ground among very vehement opposing groups of people in our country. And we just got some data, which I presume all of you have seen, that while 2,750 is higher than the limit, we're getting there. And preliminary assessment from NHANES shown here shows that the industry does seem to be responding, and we're seeing differences here in what our population is taking in. So the key point is that when we have the research and the data, as well as the resources and authorities, we can move the needle on diet and the industries that produce our food. Remembering that most of the sodium that's ingested is not coming from the salt shaker, it's coming from processed food that's baked in by the industry that produces the food. I'm not declaring victory for obvious reasons. The common quote pertains, as far as I'm concerned. American diets produce disease because unhealthy food is the easiest option. Once we get beyond these basics on diet, we have a lot of uncertainty, but make no mistake about it, getting these basics right would take us far down the road to a healthier country. How much money has been made on books and diet programs that recommend extraordinarily different dietary patterns all at the same time? We need a much stronger basic research engine to understand where to go beyond these fundamental issues. There's a clear association between the consumption of foods considered ultra-processed, for example, and negative health outcomes. But we also know there's a great deal of overlap with foods high in added sugar, sodium, and saturated fat. And we at FDA are already acting where the evidence is clear enough with, for example, the updated definition of the term healthy, which is work in process that you'll hear a lot more about very shortly. We urgently need more high quality data and research to take appropriate targeted action, realizing that any action we take will be subject to the same political influences I just described about sodium. And the ultra-processed food issue is one that I hope this workshop will clarify in terms of what the research enterprise needs to deliver. Beyond ultra-processed food, we also need a much more robust research agenda related to contaminants and nutrients in food and on the impact of different regulatory actions. It seems highly likely that dietary supplements, for example, will be a hot topic in the coming years. With over 100,000 supplements on the market and over $160 billion per year in sales, we have a lot of advertising and marketing, but very little evidence about what's beneficial, what's detrimental, and what has no effect except on the wallet. The pre-market requirement for drugs and devices forces the industry's research engines to answer crucial questions, but no such authority exists for dietary supplements or for food. We need the public research engine to pick up steam and take these issues on. I'm also really glad to see a section on implementation science. Our national culture of individualism and First Amendment rights for corporations as well as people gives us a limited tool chest of regulatory approaches in an overwhelmingly unhealthy food environment, especially for people with limited resources. The tension between individual choice, the power of advertising, and the growing evidence that tools like graphic warnings and government controls used in other countries are effective in improving health. This is a conundrum that begs a solution. What is the American solution? That, to me, is real implementation science, figuring that out. Finally, many of the more complex issues in food were unapproachable until we developed the computational power now within our reach. More detailed analysis of diet and how it intersects with individual and population biology, behavior, and the environment. This was the basis for a lot of theory up until now. Now we can begin to address these complex interactions. I was really glad to hear reference to all of us, which I hope will continue to gather steam and get the funding that it needs to answer these critical questions. We need to harness the power of big data and generate the evidence we need so we can take additional actions to help improve the health of our population. I'm wishing you the very best for a successful event, and I look forward to watching the continued joint work on this critical topic from the sidelines as I move back into private life. Thank you.
All right. Good morning, everyone. My name is Ashley Vargas. I'm a Deputy Division Director at NIH's National Institute of Environmental Health Sciences. And it is my pleasure to just take a moment and thank our leadership, Dr. Califf and Dr. Bertinoli, for their wonderful remarks and additional support in this effort. I want to begin with just a brief overview of Session 1. So Session 1 is titled Setting the Stage, FDA Responsibilities, NIH Resources, and Science to Inform Regulatory Policy and Decision Making. So what that means is we're actually going to get a masterclass this morning in how we can closer align the roles of FDA and NIH, so the scientific resources and the ability to improve regulatory decision making around nutrition and food. I'm going to briefly introduce our speakers now, just for the sake of time, as they roll into their presentations. So we're going to begin with Dr. Susan Main of Yale University. She will ground us and share her wisdom as the former director of the Center for Food Safety and Applied Nutrition at FDA. Next, we will move to the wisdom of a pair of speakers, Dr. Helen Meisner, who is the director of NIH's Tobacco Regulatory Science Program that we heard a bit about from our leadership, who will be paired with Dr. Benjamin Appelberg of FDA's Center for Tobacco Products to share how NIH and FDA have really successfully worked together to address tobacco regulatory science needs, and that this may serve as a model for an NIH-FDA partnership in nutrition and foods. Our session will then close with a canonical case study. It will be presented by Dr. Penny Chris Etherton of Penn State University and Dr. Michael Honefberg of FDA's Human Foods Program. They will discuss and share the success story of how academic research and regulatory science aligned in order to remove partially hydrogenated oils from the U.S. food system. We will close with a Q&A, and I want to thank Ms. Samantha Addis of the NIH Office of Nutrition Research, who will be moderating online for those of you online with questions, for her partnership during the Q&A. So right now, please join me in inviting Dr. Main to the podium.
Well, thank you all. It's my real pleasure to be here today. This is really the culmination of a lot of work that's been going on over the last many years between the NIH and the FDA on nutrition. So I'm just thrilled to be here, and I'll echo many of the comments you've already heard in my remarks. I do have, there's my title slide. I do have one disclosure to make on this slide. And next, I was asked to help set the stage in terms of the interagency work on nutrition. And I'm going to focus primarily on NIH and FDA, given that those are the co-sponsors of this workshop. But we do work very closely with many other federal partners, and I highlighted two of them here, CDC, and I see some of our CDC colleagues are here in the room, as well as USDA. And so CDC, NIH, and FDA comprise a huge part of the nutrition portfolio in the Department of Health and Human Services. And at USDA, it's a big place with a lot of different agencies that are working on nutrition. Some I'll specifically mention are the Food Nutrition Service, the Center for Nutrition Policy and Promotion, incredibly important to our federal nutrition work. FSIS, ARS, NEFA are all involved. I also highlighted the dietary guidelines here just to make the point that both HHS and USDA are responsible and jointly issue the dietary guidelines. As I'm sure people in here are aware, the advisory committee recently released their report that will inform the next iteration of the dietary guidelines. And this is just one example where the agencies and across the government departments work very closely together. And you'll hear more about those types of initiatives throughout the workshop here today. The last comment I'll make is if you want to see a more fulsome discussion of all the federal agencies working in nutrition, take a look at the National Strategy on Hunger, Nutrition, and Health that talks about all the different components of the federal government that have been working to address the issues of hunger, nutrition, and health. All right, so starting with FDA, you all have a pretty good sense for what FDA does, but let me make sure everybody has the same foundational understanding. FDA regulates the safety and labeling of 80% of all food consumed in the United States, roughly. Most of the remaining is regulated by USDA, and FDA also regulates infant formula and dietary supplements, as you've heard. You hear talk about food safety. I want to remind you food safety includes microbiological, those are our foodborne outbreaks, chemical food safety, a topic of great interest of late, as well as allergens in foods. And food labeling is really important to make sure that consumers are given the information they need to make informed choices about the food supply. Dr. Califf mentioned First Amendment. When FDA mandates labeling, that is compelled speech, and there are certain guidelines, guardrails around that are posed by the First Amendment. And I won't have time to get into all of that here today, but I'll refer you to an article that Jennifer Pomerantz and others wrote in Food Policy that talks about the First Amendment and how that could inform things like FDA's nutrition front of pack labeling. And other countries don't necessarily have that same constraint with First Amendment. So when you look at what's happening in other countries, you have to also be aware that the First Amendment does have ramifications for steps that FDA may take with regard to mandatory labeling. So what does FDA not do? I think I highlighted what FDA does do, but just to correct a few misconceptions that I see like every day in the media, and I hear from many others, is FDA is not responsible for food taxation. FDA is not responsible for food marketing and advertising. Those are folks like FTC and others. They're not responsible for running the federal food assistance programs. USDA has that job, and so there are representatives here who can address that issue. And FDA is also not responsible for setting pesticide levels in foods. And so when you hear criticisms of FDA, they're allowing pesticides in foods. Actually, EPA sets those tolerance levels, and FDA has the job of making sure that the food supply is compliant and enforcing those EPA tolerance levels. So thinking about setting the framework for nutrition and how FDA and NIH can work together, this is a graphic we created many years ago when I first started at FDA. Robin McKinnon and I developed this to really convey that it all starts on a foundation of robust science. If we don't have the science, then everything else is going to be problematic. And to that robust science, FDA applies whatever tools and authorities they have to try to address the issue. And you can broadly think about the tools as impacting two different pathways. The first one is primarily through labeling and education to increase consumer understanding and ultimately, hopefully, to drive behavior change to lead to improved public health outcomes. The second pathway is really using those regulatory authorities around food ingredients. You've heard about things like PHOs and sodium to really drive a safer and a more nutritious food supply for all of us, even in the absence of behavior change. Again, to drive those public health outcomes. And it's an iterative process. You see the blue cycle going back to inform the science. So evaluating the policies, revisiting policies, what needs to be done to continue to move this pathway along. And the importance here is really NIH is funding, as you heard from Dr. Bertinoli, the vast majority of that robust science that's critically important in nutrition. There are, of course, other folks who fund nutrition, USDA, Robert Wood Johnson, others, industry fund some of this research. But NIH clearly has an enormously important role to play in funding that science that then leads to the translation to try to improve public health outcomes. So you've heard some discussion already about the importance of robust science. You've heard a little bit about trans fat and coronary heart disease risk, and we're going to have a session here on that. Sodium and cardiovascular disease risk, dietary patterns in health, and the global experience in front of pack nutrition labeling. That type of science is absolutely essential to then apply FDA tools, and you'll hear some specific examples of that. But in addition to all of that research that shows us that there are these relationships between nutrition and chronic disease, in addition, regulators really need evidence and science and data to help inform how do we translate all that data. And I'll speak today about some of the issues you think about as a regulator. Things like unintended consequences. If we do this, what might happen? And have we thought that through completely? And what I'll mention here is that local initiatives can really provide much needed data, not just what happens at NIH, but what happens in the real world. Rob would say real world evidence. What happens in the real world when people try to do interventions? What can we learn from that? And so some things I hope you'll hear about today are things like New York City's initiatives to reduce trans fat and sodium reduction. The data that comes from those initiatives can be extremely important to regulators as they want to translate that science to improve public health. You've heard a little bit about regulatory science. You can see the definition here on the slide, so I won't spend time on it, but it may not be the kind of science that normally would make it through NIH peer review for innovation and things like that. I was an NIH-funded investigator for decades, but this is the kind of science we absolutely need as regulators to fill in data gaps. And it's a key part of what we do. Some of this research is funded through extramural activities, and you're going to hear about the Tobacco Centers of Regulatory Science. That is regulatory research that's being done in academic medical centers and places like that to fill the data gaps that the government needs to move policies forward. I was very familiar with the T-Course because that was funded at Yale when I was at Yale, and I worked with the investigators, and I knew the value of having the regulatory science research portfolio. Also, there's the CERCEs, the Centers for Excellence in Regulatory Science and Innovation, and the medical product centers have CERCE centers that they fund to fill in data gaps that can help with medical products issues. But notably, there is no regulatory science initiative that's been funded in food or nutrition. And I always felt that's a disparity, that nutrition doesn't have the same extramurally funded research centers like the other centers at FDA do. And so that's something I'm hoping this meeting can help to address going forward into the future. Intramurally, the FDA does some intramural regulatory science research. The one I'll point out here is the work that was done in CIFSAN, now the Human Foods Program Consumer Studies branch. And these are behavioral scientists, for example, who are studying things like how consumers interface with different test labels, web-based panels collecting data and information about label use in consumers. What's the most way to educate and inform consumers using behavioral sciences research? Focus groups, quantitative studies, that's done intramurally in the Human Foods Program. And the one I'll mention here that's really important right now is the work that FDA has been doing on front of pack nutrition labeling. We know that that will help inform the proposed rule that FDA will be releasing on front of pack nutrition labeling, again, informed by quantitative and qualitative research that's really critical, again, for translating science. Okay, so I would be remiss if I didn't say something about data needed for policy evaluation. If we go back to the conceptual framework that I'm showing right here, across these different measures that you're hoping to implement, consumer understanding, behavior change, composition of the food supply, and public health outcomes, we need surveillance. And there are data needs for all of those different issues. And just to highlight a few of them, and I know you're going to hear more about this in the sessions here today on the data needs. For behavioral surveys, I'll highlight the FDA Food and Nutrition Survey, FSANS, that collects behavioral data. There are many others that you're going to be hearing about as well today or tomorrow. Food composition surveys. In order to know if our policies are working and the food supply is changing to become more healthful, we have data needs for real-time evaluation of what's changing in the food supply. FDA uses a variety of different label databases and things to generate this information, Label Insight, which is now under Nielsen IQ as an example, but also Mintel and Circana, and you're going to hear from other speakers about that. The jewel here is really NHANES, and I know you're going to hear about NHANES as well, where we have population health surveys. And I'll make the point that the biomarker data in particular are extremely important. It can be expensive for the federal government to invest in that, but that gives us more rapid response in terms of understanding what's happening in population health. It takes decades for chronic diseases to develop and to change, so the biomarker data are especially important. And I know from my time at the FDA, funding that is a challenge. And so I will refer you to a very nice article written by Chris Taylor in AJCN last year, talking about the importance of NHANES, all of the components of NHANES, how do we invest it in the future, and how do we modernize it to take even greater advantage of the survey design. So all of these needs are important when we talk about data. All right, now setting that stage, I want to do a little bit of a case study, and I want to talk a little bit about ultra-processed foods. And I know fully well you're going to have a whole session on ultra-processed foods. But my goal is really to talk about it from the perspective of a regulator. Others are going to cover the science and all that, but how do I think about ultra-processed foods as a former regulator? And Dr. Califf highlighted the tension between we want to move fast, we've got chronic disease problems, we're all concerned about this issue, we want to move fast, but there's a tension between moving fast and getting it right and making sure that we have the data underlying any potential action that FDA might be considering. And so the beauty of the regulatory science centers is that they can help fill those data needs more quickly so that you can get that more urgent action. You can take those regulatory actions informed by the best available data using studies that have been specifically designed to fill those data gaps to enable regulatory actions. So thinking about scientific gaps and ultra-processed foods, many people have written articles about the scientific gaps and research, roadmaps moving forward. This is a list that I pulled together. This is Susan Main's list of scientific gaps from the perspective of a regulator. What would I want to know? What I'd really want to know is, Kevin Hall's signature Sentinel studies. What's driving energy overconsumption? We really need to understand that because that has ramifications for the corresponding regulatory actions. Is it energy density? Is it hyperpalatability? And how do we measure that? Is it nutrient combinations? We know that sodium, we know saturated fat, added sugars all play a role, and FDA is taking action on those 3 nutrients, as you've already heard. Is that what's driving it? Is it the fact that ultra-processed foods don't have all the same healthy food groups in them? It's the lack of vegetables and fiber and everything else. Is it non-nutritive ingredients, food ingredients and things like that? The data here are extremely limited, but you're hearing a lot of people crying for public health action, but we don't even know if that's a key piece of it. Degree of processing, I know Dr. Ferruzzi is going to be talking about that. What do we know about processing and how does that influence chronic disease risk? And is it a combination of some components of the piece above there? Is there an adaptive response? The intervention trials are short by design because you can't fund metabolic studies for a long period of time. So we have to invest in longer term studies, cohort studies, epidemiologic studies. How do we measure ultra-processed food in a valid way for those types of epidemiologic real-world evidence? And what about heterogeneity? The topic of ultra-processed foods covers 60–70% of our food supply. Do all of the foods have the same effects from a regulatory perspective? Do we use the same regulatory action for all? Or do we really need to think carefully about heterogeneity when we think about regulatory policy for ultra-processed foods? So I'll refer you to an article that a couple of my colleagues and I published just last month. Dr. Erica Lofeld is here and will be speaking later. Stephen Moore, both from the Cancer Institute and myself. And we were asked to write a perspective piece on ultra-processed foods and cancer. And I'll refer you to it because we talk about some of the research needs, some of the data needs to really address this issue. But a key, I think, lesson with regard to heterogeneity comes from the cancer literature. And more specifically, I'm going to refer to the subgroup findings. And that is, when you look at subgroups of foods in relation to colorectal cancer risk, we consistently find that major sources of fiber and calcium are inversely associated, not positively associated. So our whole grain breads, our whole grain cereals, yogurts, dairy, calcium, vitamin D are inversely associated with colorectal cancer risk, whereas consistently processed meat increases your risk of colon cancer. But they're all in the same category when we think about ultra-processed foods. So are we driving consumers away from the kinds of foods that can actually be protective against risk? So that is something we have to really think about. You can't throw out decades of scientific research that we may not understand some of those heterogeneity effects within subgroups. So that's clearly a research need that I think will come up further in this workshop. So what do we do about, what is the regulatory spectrum? I'm always interested when I hear folks say, the evidence is overwhelming. Ultra-processed foods are increasing our risk for a whole multitude of chronic diseases, so it's time to regulate. We need to regulate, we need to act. And the question is, what does that look like? What does that mean, cries to regulate? And I think it really depends upon the data that come out of the previous slide, the mechanistic, 2 slides back, the mechanistic understanding, so that we don't cause unintended consequences when we target such a broad category of our food supply. People ask me, is FDA going to ban ultra-processed food? Well, that's 60, 70% of our food supply. So obviously that's not going to be on the table in the foreseeable future. But from a range, things like general dietary advice, consumer education. Again, we need to understand some of the data so we don't give consumers inappropriate dietary advice. Labeling, what labeling initiatives could be undertaken. Already you've heard front of pack is underway at FDA. We're all anxiously awaiting the proposed rule on FDA front of pack labeling. FDA Healthy, I think, is going to be coming shortly. So we're anticipating what that can do in terms of labeling. These will take steps towards educating consumers about ultra-processed foods. But what about things like this food is ultra processed, or this food is high energy density, or this food is hyper palatable? Those would be further down the regulatory spectrum, but obviously would have to depend upon data and information. That would be compelled speech that would be subject to high requirements about the robustness of the science underlining that kind of compelled speech. Or what about the ingredients? If it turns out it's ingredients in these ultra-processed foods, FDA can take action on those ingredients, either individual ingredients or even considering classes of ingredients. I will say classes of ingredients is tough from a regulatory perspective because that would make the assumption that all of the chemicals in that class have the same biochemical function. That typically is not the case, but I'll simply mention that as something to think about. All right, so unintended consequences. Could consumers avoid food groups that are thought to reduce chronic disease risk? That certainly is a concern. Could it adversely impact population nutritional status? If consumers think, for example, bread that they buy from their grocery stores and ultra-processed food, it's also a key source of things like folic acid, which are fortified in our food supply. So if consumers make bad choices, will there be unintended negative consequences on our population nutritional status? What about plant-based protein foods, plant-based milks, plant-based meats? We have little data on how they relate to chronic disease risk. And by putting them into the same category of ultra-processed foods, what does that mean in terms of health? And what does that mean for considerations like our planetary health? Infant formula is an ultra-processed food. So if we tell consumers avoid ultra-processed foods, will that then lead consumers to have a lack of confidence in infant formula in the market and then try to make it themselves? And we've seen what happens when consumers try to make their own infant formula. They don't get the nutrients right. There's food safety concerns. That would be a very deleterious consequence. Food inflation, as we constantly hear from the food industry, reformulation costs money. And so if we're constantly asking the food industry to reformulate, that can certainly contribute to food inflation. Could it exacerbate disparities in food access and equity? We know that these ultra-processed foods are more available in places like food deserts, and could that have unintended consequences? And lastly, what about food safety risks? If consumers are avoiding things like foods that have preservatives, could that lead to food safety concerns? There's also issues about food loss and waste. A lot of these preservatives have a big impact on shelf life. And so what does that do in terms of food loss and waste? So these are the kinds of considerations that regulators would think about when we're thinking about the proper regulatory action to take based upon the science and data that we have available. All right, my last slide. I think you've heard that we need a call for data, a lot of data. We need high quality measures in that data as well. And so when we think about something like ultra processed food, that's difficult to measure off of something like a food frequency questionnaire. And so you need robust data in order to inform that regulatory policy. So making those investments, I would hope that we could add good dietary measures into some of these extremely valuable cohort studies, including things like food recalls that can be used to help measure ultra processed food intake or other nutrition exposures for the future. So that's critical. We need real world evidence with valid measures, and it's really that totality of science that we need. So we're going to hear from investigators about the intervention trials, the observational data, what can they tell us? There is no perfect study design. They all have strengths and limitations, but it's that totality of evidence that we're going to be looking for, an investment in that in order to move the needle on going forward into the future. So with that, I will move it on. I think we're going to talk next about tobacco regulatory science with Dr. Meissner.
Thank you. And thank you for the invitation to participate in this meeting. You know, what attracted me to tobacco regulatory science was the idea that, and the opportunity to directly inform national health policy. And that's pretty unique, I think. And I'm delighted that there's interest in forming a similar partnership in nutrition. So let's start at the beginning. In 2009, President Obama signed the Tobacco Control Act, which for the first time gave FDA the authority to regulate tobacco products. Not long after that, and I believe this was mentioned earlier, in 2010, this is a photo of Commissioner Peggy Hamburg, HHS Secretary Kathleen Sebelius, and NIH Director Francis Collins. They established the joint NIH-FDA Leadership Council for Regulatory Science. And one of the working groups under that Leadership Council was for tobacco. Some of the first tobacco regulatory science projects that we funded were through supplements of existing NIH tobacco grants. But as the Center for Tobacco Products at FDA grew, and it became apparent that this was going to be a really large research program, Dr. Collins thought that it could stand to have central coordination within the Office of the NIH Director, since it involved multiple NIH institutes, and he appropriately assigned it to the Office of Disease Prevention. And in 2013, TRISP was officially established. So the Tobacco Regulatory Science Program, or TRISP, is the central hub of the NIH-FDA collaboration, as I mentioned, in the NIH Office of the Director. And we serve as the primary liaison with the FDA team in the Office of Science at FDA. And we work directly with our colleagues at the Institute, so the scientists, budget, grants, review staff, and also policy and finance staff. We advise the Center for Tobacco Products on the best approaches to meet their research objectives. We have adapted policies to meet CTP goals. Our office issues all of the funding opportunities, and the institutes sign on to that, and we coordinate administration of the grant portfolio. So this slide illustrates that there, and this was mentioned, I think, in Susan's talk as well, there are areas of common interest for research at FDA and NIH. So in tobacco, the areas of addiction and toxicity, appeal of products, and so on. But there are areas that are uniquely NIH and uniquely FDA. And this is really an important point to think about. So mechanistic research, for instance, which is the staple of much of what NIH supports, is not something we can generally support through the Tobacco Regulatory Science Program. Regulators need to know a constituent causes harm, but they may not really need to know the biological mechanisms that create that harm. And so that's, a different mindset that's important to consider. Likewise, improvements in treatment and clinical practice, these are not things that we fund in tobacco regulatory science. Likewise, FDA has interests that would probably not normally do very well at NIH. For instance, regulatory impact analysis, very important. Susan mentioned this. I know Ben is going to provide an example of that. This is, probably would not fare well at an NIH study section, or cataloging the toxicological profiles of different constituents. Again, important for regulators, not so much for, innovative research to advance our knowledge. So something to think about. And, I think as researchers, you should be asking yourself, what do they have the authority to regulate? What evidence is needed to support that regulation? And how do the outcomes of my research support the proposed rule or regulation or regulatory decision? And that's a different mindset than what we normally have here at NIH. And while we've funded decades of tobacco research, the tobacco control community didn't necessarily get what regulatory science was. And so we spent a lot of time at the beginning educating the program staff here at NIH, extramural scientists, reviewers, and others on the differences between NIH research and what regulatory science FDA could support. To this day, NIH and FDA staff review the aims of all of the applications that come in for responsiveness to FDA regulatory authorities. If they are not responsive, and we know FDA can't fund it, it's not going to go to review. We've adapted NIH review criteria and other policies, so innovation, not necessarily the most important thing for informing regulation. So we did need to work with the policy folks at NIH to adapt these to really meet regulatory needs. Peer review specifically for regulatory science. Tobacco regulatory science covers a very broad spectrum of research, from health effects and toxicology to marketing, behavior, communications. So you don't have this kind of expertise all in one NIH study section. And after a couple of reviews, we realized pretty quickly that we needed ad hoc special reviews specifically for tobacco regulatory science. And before every review, we do reviewer orientation, because we do not want reviewers to penalize applicants for not being mechanistic, or for proposing something that may sort of seem ho-hum in terms of advancing the science, but not necessarily ho-hum in terms of informing regulation. So I added this slide just really to say that while FDA is funding the grants that we are supporting in tobacco regulatory science, they still are NIH grants. And so our staff are the face of the research to the extramural community. We participate with FDA in developing the funding opportunities, communicate with investigators, and jointly monitor the progress and approve it. So I'll just mention a couple of things. I think the program, we've been at this over a decade. I think the program has been quite successful. And I'm just going to name a couple of things that I think have contributed to the success. Resources, of course, money speaks, right? Support for the grants, but also staff time. And we were lucky at the beginning to have the support of the senior leadership at the participating institutes. It was, it was great to see the director of NIH and the commissioner be supportive of this meeting, but there are other senior level folks at both agencies that whose support is important. We need their support to build new relationships, acknowledge that there is an increased workload related to the program, and establishing a centrally located coordination office to mediate any issues that may arise. Also, staffing, having a core set of staff that really understand what the program objectives are. It helped us a great deal that when they were establishing this program at FDA, that there were former NIHers who were working at FDA to set up the research program. The staff in my office had experience working across institutes. And believe me, if you know one institute, you know one institute. They operate differently, and we needed to come to consensus on how to move forward. And it may sound boring, but we spent a lot of time in the beginning focusing on policy and infrastructure building. And finally, just a no-brainer, an atmosphere of collegiality and respect, understanding the needs of both organizations. We did workshops at FDA on the grants process, convening regular meetings, having open communication. And I see I'm out of time. This is our website. All of the research that we funded can be found here if you're interested in taking a look. And I'll now turn it over to my counterpart at FDA, Ben Appelberg.
Okay, great. Hi, everyone. I am Ben Appelberg. I'm the Deputy Director for Regulatory Science in the Office of Science at FDA's Center for Tobacco Products. And I'm really pleased to be here to talk about the Tobacco Regulatory Science Program from FDA's perspective. So first, just a little bit of background about FDA's tobacco regulatory activities. So the Federal Food, Drug, and Cosmetic Act, as amended by the Tobacco Control Act, gave FDA broad regulatory authority over the manufacturing, marketing, and distribution of tobacco products. And so there are a number of key activities that we undertake laid out here. So, for example, reviewing tobacco product applications to ensure that new products meet public health standards, compliance and enforcement activities to ensure that manufacturers and retailers follow the law. Public education is an important part of our work at the center. So educating the public, in particular youth, about the dangers of tobacco products. And then finally, issuing rules and guidances to implement the tobacco control laws. And I'll talk about some examples of those. And what's important to know, and sort of context for our research program, is that the statute requires the center, FDA, to apply a population health standard when making regulatory decisions related to tobacco products. So this requires us not just to sort of understand the impacts of a product on individual tobacco users, but really understanding the impact to the population as a whole. So both the risks, for example, to young people, to individuals that aren't currently using tobacco products, as well as possible benefits, for example, for smokers who might switch to a potentially less harmful product and benefit their health. And so because of this, when we're sort of talking about tobacco product regulation, whether it's application review, whether it's trying to promulgate tobacco product standards, it really requires quite a multidisciplinary kind of lens, integration of really a wide range of disciplines and lines of scientific evidence. So this includes really understanding features of the products themselves, how they're manufactured, designed, engineered, how they're marketed in a way that could influence the appeal of the product, the level of addictiveness or abuse liability, their toxicity, and ultimately what this all means for impacts to use behaviors and ultimately disease risk. And really because of that sort of multidisciplinary and varied nature of the center's work, we've have a sort of set of research priorities and a research program that's pretty wide-ranging in its nature. So, for example, on the left, this is shows the website that lays out kind of our current research priorities. And we've spent a lot of time both with NIH and others to really communicate to the research community sort of what we mean when we're talking about tobacco regulatory science and the kinds of research questions that are most likely going to help inform regulation and advance the science to drive regulation. So I've just highlighted a few examples of those publications here. Broadly, the research program at CTP really is pretty wide-ranging. So we fund research through a number of different partners. So there's collaboration with federal agencies, for example, on national surveillance systems. So this includes our work with CDC on the National Youth Tobacco Survey, other parts of NIH on adult tobacco use monitoring. We have a large nationally representative cohort study that's funded through partnership with NIDA at NIH. So a lot of that work, we have work that our staff do through contract, partnerships, contracts with research organizations. And then, of course, as I'll talk about now, as we've been talking about, the grants and cooperative agreements funded through our partnership with NIH. And just to give you a sense of the scope of this research program, what I've highlighted here is just kind of a sense of the sort of magnitude, what we're talking about. So from fiscal year 2010 through 2024, we funded over 750 different research projects. And as you can see, a majority, more than half of those projects were funded through this partnership with NIH. This work has really produced a vast amount of scientific evidence, is reflected in a number of publications, about 4,400, of which about 300 were just in the past fiscal year. So one of the cornerstones of the partnership with NIH are these tobacco centers of regulatory science. Susan mentioned this. These are really multidisciplinary centers made up of scientists with a broad range of expertise. Generally, these centers include at least 3 different research projects, each of which has a number of different scientific aims. And then there's an integrative theme that ties them all together. There are cores that are administrative core and maybe other cores to help manage the work. And in addition to providing the science to inform regulatory decision making, they're quite adaptable in terms of working on emerging and developing needs and issues. And they also have included a program for career development for younger researchers to get into the field. And I'll just note that the first set of T-Cores that were funded in 2013 were center grants, and then subsequently for the second and third rounds, we've worked with NIH to make them cooperative agreements, and that really allows us through the NIH program officers to be a little bit more engaged, to have some more collaboration and exchange with the scientists that are leading this work. So just to give you an example of one of these centers, highlighted here, the Center for the Assessment of Tobacco Regulation, also known as CASTOR. The goal of this center is laid out here: to provide evidence-based and expert-informed modeling of the behavioral and public health impacts of FDA tobacco rules or other regulatory actions, focusing on impact analysis, behavior, and health effects of scientific domains. And you can see the centers that are involved. And just to give you a sense of the work that's come out of this center, one of the papers was this update of a model that looked at the public health impact of a ban on menthol cigarettes in terms of tobacco use and future morbidity and mortality. So in April of 2022, FDA issued proposed rules, one of which was to prohibit menthol as a characterizing flavor in cigarettes. So this is, particularly informative to that kind of rule, and was cited in the proposed rule. Another proposed rule that issued at the same time was a product standard to prohibit all characterizing flavors other than tobacco in cigars. And so when we think about the impact of our research program, we're regularly evaluating that, including the extent to which it informs regulatory decision making. So one way we've kind of looked at that is looking at those two proposed rules in the preambles, where the scientific rationale is laid out. Really, the scientific justification argument for those. We identified that about a quarter of the publications that were cited in those rules were the result of CTP-funded research. And a significant fraction of those came out of the TCORS grants. Really quickly, just the last few slides just highlight some of the other types of scientific research questions that we fund that we feel are sort of the most critical for addressing tobacco regulatory needs. So this is a recent notice of funding opportunity for some really discrete, specific questions to help inform application review and product regulation more generally. So for example, nicotine pouches is a product that's become sort of an emerging product. So understanding the extent to which data that's collected on abuse liability might inform the likelihood that smokers or other smokeless tobacco users might be able to successfully switch to those products. Understanding the characteristics that increase or decrease the appeal to young people, the health effects of e-cigarettes is a critically important topic, as well as questions related to specific ingredients and constituents in the products and what those the influence of those are. This is really important kind of evidence for us to make determinations about those kinds of applications. The last two slides, just to note some other examples. We recently issued an award to look at public education related to the continuum of risk. So this research is really going to look at how FDA can accurately convey this kind of concept of the continuum of risk without having negative consequences on young people. So that kind of formative scientific research is really critical to our public messaging. And finally, I'll just emphasize what Susan talked about when she talked about surveillance and evaluation. We have these partnerships with federal agencies to conduct national surveillance on what's happening in terms of tobacco product use and its predictors. But that can take time and be kind of slow to generate the evidence and report on it. So we've recently funded a Center for Rapid Surveillance of Tobacco that really allows for the integration of multiple lines of data, including sort of non-traditional sources, to be able to identify emerging issues that can be brought to the center's attention so that we can figure out what to do next, gather more data or take regulatory action. So those are just some of the examples. I know I kind of sped through that because we didn't have a ton of time, but thanks again for having me here, and I'm happy to answer questions in the Q&A.
Okay, thanks so much for the opportunity to be here today. and to talk about the robust evidence that led to the trans fat ban. Just a brief comment, the food sources of trans fat that I'm going to be talking about and the studies presented come from partially hydrogenated fats, oils, and margarines. So today I'm going to give a very brief background of early trans fat research that led to the randomized controlled clinical trials, observational studies, and mechanistic data that led to the trans fat ban. Then we'll talk a little bit about current dietary recommendations for trans fat. And one more brief comment, in the box on the bottom, you see that trans fat is the isomer on the right, and it is an unsaturated fat. It's distinctly different from the cis isomer in that it has hydrogen atoms on opposite sides of the double bond compared to the cis isomer. Okay, so interestingly, the early research on trans fats goes back almost 75 years. In the 1980s, there was research conducted that showed that trans fat concentrations in tissues from patients who died from coronary disease were higher than those who died from other causes. And then there were 3 classic clinical studies done in the 1950s to 1961 that showed that trans fat increased total cholesterol levels. But it wasn't until the 1990s that the major clinical study done by Katan and Menzik and the observational evidence done by the Harvard group opened the floodgate to all the research that followed that led to the trans fat ban. And so this is a very seminal study published in the New England Journal of Medicine in 1990 on a total N of 10 participants that showed on the left-hand side that compared to saturated fat and oleic acid, trans fat lowered HDL cholesterol, and on the right-hand side, the trans fat diet and saturated fat diets both increased LDL cholesterol compared to the oleic acid diet. So a lot of studies were conducted afterwards, and in 2003, Menzik and colleagues conducted a meta-analysis of 60 controlled trials of dietary fats and looked at the effect of replacing dietary carbohydrate with different fatty acids. On the left-hand side, the gray bar shows an increase in the total cholesterol to HDL cholesterol ratio with trans fat compared to saturated fat, which is a black bar. And you see the unsaturated fat bars show a decrease in this ratio. And the reason why this ratio increased is because on the right-hand side, we see that trans fat and saturated fat both increase LDL cholesterol. But on the right-hand side, we see that trans fat, which is the bar to the farthest right, it basically lowers HDL cholesterol compared to the other 3 fatty acids. So we're getting evidence that trans fat then is markedly hypercholesterolemic and basically lowers HDL cholesterol compared to other fatty acids. So what I just talked about was exchanging trans fat for dietary carbohydrate. What happens when unsaturated or saturated fatty acids are isocalorically substituted from trans fat? On the left-hand side, we see increases in LDL cholesterol, decreases in HDL cholesterol. And likewise, when saturated fats are replaced with trans, LDL increases and HDL decreases. So now turning to the observational evidence, this study was published in Lancet in 1993 from Nurses Health Study, and you see a clear dose-response relationship between trans fat intake and risk of coronary heart disease. And in this study, the authors suggest that based also on the lipid lipoprotein evidence that because of the current trans fat intake then, that would account for about 30,000 deaths per year due to trans fat intake from heart disease. And so that looking at a substitution analysis, this comes from Nurses Health as well, what we see is that when trans fat replaces saturated fat at 2% energy, there's an increase in coronary heart disease risk by 5%. And you can see that, monos and polys substituted for saturated fat decrease risk considerably when 5% are replaced, as is the case with dietary carbohydrates from whole grains. So you can see that trans fat is even worse than refined carbohydrates, shown in yellow here, when it replaces saturated fat. So this slide here on the left-hand side shows changes in total mortality associated with increases in different fatty acids. And the blue bar shows total mortality, trans fats. And you can see that trans fats are the worst fat of all compared to saturated fat in red. And then the good fats are monounsaturates in the green and the blue. And then on the right-hand slide, on the far right panel, we see that trans fats increase cardiovascular disease mortality and also increase neurodegenerative disease mortality. So there are proposed mechanisms back in 2006 for why trans fats are eliciting adverse health consequences. And they could work at the hepatocyte level, that is, increase atherogenic lipoprotein synthesis. B, work at the endothelial cell levels, that is, cause endothelial cell dysfunction. C, at the adipocyte level, increase free fatty acid levels. And D, have an adverse effect on monocytes and macrophages, i.e. increase inflammatory responses. And this was proposed in 2006. And I'm going to just show you the evidence that has come out that has supported, all of these mechanisms. So at the hepatocyte level, we showed that trans fats increase LDL cholesterol, decrease HDL cholesterol. The far left panel here, you see a dose-response relationship with trans fat intake and triglyceride levels. In the middle panel, you see a distribution of lipoprotein A levels related to trans fat intake and saturated fat intake. And trans fat are in the white bars, and saturated fats are in the stippled bars. And you can see that there's a clear relationship here, and trans fats are worse than saturated fat in terms of lipoprotein little a. And on the far right, a very nice study done by Alice Lichtenstein, which showed that LDL particle size is decreased on a trans fat diet. And we know that small, dense LDL are very, very atherogenic. So in terms of endothelial cell dysfunction, very nice study showing that when trans fats replace saturated fat, the flow media dilation of the brachial artery is decreased, indicating artery hardening and stiffening related to trans fat intake. Now, there's a lot on this slide here. So basically, this comes from Alice Lichtenstein's work. I've circled SM, that's stick margarine, that's the highest trans fat intake. And what it's showing in the black bar is an increase in free fatty acids that likely is related to a decrease in ASP. And ASP is the determinant of the rate of adipocyte triglyceride uptake. So adipocyte triglyceride uptake is decreased. There's an increase in free fatty acids in the plasma. And what we know by the third bullet point on the right is that an increase in plasma free fatty acids increase the risk of metabolic diseases. Okay, what about monocytes and macrophages? There's evidence to show dose-response relationships between trans fatty acid intake and increases in ICAM, VCAM, and E-selectin. And then at the bottom box, a very important clinical study done at USDA Beltsville basically showed that trans fats diet increased CRP and E-selectin. 1 minute. Okay, so we're on dietary guidance now. And so actually in the 2000 guidelines, there's a sort of suggestion to decrease trans fat intake. And, the recommendation is to choose vegetable oils rather than solid fats. But it wasn't until 2005 that there was clear guidance to keep trans fat intake as low as possible. And finally, from the ACC/AHA guidelines, what we see is that there's one nutrient that causes harm, and that's trans fat. And trans fats should be avoided based on a lot of evidence, as you can see in the box on the right. So with that, I thank you very much for your attention, and just want to say that because trans fat has been taken out of the food supply, you can see that it's been estimated that it might avert 72,000 to 228,000 coronary heart disease events per year. Thanks.
Okay. Hi, everyone. So my name is Michael Honigford, and I am from the food ingredients part of FDA. So I'll be coming at this from that perspective. So thank you for having me today. And thank you to Penny for the great overview. That great discussion of the robust science we have on the effects of trans fat really lead into the regulatory discussion that I'm going to have and explain how FDA used that in order to take action on trans fats. So very, very briefly, I need to first start with FDA's authorities to ensure a safe food supply. So how do we regulate food ingredients? Well, we were given some laws by Congress. And the key one I'm going to be talking about today is the 1958 Food Additives Amendments to the Food, Drug, and Cosmetic Act. These amendments gave us a definition for food additives. It gave us the authority to ensure that food ingredients added to food were safe, and established a pre-market review for these ingredients. So from this law, food ingredients can either be a food additive or they can be generally recognized as safe. But in either case, they have to meet our safety standard of reasonable certainty of no harm. So with the Act, food additives, we were given a definition. Essentially, anything that can become a component of food that is intentionally added falls into this bucket of food additives. And we have a pre-market review process for them, a petition process where industry can come and ask us to grant an approval. But also within this definition is a provision called the GRAS provision, or generally recognized as safe. So this definition carves out kind of an exception if ingredients were generally recognized by qualified experts to be safe, or if they were in use prior to 1958 when that amendment was passed. They are exempt from pre-market approval by FDA. So today, for GRAS ingredients, FDA operates a GRAS notification program. So industry can come to us if they want to use an ingredient, inform us of their conclusion, and we can respond to them. So with that high-level background on how food ingredients are regulated, I'd like to get into how partially hydrogenated oils fit into this regulatory scheme. So partially hydrogenated oils have a very long history of use. Long before 1958, they were in the food supply. The most commonly used partially hydrogenated oils were partially hydrogenated cottonseed oil, partially hydrogenated soybean oil, and you wouldn't have seen those in FDA's regulations because of their history of use, much like other common historically used ingredients. FDA did affirm a few partially hydrogenated oils as GRAS in the late 1980s, essentially based on their similarity to these commonly used historical ingredients. So as I described, FDA has an established authority for food safety, and we also have a framework to ensure the safety of food ingredients. And we have the authority to take action when warranted. So as I'll demonstrate in this talk, this concept of GRAS, or general recognition of safety, is time dependent. And if we determine that the science no longer supports the GRAS status of an ingredient, we can take action. And for example, as I mentioned, we have a notification program for GRAS ingredients today that does not involve rulemaking like it does with food additives. So if we do identify that something's no longer GRAS, we could more efficiently take action. So I'm going to walk through the timeline that I just briefly showed on the last slide and kind of walk through what FDA did through the years in response to the evidence that came out on trans fats. So our earliest action was our actions on trans fat labeling, and this was very effective. So Penny talked about the science in the 1990s, and that led to FDA proposing a rule in 1999 to list trans fat on the ingredient label and on the nutrition facts panel so that consumers could monitor the amount of trans fat they were consuming. And this was based on the evidence that Penny described, the relationship between trans fat and its effects on LDL. And this proposal was finalized in 2003 and became effective in 2006. Now, we saw a lot of reformulation in the early 2000s. As soon as we announced that proposal, industry started responding. They didn't want to list trans fat on their ingredient or on their nutrition facts panel. So, as I said, this was very effective. We saw intake of trans fat be reduced about 80% as a result of the labeling actions. So kind of summarizing the key information that existed on trans fat as we got now into the early 2000s. There were the feeding trials that showed the linear relationship between trans fat and cholesterol levels. There were the observational studies, and as well as the expert panel reports that were coming out during the 2000s. So all of this data led FDA to consider using our regulatory tools to start to look at focus on ingredient safety as a way to further reduce trans fat consumption, and taking a look at partially hydrogenated oils as that primary source of the artificial trans fat in the diet. And like Penny already discussed the scientific evidence in detail, so I have this as a summary really to demonstrate the robust amount of information we had in order to take regulatory action. We had 23 controlled feeding trials in the literature. Those were supported then by the prospective observational studies. And then we see consistent evidence across both those types of studies, which show, is a very strong evidence. And then again, we have all these expert panels coming out suggesting that even the current intakes of trans fat were still too high, even after the labeling had taken effect. And as I mentioned, one of the key components of food ingredient safety, if you are going to be generally recognized as safe ingredient, is this general consensus among qualified experts. And it was clear that this was no longer the case, that there was no longer a consensus that partially hydrogenated oils could be considered safe. So in 2013, FDA published our tentative determination regarding partially hydrogenated oils. We said that PHOs, we believed that they could no longer be considered GRAS based on the health risks. We explained in detail our supporting evidence, the scientific studies, and the panel opinions. We have a regulatory process set out in our regulations that allows us to make a determination if we find there's new data available concerning a food ingredient. This process does include a public comment period, so we did receive a number of comments. Some of them were very technical, and we reviewed all of the comments and responded to them. And we concluded by stating that we believe that elimination of PHOs could prevent thousands of coronary events and deaths. So moving forward, after we evaluated all of the comments, we published our final determination in 2015. And we said that PHOs could no longer be considered GRAS for any use in human foods. And as I mentioned, that regulatory distinction, therefore that makes them food additives. There were no authorizations for them as food additives, therefore they would not be allowed to be added to food. We set a compliance period of 3 years, so industry had time to phase out their use of PHOs. So that was to go to 2018. So continuing a few years later, one of the further actions we took was the denial of a food additive petition. Industry had asked us to consider approving some low-level uses of PHOs. However, when we reviewed that petition, we could not find that we could state that any level of PHOs were safe, and therefore we denied that petition. And at that same time, we extended the compliance date to ultimately 2021 is when all PHOs needed to be removed from the food supply. And just last year, we did what we would call some final housekeeping actions in our regulations just to clarify any, basically any mentions of PHOs that still existed in our regulations. There were a few standards of identity regulations that included PHOs as optional ingredients. We removed those. We revised those GRAS regulations that I mentioned earlier from the 1980s and took out those uses of PHOs. There were also some pre-1958 authorizations that we needed to clarify. And so we did all of that in this 2023 final action. So in conclusion, I hope I was able to explain how FDA used the robust body of literature in order to take our regulatory action and kind of show how we do have a regulatory framework where we can address ingredient safety through our framework and our authorities. So thank you.
Okay, let's take one more second to thank our speakers for truly giving us a masterclass this morning. Okay, and I am going to take the sitting down just so that I can see the screen, but I wanted to switch the slides, so I'll be right back. Okay, so now we're moving into our Q&A for all of our speakers. So speakers, when you want to respond, just click on the speak button on the microphone. So we have questions hopefully coming in online. For those of you that are online on Zoom, please enter questions through the Q&A. For those of you in the room, please line up in behind the microphones here. We have 2 in the room to start the questions. And as moderator, I will start the first one. So I hope many of our viewers and many of those in the room really got a sense of the story that could be had when NIH and FDA work together. And so I wondered, for those of you in the room, you all have had various roles. Are there any things that you want to say as far as advice goes to researchers who really want to see their publications and their research contribute to public health and regulatory decisions that they may not already think of? So I'll pause and ask if there's any advice people want to give to researchers. Dr. Meisner?
One thing that we say over and over again is to think specifically, what regulation am I trying to provide the evidence for, and how can it, how can my outcomes be incorporated into that proposed rule? All of the evidence that was provided here, how was it used? It's a good idea to take a look at some of the proposed rules and regulations, or even ones that have been finalized, and see the type of information that is used to support the rule.
I think the other thing I'll add is it's not just like the science per se, but it's also the econ and the public health impact. And it's really publications from multiple disciplines that are so critical in the rulemaking. And like when I joined FDA, I didn't know what a regulatory impact analysis was. So it's kind of becoming educated on the types of data that FDA must use in taking these public health actions and getting publications from that broad spectrum to help move regulatory policy forward.
And just real quickly, I think it goes without saying, do the highest quality research you could possibly do, and try to get your study cited by the Dietary Guidelines Advisory Committee, and they have specific criteria that they're looking for before they will use your study. So pay attention to that.
Thank you. And now we'll go to our first question in the room. Please just state your name and where you're from before your question. Thank you.
Sure. Shu Wen, University of North Carolina in Chapel Hill. So it was great to get the example of the partially hydrogenated oils. And if we look at the timeline in terms of how long it took from the initiation of some research on that issue all the way to when everything got tidied up, the FDA side, that was 30 years. Is there a way, like in your view, and based on past experiences in working in this space, how can this process be sped up if there were additional ingredients or classes of ingredients that required more regulation, in your perspective? Thanks.
I can start. I think, there's multiple ways you can speed it up. One is really for an administration to make it a priority. If an administration says this is a priority, and, administrations come in and they got 4 years, and don't guarantee they're going to get reelected. They got 4 years to get something done. They can really drive a lot of what federal agencies prioritize and move along more quickly. So I've seen rules that have taken, so long to clear through the federal process, and I've seen rules that can go pretty quick. And so a lot of what the administration prioritizes can impact that. The other thing is Congress can impact that too. And that's because federal agencies can say, we really want to do this, but here's a problem we are facing and we don't have clear authority or we don't have this and we want to be more nimble, but you need to help us do that. And so working with members of Congress can really affect how nimble federal agencies can be in terms of taking some of these actions, because these are spelled out in law and there's procedures and things that they have to follow. Congress can modify those. So I think administration, Congress, others can really, really help. And then research funding, the appropriators, the people who are funding NIH research and people who could fund regulatory science. So it's the legislators, the appropriators, and the administration can all impact those timelines in a really important way.
Hi, Kevin Hall, NIH. Maybe it's a narrower set of questions, basically also on the timeline issue. When, and this for Dr. Chris Etherton and Hollingford, when do you think that the threshold was crossed between when we had sufficient scientific evidence to act? And what did that body of knowledge look like as we kind of look forward to other aspects of our food system?
I think the other thing I'll add is oftentimes when the agency is concerned, it's not just one policy they're trying to implement, it's multiple policies that interact together to get to a desired outcome. And so you heard the mention of the labeling, you heard the mention of dietary guidelines, taking food ingredient authorities, something like sodium reduction that we worked hard on in my time at the FDA, multiple policies. It's been on the label, that's not enough, changing how industry can declare it on as a food ingredient, then working towards the voluntary targets. There's multiple policies going on at the same time to try to drive that public health outcome. You may hear about one particular policy, but there's often many things happening at the same time to try to drive those public health changes. And I think you're seeing that with ultra processed food. There were agencies are moving forward right now with some of those things, but there's more that we can and should be doing, but we need data and science to do that.
And we're getting a lot of questions in the chat, so thank you for entering those. These questions are around if the FDA still regularly monitors the PHO regulation or is it assumed it's not needed? And also asking if what action the FDA is taking on products that still contain those trans fats.
I will say we do monitor. We do label reviews of imports. We do monitor ingredient labels. We have inspectors go out. They're aware that PHO should not be used in food anymore. And we do have a number of compliance tools that we can use to take action if we see it on a label or see it during an inspection or something. So yes, we are definitely looking at that.
And then this question is for Dr. Main. Can you speak a little bit more about the First Amendment implications for food labeling?
Yeah, I think one of the really good examples comes from the tobacco labeling experience that we've had. And perhaps you could speak to what's happened with tobacco, where again, compelling speech is requiring industry to put something on their packaging and that has to be able to survive First Amendment challenges. And I think one of our best examples is the tobacco graphic warning label. So do you want to speak to that?
Sure. Yeah, I'll just add to that. I mean, we run into this often at the Center for Tobacco Products in terms of what Susan's talking about, in terms of the sort of heightened scrutiny that, whenever you're sort of dealing with compelled speech, whether it's warning labels. We also have, authority to review claims that, companies want to make for their products, and it sort of has that same implication. So that it's been litigated quite extensively, the health warning work over a period of basically much of the history of the center at this point, but we're things are heading in a better direction at this point. But yeah, that is just a factor that we have to take into account, and then relates to kind of the consideration of the like folks were talking about, the types of regulatory tools, the types of tools in the toolbox, and really trying to approach things from a variety of in a variety of different ways.
And I cited Jen Pomerantz's paper that was published as well that kind of goes through the legal risk depends upon the type of label, and they have varying degrees of risk, and that's covered nicely in Jen Pomerantz's paper that I cited.
Thank you. We'll turn to our next question in the room.
Yeah, I'm Rafael Perez Escamilla from the Yale School of Public Health. And this question is related to the last comments. In your experience, what has been the role of the legal system in either making it easier or more difficult for the FDA to do its job? And what can be done if the latter is true to change that situation?
I can start from our perspective at CTP. I mean, we're in a situation where we're regulating a pretty litigious industry, so there's legal concerns are sort of always at the forefront. I mean, obviously we're gathering science to make the strongest science-based, evidence-based decisions, and that's what's going to hold up to the strongest scrutiny, is to be able to demonstrate that we're following the science, that we're following the statutory requirements that are laid out by Congress. Yeah, I mean, the legal, political, I mean, there's all kinds of considerations that are sort of always, I think, at play in the regulatory decision making.
I guess the only thing I'll add is if the agencies have clear authority or a mandate from Congress to do something, that's going to be a very different situation than if an agency is trying to say, this is kind of tenuous and, we're not, it's going to be a bigger stretch, right? So again, working with Congress to have explicit authority to take public health actions is going to make that legal hurdle a whole lot easier to overcome. So I think that's really important.
You all described the process by which evidence arose of the adverse health consequences of consumption of PHOs and how that worked through the regulatory process. I would be curious to hear a little bit about, presumably, these fats were being used for some purpose that had a benefit either to industry or consumers in terms of taste, texture, shelf life, whatever. During this process, talk a little bit about how the benefit side of that equation came into the conversation, if at all.
So for food ingredients, there's not really a risk benefit like there are with drugs. And so, food ingredients need to be safe. They need to meet the reasonable certainty of no harm standard in order to be added to food. So there's not really that consideration. I think what we did consider was, in allowing a compliance period for industry to reformulate and to find alternatives, because we did recognize that there were very real reasons they used these for shelf stability and for mouthfeel and texture, and they did remove a lot of them. And my understanding is that last 20% was the most difficult, and that's why they weren't removed yet. So there was some R&D, and my understanding, work that needed to be done to remove those. But yeah, I mean, I think allowing that phase out period sort of allowed that to happen.
And
Yeah,
the
there's
other
no-
thing I'll add to what Michael was going to say is, having a dialogue with industry is part of it. I mean, FDA hears from all of their stakeholders, their public health stakeholders, and industry, all of the above. And that's really important because of issues of unintended consequences, things like that. So in the case of the partially hydrogenated oils, there were questions about supply chains. So if we take all these out, is there enough suitable substitute for the food industry to continue to make their food without messing up all the supply chains? And so that can impact things, not like the regulatory decision, but the compliance periods that are needed to accommodate a regulatory action. So stakeholder engagement is key. It's notice and comment. So there was lots of public comment that the agency hears to really address these issues of unintended consequences. And I recall the supply chain dialogues that we had at that time.
Right, and I think that came into play. You probably saw me mention we extended that compliance period, and that was in dialogue with industry. They had petitioned us for some uses, and we're waiting on our decision on that petition. And so, there were still some uses that they needed to phase out. So that was part of that dialogue.
Thank you. We'll have our last question from in the room. Please identify yourself, lady.
Thank you. Shariki Kumanyika, University of Pennsylvania. There are some differences in what's allowed in Europe and the US. And I'm just wondering if maybe Susan, you could talk about this. What's the difference in the data sources that are considered by EFSA versus, FDA? So.
I can, and I think Michael can deal with that one as well on the food ingredient side. A couple things. I mean, the regulators may arrive at different conclusions having looked at the same information, and we've seen that play out with different food ingredients. But having said that, I think they all agree on the importance of toxicity testing and data and information, but sometimes people will interpret it slightly differently. And so if under US regulations, we look at underintended conditions of use. And so dose is a hugely important aspect. And so simply showing that something might have an adverse effect, if the safety margin is so large that we're not concerned that that's going to be an issue, then that's how the US may look at that. But other countries may look at that a little bit differently. But dose and intended conditions of use are critically important to how FDA looks at food ingredients. And so you hear people say, well, this thing causes cancer. Yeah, at doses that could never be achieved in human consumption. And so those are the kinds of issues that FDA would think about. But Michael, you can augment that.
No, that essentially sums it up. I was going to say, sometimes people point this out, not realizing that, industry comes to us for approvals, and sometimes it might just be that they asked for approval in the US or didn't ask for approval in the US and asked for approval in Europe. So sometimes those differences aren't even based on safety. They could just be based on, the regulatory, approval process and, the different regulatory frameworks.
All right, please join me in thanking our speakers and panelists one more time. So we will reconvene after lunch at 12:45 to continue our discussion on, as Dr. McKinnon said this morning, charting a new path forward for nutrition and regulatory science. Thank you.
Now we're recording. So welcome back, everyone, from lunch. Hopefully you feel very well nourished. Really excited for this afternoon session. But this morning session was really a great, as my colleague Ashley Vargas said, a great masterclass in nutrition regulatory sciences, and really a great opportunity for two case studies around tobacco regulation as well as partially hydrogenated oils to really set the stage for our two conversations today. So my name is Allison Brown. I'm a program director here at NIH's National Heart, Lung, and Blood Institute, and really excited to welcome you to this session, but also serve as the virtual moderator. So the afternoon session is titled Key Research Gaps and Opportunities in Nutrition Science to Inform Regulatory Policies and Decision-Making. So we have actually two presentations and panels that encompass today's session. So the first one is on ultra-processed foods, and the second one is on methodologies for incorporating nutrition science into risk analysis and food policy. But I'm really excited to introduce the speakers for our first set of presentations on ultra-processed foods. And it goes without saying that ultra-processed foods has really been widely discussed in the news as well as the research community over the past several years. So we're really excited to have four experts to talk about the topic, but really focus on what we know, but key is what we don't know and the opportunities for research in the future in this area. So first we'll have Lauren O'Connor, who's at Texas A&M Agriculture, Food, and Nutrition Evidence Center, followed by Mario Ferruzzi of Virginia Tech. Then we'll have Maya Bendevelu based out of University of Rhode Island. And last but certainly not least, we have NIH's very own intramural researcher, Kevin Hall of the National Institute of Diabetes and Digestive and Kidney Diseases. So similar to the previous session, we welcome a Q&A from the virtual attendees, and I'll be serving as the virtual moderator. And after each of the 15-minute presentations, we will take questions from the room, from the audience members here physically. So just really excited to welcome our first speaker, Lauren O'Connor.
All right, thank you so much for having me. And Susan did a fantastic introduction to this topic this morning, which I think you'll see a lot of overlap in concerns and research gaps. But clearly there is an association between ultra-processed foods and health outcomes. We can't deny that. And because this is a regulatory focused meeting, and what I've been seeing today is that in regulation, you're really in the weeds, and that's where I love to be. So that's where I'm going to get into today. And just just dive into, do we really know as much as we think we do about what an ultra-processed food really is? So if you've been living under a rock, we classify ultra-processed foods using NOVA. It groups foods into 4 different groups based on a proposed level of increasing processing along this continuum. The culprit of interest is always group 4. The other ones kind of just go by the wayside most often. And an ultra-processed food is defined by a formulation of several ingredients and food substances that have no or rare culinary use, as well as there's a lot of focus on the food additives that have a cosmetic function that improve sensory properties of the food. But in order to really interpret this definition of ultra-processed foods, I want to take a step back to discuss the difference between food processing and food formulation. So processing is really the methods and the techniques that are going into making a food, including processed foods. Whereas formulation is, it's essentially the recipe, it's the ingredients. There's a very common misconception that I think is causing a lot of confusion and headbutting on this topic, is that people think food processing and food formulation are interchangeable terms, but they're not. And I do think this is a pretty difficult concept to understand if you're not a food scientist, and I'm not a food scientist. Mario is, he can correct me if I'm wrong on any of this. But so in order for me to really process what the difference between these terms, I'm gonna give you a kind of a silly example. So if you take a sugar cookie recipe and you want to reduce the sugar, so you're going to replace some of the sugar with Splenda, you're changing the formulation, but you're keeping the processing the same. You're still mixing it and baking it at whatever temperature. But you can keep the exact same formulation and also process foods differently. So you can make a delicious sugar cookie by baking it, or you can make something really weird by fermenting and smoking it. So silly example, but what I'm trying to show is that formulation can differ, processing can differ, and they're not interchangeable processes, but they're both processes that are essential to making a food. So if we get back to the NOVA definition and just look a little bit more critically at this, the definition of UPF is literally defined by the term formulation. It is a combination of ingredients and food substances and food additives. So my perspective today, which I think is beginning to be a trend that I think many of the panelists would agree with, is that the designation of being an ultra-processed food really isn't so much about the processing, it's really more about the formulation. So let's test my hypothesis. So does processing actually increase along this continuum? Stole this from Mario. So this is a diagram that he put together about the steps that go into making a nut milk, so like almond milk. And any food processing classification system, any clinician, any researcher would probably say that, yes, this is a highly processed food. I think we can all agree on that. But if you look at cow's milk in NOVA is considered a minimally processed food. Most clinicians, researchers, et cetera, I think would agree that we usually call this a minimally processed food. But if you actually look at the steps of processing, there's actually more steps that go into making cow's milk than there is to making nut milk. And this is not to shame either food. Both are important components of a healthy dietary pattern. But the point is that why do these two foods sit on the opposite ends of this spectrum where processing is supposed to be increasing along this spectrum from group 1 to group 4? Now, this may be the exception and not the rule, but the point is that we don't know. We need research to really understand whether foods in group 4 truly undergo more processing than groups in 1, 2, or 3. Because if we're saying that processing is the issue, if that is what we're trying to regulate, then we need to make sure that our verbiage and our language is matching what the system is actually picking up on. So this is the full and operational definition of ultra-processed foods. Do not read this. I'm going to walk you through it. There's a lot of critique that there's not a definition of ultra-processed foods. There is. There's a lot of thought and that went into this. It's a very, well thought out process, but it's challenging to operationalize. So let's pretend that we all ran a trial and we have a large data set of dietary intake and we want to calculate how much calories someone's consuming from ultra-processed foods at baseline. So the first step is to identify whether the food that this person ate on this day is an industrially manufactured food product made of several ingredients. So again, there's that formulation piece front and center, or whether it's homemade. This is pretty challenging because homemade means different things to different people, and our dietary assessment tools don't ask this question generally for that reason, is because it's, you know, opening a jar of tomato sauce at home versus making it from scratch. People might consider those both being made at home. So let's just say that the food that of interest is, yes, it's an industrially manufactured product made of several ingredients. And let's just use tomato sauce as an example. So once you have the food, you have to then look at the list of ingredients to see if there's a hallmark or characteristic ingredient that would make it an ultra-processed food. So again, there's this ingredients, this formulation focus, and there's some examples here that I pulled out from the definition in the blue boxes of what those characteristic ingredients might be. But again, this is easier said than done. It relies a lot on the dietary assessment tool being used. And I think Dr. Main mentioned this as well. Using a food frequency questionnaire, you don't have itemized foods that the person's consuming. You have large food groups. Even if you do have a 24-hour recall or 24-hour record, you have to rely on the underlying database that is connecting to those tools and providing that nutritional compositional information. I can tell you there's people in this room like Kyle who know a lot more about this than I do, but we do not have this type of information in our dietary databases. There are some data on artificial sweeteners in some dietary databases, but what you need for accurate NOVA classification is not available in dietary databases. What you really need is a food label. And we all know that this is a rare thing to have when you're running large prospective cohorts or large trials. It's very difficult to get the food label. And if you have the food label and you can identify the ingredient or the additive, you then also have to know the function of the additive and the ingredient. This is a list of how additives are classified in NOVA, but just to focus on group 4, you have to identify whether that additive has a cosmetic function. So is it making the food look or taste better? Again, I'm not a food scientist, and I think this would be a pretty challenging thing to figure out. And I work for Texas A&M, but I work remotely. I live in this area, and the Library of Congress is probably my favorite building in the whole city. And I was there this past summer, looking through the food additives data book, because that's what I do in my free time. And I was looking at some additives to try to figure out, like, okay, could I, with my advanced knowledge on this topic, could I figure out whether this additive would be being used for cosmetic purpose or not? And you can see here, this is just one example of propionic acid, which it has antimicrobial purposes, which would not make it ultra processed, but then it's also a flavoring agent, which would make a food ultra processed. Just a second example here, sodium nitrate, same thing. It contains, these additives serve multiple purposes. And if you're not the one creating the recipe for these foods, how do you know what that purpose is? And how do you make that designation of ultra processed versus just group 3? So with that said, if you have all of the brand names, the food labels, you know what the additive is used for, you have all of this information, there's still a lot of variety even just within a food. So again, not trying to shame foods here, but if you were to classify each one of these 4 foods, each one of these 4 pasta sauces, they could fall into either group 1, group 3, or group 4. But my question is how meaningful is that? Are these foods really that different where they would affect health in a differential way that we should be aware of? But the biggest point is that we don't often have brand names and food labels unless you're running a new study or we develop new tools, which I would argue is a huge area for new research. But without this information, these four pasta sauces, which technically fall into 3 different categories, in a dietary data set would actually probably all go into one category. So you're losing that nuance because you don't have the information that you actually need. But there is some guidance available for researchers. The NOVA group put out a really great commentary in Nature Food about best practices of applying NOVA. This is led by Eurydice Martinez-Steele and Neha Kampor. And it gets into the sensitivity analyses and the checks and balances that you can do with your dietary data set to kind of overcome some of these assumptions and lack of data. So this is a really great resource if you're trying to do this with your own study. Another really great resource, also led by Eurydice Martinez Steele, as well as Kirsten Herrick, who's here today. And when I was at NCI, this is a static data set that links to ASA 24 and NHANES. And it essentially just standardizes the assumptions that I discussed so that it can ease comparability across studies. So there's still misclassification going on, but at least it's the same misclassification. So we can compare across cohorts, across outcomes, et cetera. And so this resource is available through NCI. The last thing I want to touch on, which Dr. Main also did as well, is the heterogeneity within this food group. So you often see these pictures when, in the media or someone's presenting on ultra-processed foods. What you don't see is these: whole grains, infant formula, baby foods, plant-based proteins, tofu, foods that we recommend Americans to consume more of, that they're not consuming enough of, and grouping them with foods that we want them to limit or just have on special occasions. So does it really make sense to combine these foods under one umbrella term? And as far as regulation, from what I've learned today, that's not a good idea. Oops. So there's been a lot of push to look at subgroups of ultra-processed foods, which I think is a really good step in the right direction. Because what we see, this is from the Nurses Health Study, is that the foods that you would expect to have a positive association with health do: breads, yogurts, cereals. And then those, kind of same old culprits that have been on our radar for decades, like sugar sweetened beverages, processed meats, those are the subgroups of ultra-processed foods that are associated with negative health outcomes. And this is observed in the EPIC cohort with type 2 diabetes, as well as also with cancer and cardiometabolic outcomes. So again, these foods in red are already on our radar. There are various public health agencies that already recommend to either limit, avoid, or reduce intake of processed meats, especially for cancer risk, as well as artificially and sugar sweetened beverages. So it begs the question, in my opinion, is what is this added value of the NOVA system based on what we already know and what you've seen today with what the FDA is already regulating and trying to push forward in the food system. But there's still a lot that we don't know, and definitely, our focus today is what research is needed. And so this is the research roadmap that myself and Kelly Higgins developed while I was at ARS. Mario, Kevin, and Kirsten were also there involved in this. And it's 6 questions that are focused on where the field needs to go in order to be able to understand, are ultra-processed foods adding value to our current dietary guidelines? Not going to read these off to you, but and so these is just a summary of the research gaps and resources that I shared. But one thing I just want to mention is that these challenges are not new, in my opinion. There's challenges with dietary assessment across any food group. There's challenges in definitions across any food group dietary pattern, like Mediterranean pattern. Red and processed meats have done a lot of work in that area where the definitions and descriptions there are very challenging as well. So this is not new, but it does have important nuances that need to be considered. And that's it.
All right, thank you. And thank you, Lauren, because like I said, I think between Susan and you've taken care of 95% of what I need to talk about today. But I think part of this is the repetitive message of really what are we actually debating? What are we thinking about in terms of providing guidance and in some cases regulatory structure? I think it's important to kind of step back and think, in the simplest sense. I was asked to talk about why we process foods. And in the simplest sense, it really is to convert raw materials for the same reason you do them at home and to make things fit for human consumption and for the absorption of nutrients and everything else. So it's really a series of techniques and methods that we apply, which is about as far from the definition of ultra-processed foods as you can get to a point. And I'll get into that. Again, I'm a food scientist. Lauren's alluded to that. I do a little bit of nutrition, but For 11 years, I did the food science for the non-food scientist course at IFT, and I spoke extensively in the context of why we process foods and why that's important. But realistically, when we think of the broader food supply, and I'll get to that in a second, we really focus historically over thousands of years. A lot of the processing techniques, cooking techniques, preservation techniques were there for preservation. Make sure that that food was available for us as we needed it, and we weren't wasting it, for safety, so we don't get sick, for quality, including nutrition. And I would also argue for diversification, because, you know, we do get bored if we eat the same thing every day, every meal. So it's inherently perishable food, and processing is a way that we use to preserve that and move it forward. So the increasing in shelf life, the increasing in sensory quality, this may go back to some of those hypotheses. As we alter those sensory qualities, are we altering that balance? And these are good questions, but this is why we process food. Convenience is another piece in that, right? You'll hear that term. Why are consumers purchasing? Nutrition is a distant third. If you look at the IFIC surveys, it's actually price and convenience still are two of the top. Flavor is certainly top, but price and convenience are right on up there in terms of factors in the lives we lead. So I'll introduce you to the concept of Fat Tom. I always usually try to make a joke about Fat Tom, but I tried not to do that because we're here in Washington and that could be misconstrued. But realistically, those are the factors that we try to manage when we are thinking about raw commodities or highly perishable foods, and we want to monitor what they're composed of. That includes the acidity levels that they're in, the temperature and the time in which they're held or processed. You're controlling oxygen and moisture. All those things are there to control microbial growth and spoilage. And so we're using processing and formulation in many ways to manage those factors and to convert highly perishable foods or perishable foods into stable foods, or highly perishable to perishable foods. So these are combination of techniques that are very traditional and have been around for a long time. It is important to step back. We could do this at home, but really the basics of the food system in the US, we forget sometimes, right? It starts from basic producers, farmers. So now I'm in a college of agriculture again, and I have to tell you, I hear a lot from the production side, and they're asked to produce. I was just looking at the numbers this morning in preparation, 15.2 billion bushels of corn this year, maize. That amounts to about 850 billion pounds of corn produced. If we don't process it, what happens to it? Why did we produce it to begin with? There's a whole policy debate there, I understand, but those have to be converted, and they have to be converted for multiple uses, some of which goes into energy and fuel, some of which goes into animal feed, much of which also goes into human consumption. And we have a large availability of food in this country because of that very efficient production system. But that goes into production of food ingredients as well into the value addition and food processing. When we think of food processing, and this is where the NOVA category fits, this is what the consumers see, but they're starting to learn more about what's on the other side, the right side of the screen, the food ingredient side, which is where those commodity streams fly in too, right? So just to be clear, many of the ingredients that we have in food are actually part of that. Lauren already talked about the distinction between food processing and formulation, but I want to add one wrinkle to that. You often hear the term, besides ultra-processed food, just processed food. And processed food is the combination of the two, right? So if you look at what that is at the bottom, this is coming from IFT, it's the combination of food ingredients and use of processing steps. That is what's been converted into the ultra-processed food definition. So they took what IFT has kind of used as a gold standard of distinction between food processing and processed food, and added the ultra piece to it because of the nature of some of the ingredients or processes that are chosen. What are the components of processing? These are unit operations, and Lauren kind of already touched on this. There's lots of different distinct unit operations, and I used to joke when I taught in food science that it depended on which university you went to. If it was taught with an engineering hat, they really talked about unit operations from the most basic sense, versus a product development hat, which taught about it from more of what I would say layman's terms. But these are just examples of different unit operations you would see in a typical food plant, including dehydration or baking or frying, freezing, coating, etc. The key thing is that it combines, no single food process is usually using a single unit operation. It's a combination. And I think this is where, if you think about the steps it takes to make your sugar cookies that you're going to smoke, I'm going to try this now over the holidays. This is what happens when you're a food scientist. I'm like, that's a brilliant idea. You'll see that there's multiple steps to that smoking in and of itself. But it's an integral part is the formulation part. So ingredients have lots of roles, right? So if you think about the use levels, and they're dependent on several factors, right? So how much do we use? There are limits, and the FDA has some of those limits for additives. If you think about it, flavor, aroma, texture, food safety, shelf life, and nutrition, these are the distinctions Lawrence was making. If you're enhancing shelf life, it may not be ultra processed, but if you're enhancing flavor, it is, which is interesting because preservatives get linked a lot in this. But predicting the functionality is critical, and not everything, and I'll get to this, not everything that is added is always not functional in a natural sense. But ingredient categories that are there, obviously sweeteners, texturizers, thickeners, gelling agents, preservatives, coloring, flavors, those are the ones you hear the most about. And I'll talk briefly about those because they seem to be the ones that are highlighted extensively as ones that are doing very bad things to us as individuals. And I'll touch briefly on that in a second. But just to go back to the premise, and Lawrence said this, it is a combination of steps from raw material to formulation to process. I'll add packaging to this, and I'll add the final product as presented to the consumer to this. And this goes back to maybe some of the questions of, is it the food environment, or is it the food itself, or is it the ease in which it can be consumed? There's lots of testable hypotheses here that may not actually involve the formulation or the process. But that being said, I think this is the Lane paper in BMJ that came out earlier this year that made a lot of headlines, this umbrella review. There's no denying that people are increasingly publishing and becoming aware of this association. And in fact, what happens is papers such as this and others will then lead to the press releases that we are often asked about as professionals in this field. And you can see here the two big links. One, the harmful additives that have infiltrated our system, and then the Washington Post, melted, pounded, extruded. And the example here is a whole grain kernel of corn versus a corn chip. And I don't know how many of you have eaten a whole grain kernel of yellow dent corn #2. But if you haven't survived, I need the number for your dentist, because that would not be an easy eat. So that's really a strange comparison to make. But if you've gone to their website, it's a really unique visual of how they talk about the process, the physical process. Now, this is Jan Ubnik and Adam Levine, who have actually written a nice review about the complexity of this story, which is the steps that you can go in the food space, and you can see the process culinary ingredient steps, the ultra processing steps that can all exist within the same food. What you see in breakfast cereals, what you might see in texturized vegetable protein, which falls into the category as Lauren talked about, or potato chips, which generally fall into the processed culinary ingredient, not an ultra processed category, but not something we tend to want to recommend people over consume. So again, the complexity of this is there, and it leads, as Lauren just said, very, very confusing linkages between things that may be ultra processed, but could be with multiple process steps, but it could be categorized as generally foods that fit into the MyPlate plan or minimally processed. As you can see, the number of process steps versus number of formulation steps. It doesn't necessarily always line up. There are general trends that we see there, but it doesn't always line up. So just to give a couple of examples, if you go back in time, if I had to go back to Mesoamerica 1,000 years ago, would anything they're doing be ultra processing? The answer is yeah, it would be. If you've had nixtamalization or nixtamalized corn, if you had masa, if you had corn tortillas, you've seen this process. It's whole corn that's soaked in lye. This is traditionally potash, right? The ashes from a fire, you would soak it in lye, you would heat it, soak and wash it, and then you would have whole hominy. The pericarp is removed from that. You get a textural difference in the corn. There's an appearance difference, so you're affecting those two pieces. That could be turned to masa flour or dough, which is sold and turned into corn tortillas. That, my understanding, would not necessarily be ultra processed per se, but if you look on the labels, it would be, right? It has hydrated lye as an ingredient, so it definitely would be there for visual purposes, flavor purposes, etc. And that is if you bought it from the store or if you bought the ultra processed version, which has additional ingredients, preservatives. Why are they there? Well, they're there to increase the shelf life from 2 days to 45 days, right? And that's a guess. That's my guess based on the shelf life that's on these labels. But at the end of the day, are those products that different? And that's where we get back to. Another example, if you actually take those and slice them up and fry them at home, there's your ingredient list. And if you look at the ultra processed version, corn, vegetable oil, and salt. It's very difficult for consumers to discern this, right? So I'm not making judgment on one being better than the other. I'm just telling you this is the complexity we're dealing with. Again, what is the big difference? The shelf life. So 6 months at room temperature. If you've ever made these at home, 2 days, you have a hard time chewing through them. So food additives are really part of that distinction. And I just point out, we're not far from the FDA. This is 1902, Harvey Wiley, former Purdue faculty member where I started my career. The Poison Squad was there. Look at the ingredients of study with that $5,000 grant: saccharin, bleach flour, caffeine, and sodium benzoate of sodium. So we haven't changed so much since 1902. We're still asking questions about these ingredients. Lauren talked about I don't go to the Library of Congress, but there's a great website that has substances added to food. And we've already heard about that. This is my cheat sheet book. If anybody ever wants it, I go in Hui, if you're a product developer, it's a great dictionary of food ingredients if you're ever wondering what's in there. But the whole point, and I think some of you have seen James Kennedy's slides before out of Monash, foods are composed of chemicals, okay? They're composed of chemicals. You can go through what he has listed here in terms of flavors and colors, and we have E numbers all over the place on the strawberry, right? And the chemicals also appear extensively in natural foods. We talked about propionic acid. I'm glad Lauren used that as an example. It's one I use often. That is Swiss cheese. It has greater than 40 milligrams of propionic acid per 1.5 ounces. The limit in the FDA is 3,000 milligrams per kilogram of food. So you're not that far off in terms of what you're consuming there. You hear the hypotheses related to emulsifiers in foods and those added. Well, there's two right there that I could pull out. The 700 milligrams of lecithin per normal egg. And 3% of the total lipid content of olive oil is mono and diglycerides, which are emulsifiers that have been linked to many effects. But apparently eggs and olive oil are still pretty healthy. And by the way, that's why Swiss cheese doesn't molt. It's the propionic acid, if you haven't noticed. So I'll quickly wrap up here. There's a lot, we heard this morning, the ag and food industry can adapt to regulatory guidance. I think the PHO, the sodium and sugar reduction, the voluntary reductions people have undertaken have been very good. I'll point out, synthetic food dyes are completely allowable in the US, but we've seen the majority of new product launches are leveraging natural color solutions because consumers have asked. So the industry can react. And just some considerations, it should be a unifying conversation to make the food supply better and more widely accessible to all. Cost is a big factor in what we do. And I think it goes back, I mentioned it to a few of you already. This is like having that schoolyard brawl and the teacher breaks it up and the kids forget what they were fighting about. They were so interested in just winning the fight. I think we have to have ongoing discussions about how to improve foods and build on what we've already done. We do have gaps in basic and clinical research. A lot of the work has been based on epidemiology, and I took out the slide that said this, but Don Landry, who was a former chair of medicine at Columbia University, said a quote to me in the ear the other day, and he goes, epidemiology is a great invitation to do some more science. And that's what we need here, right? Is more science. So the other thing we must consider is waste, and shelf life is important. I said stretching the availability and the cost of that food, or reducing the cost of the food, is key. Food losses and waste contribute so much to what we see, and a lot of that is happening in the household. It's also happening in food service. This is a great study just published by Reed in 2020. By Quentin Reed. And I think what's really interesting about this is it talks about the different points, but also really doesn't get into the fact that, we're very different than the developing world, which is losing it on the farm and in post-harvest. We're losing it in the stores and at home because of what we buy, and maybe you clean out your fridge every 2 or 3 weeks. With that, I'll say also,
we must remain, the cost piece is important. As we reformulate, it's going to impact what we do. So clear guidance is going to be important, but also clear plans to make sure we're managing the availability of food to the most critical in need in this case. It's a right to have high quality food across the population. And I think the food industry and the ag industry is there to support that, as we've shown that in some of the already changes that we're making. So clear guidance would be useful, but at the same time, I don't want to lose the practical piece of what we have. So I'll stop there.
Great. Thank you so much for having me. And in the spirit of repetition, I'd like to say, for the 95% that was previously covered, I think now it's 99%, and I'm about to add an additional 1%. So, okay. So I'm going to briefly be covering some of the global trends in ultra-processed food guidance, and then look at how those global trends might inform what we consider in the US. And then I'll conclude with some research and implementation gaps and some of these considerations that we've been discussing in terms of what do consumers see when they hear this kind of guidance, and how might that be confusing and have different unintended consequences. So again, unless you've been living under a rock, we're all aware of the very strong epi-evidence that links ultra-processed food consumption with adverse morbidity and mortality. And we're seeing that with that accumulating evidence base, there is action that's being taken, particularly globally and especially in Latin American countries. And typically it's being done in different ways, where there are either the eat more messages around some of the minimally processed foods or some of the eat less messages. And right now, as of a 2022 review, we see about 106 dietary guidelines evaluated. 45% are explicitly mentioning food processing in some form. But the way that they're addressing it, even though those guidelines differ, fall into sort of three bigger buckets. So first we have referring to characteristics that imply the presence or absence, which requires quite a bit of subjectivity and interpretation. We have discouraging specific ultra-processed foods, and then one that's more referring to the broader category of ultra-processing in general. Now, I'm gonna, we talked about increasing intake of minimally processed foods, but if we focus more on the eat less messages, we can see where some of the classification challenges kind of continue to arise. So while 52% or more than half of discouraged foods are ultra-processed, we also have these discouraged NOVA 1 and NOVA 2 groups, particularly things like butter and certain high-fat meats. And so if we want to have a language around eat less of something, we need separate guidance for these other foods which don't fall under the ultra-processed blanket. Similarly, if we're looking at things like cakes and breads, and you saw from many of those ingredient lists, it can be very difficult to disentangle which ones are in fact ultra-processed and which fall into a classification that wouldn't require additional regulation. And the same can be said of nearly a third of sweets and sugars can't be clearly identified as ultra-processed without more information on a label. So that brings us to the US, where it makes that much more complex when you look at the just sheer number of products in the US food supply. And so one of the things, and this is sort of a personal opinion that I think is important to consider, is what if we prioritize our policy around something where there's more universal agreement? For example, when we're looking at some of these nutrients of public health concern where there's already a lot of movement, we might have the greatest and most expedient, going back to the earlier talks today about the 30-year lag on improving public health outcomes, because most, not all, but most ultra-processed foods are high in nutrients of public health concern. And I use this Uncle Ben's here as an example because the average consumer in trying to navigate a grocery store would need to hold a lot of pieces of information in their head when making a decision. So here we look at the American Heart Association guidance, and we can see we're encouraging intakes of whole grains, and we're encouraging intakes of non-tropical liquid oils. And from the front of the package, you might say, okay, this meets that goal. If you look and have more in-depth knowledge and you look at sodium levels, you might say, okay, I shouldn't have that. But then if you add this other level of ultra-processing, you'd have to know that the additive that's in there is a cosmetic additive and thus would be moved into the ultra-processing. Now, that's also from the regulation standpoint, from what we've learned, that costs money to add this new criteria compared to something that we already might say requires a limit based on sodium alone and has a very simple message. And ultimately has the same net effect of discouraging consumption of certain products that we'd want to discourage. And nutrient-based guidance may be especially worth considering in the US context, where approximately 73% of the US food supply is ultra-processed. Now, this is using a different estimate than NOVA, that they looked at sort of the degree of ultra-processing using the Grocery DB database of 50,000 products. But again, in this same analysis, just like we've seen in a paper I'll cover shortly, most, but not all, ultra-processed foods in this case would be unhealthy based on thresholds for sugar, saturated fat, or sodium. And Lauren already shared this paper, but I think it's important to think about this when we're trying to sort of differentiate between different types of either front of package labels or, regulatory actions, because there's much more heterogeneity at this point in the association between ultra-processed foods and health outcomes than in the associations between nutrients of public health concern and health outcomes. And again, just like we have, recommended for the last 20 years, we're seeing that particularly like whole grain breads, yogurts and dairy-based, products have an inverse association with certain disease states, whereas some of the key drivers like processed meats and sugar sweetened beverages, as well as artificially sweetened beverages, increase health risk. And I really think in this context, getting it right really matters. And this has come up in this question of what is processing versus formulation. Is a processed food the same thing as an ultra-processed food? And just the myriad headlines that we see every single day about, is this bad for me? And whether or not that's going to affect consumer confidence in the food supply. And I think, while not everyone will agree, despite that very long operational definition that was shared, I personally still find identifying ultra-processed foods difficult and confusing. Whether it's looking at the codex, which I've looked at extensively as well, and seeing that it can be classified as a preservative or as a coloring agent or multiple things, that multiple classifications make it difficult. And we know that if we try to use euphemisms for ultra-processed, that can also be confusing and obviously not valid for a regulatory standpoint. Additionally, kind of to your analogy, Mario, about the brawl, most discouraged foods are ultra-processed, but some are minimally processed. So why only provide guidance around the processing level when we can have other terms that are clearer to communicate and really have the same net effect? I think, again, that why getting it right matters is that when we don't have clear, consumer-friendly, actionable guidelines, there is a risk of perpetuating consumer confusion and mistrust about dietary guidance, which we did see with the low-fat diet debacles. But just because we have this confusion doesn't mean that I think we're at a place of inaction. And I think this is why this kind of meeting is really important, because I think there are a lot of things that people agree upon, and we focus more on the areas of disagreement. And given that there are unequivocal adverse associations with foods high in nutrients of public health concern, which also largely happen to be ultra-processed, if we use that as a jumping off point, we can probably have incredible forward momentum on this tremendous public health problem. Which brings me to some gaps. So when we think about how to change the ultra-processed landscape, I think it's important to remember that the key to effective problem solving broadly is to know what the dimensions of a solution look like. We look at the average healthy eating index score in the US, and it's in the low to mid 50s. Our bigger problem goes back to where this meeting started, is that the average US diet is really, really poor. And a huge public health win would be to have a big shift in what people are eating at this level. So then we need to think, if we're going to do that, what's the context of what does the US diet look like right now? Is this solution feasible that we're proposing? Can we optimize it as more data comes in and not have it be a static one-time only fix, but something that's iterative and then evolves as new research comes out? So when I go back to thinking about those three primary approaches, what's best suited for the US context? So again, in the US, if we use the NOVA classification, 60% of calories in the US diets come from ultra-processed foods. But if we try to focus on regulating all ultra-processed foods, we could have unintended consequences for healthier ultra-processed foods, which also support the multifaceted goals of the food system to have a food supply that is affordable, safe, accessible, and maybe will sustain planetary health. If we focus solely on ultra-processed foods, we'll miss 16% of the foods that are considered high fat, salt, and added sugar. Now, if we focus only on the HFSS foods, we also miss 16% of the foods that would be considered ultra processed. But I think that does beg the question of, is that good enough when we're seeing heterogeneity in some of these associations? Is that 16% gap moving us in the right direction from a population health standpoint? And just, putting it another way, 50% of the food supply is ultra processed per NOVA. 43% is high fat, sodium, and sugar. So is that 7% disparity worth fighting for? Is it worth the inaction that we're currently at? So when we think about the next step of feasibility, I think looking at Chile's law, which focused exclusively on HFSS labeling and marketing restrictions, can provide good insight into both feasibility and effectiveness. So for those who are not as familiar with the exact elements of the law, these different warning symbols were required to be put on foods that exceeded certain thresholds for these nutrients. And those nutrient labels, the thresholds for the nutrient label, become stricter over time. And products with these front of package labels are also restricted to child-directed marketing restrictions, as well as banned from sale and promotion. And initial research does show that this has been effective. So in a pre-post study, the calorie, sugar, saturated fat, and sodium content of what's being purchased has all decreased. And as that phase 2 has been implemented, we're seeing a similar trajectory. So to me, that's very compelling evidence that this does have an effect on population health. So in the US, again, I'm reiterating this point, but 43% of the foods in the Nielsen database are HFSS, 50% are ultra processed. If we just use the HFSS criteria, we're going to miss 16% of ultra processed foods, but maybe some of those foods are foods we might not be ready to discourage yet. So again, is there enough evidence for regulatory policy efforts to address non-HFSS UPFs? And just to add, if you were to add the non-nutritive sweetener piece to this, depending on evolving evidence, then there would only be a 9% disparity between HFSS and ultra processed. So that brings me to some research and implementation gaps, and I won't go through these in detail since we've heard about them, but we do really need to understand, if we want further action, what are the nutrient and energy independent mechanisms? Can we look more at evolution beyond a binary, this is ultra processed or it's not, when there's degrees of ultra processing? The distinct effects of processing and formulation, as well as the health effects of high quality, high ultra processed patterns and high quality, low ultra processed patterns, which Dr. Hall, I'm excited, is going to share more. Some information on threshold as well as life stage effects. And then from implementation, we really don't know yet what consumers understand and how the information can be best communicated to them. We do need a precise operational definition that it doesn't require a PhD to discern, and even with a PhD, we're not necessarily able to do so. But even as we do this, we can't do this in a vacuum. We need to, if we're going to be doing this, we need to be increasing access to healthy, convenient, affordable alternatives to promote nutrition security and have a plan for managing evolving guidance. We know that this isn't the end, this is the beginning. And as we do this, thinking about this from a multi-level standpoint, where we can't just be looking at incentives or taxes, we need to be doing education, marketing regulations, and changes to procurement policies to support this kind of effort. So policy changes do need to balance the benefits, risks, and harms. From a beneficial standpoint, we can increase consumers' right to know, which they want, and satisfaction. This can drive food system change, which we've seen, and we can drive environmental benefits if people are willing to pay. However, adding a new label to foods, especially when it's overdone, may make it harder to process and evaluate information. We know that labels cause halo effects in people to make other non-product related inferences, and it can increase consumer fear and demand, as well as increasing food prices. And then the worst case would be it stigmatizes foods that don't harm human health based on current evidence and could greatly reduce productivity. So I'll leave you with my thoughts are, if we target HFSS foods using a combination of labels, taxes, and marketing restrictions while simultaneously enhancing access using subsidies and availability, we can make a huge dent in population health while we evolve targeted research looking at additives and other UPF dimensions to update guidance. Thank you.
So thanks so much for the opportunity to talk to you today about our are research that's really trying to focus on elucidating mechanisms of ultra-processed foods and diets that are high in ultra-processed foods. I was really heartened by some of the comments from Susan Main this morning and the really important nature of understanding mechanisms and how that can be useful for regulations and other policies, and for Penny Chris-Etherton's remarks about the really important work of these really small trials early on in the development of trans fats, things that actually wouldn't have passed the stringent criteria for the Dietary Guidelines Advisory Committee, for example. There were only 10 subjects for 3 weeks at a time, wouldn't have met those criteria. And neither will any of my studies that I'm going to tell you a little bit about. But I think, again, from what we heard this morning, we're in this situation where we have a epidemic of diet-related chronic diseases. This is not a small problem, right? This is costing hundreds of thousands of lives, more than $1 trillion a year to the US health system. And over again, we've seen that diets that are high in ultra-processed foods, and that's using some sensitivity analysis here, that basically are part of the problem. And so our public health researchers are saying, UPFs are really bad news for the health of the country. And many of the analyses are even trying to look at the effects of ultra-processed foods beyond nutritional profile of the diets. There still seems to be some signal there beyond just the traditional culprits that we often think about. We heard from Commissioner Califf this morning, and he made this remark earlier this year, that the observational data shows there's a pretty clear association between ultra-processed foods and a lot of bad health outcomes. But before the FDA can do anything about that, we're going to need a lot more research. And so what I'm going to describe to you is some of our research that's been published and some ongoing research with some interim analysis that I'll share with you today about what we're trying to do to look at some of the mechanisms of diets that are high in ultra-processed foods. And so we were the first folks to do a randomized control trial of basically, the way I like to think about this is food environments that have varying different exposures to ultra-processed foods. And what we do is we bring people to live with us at the NIH Clinical Center for 24 hours a day, 7 days a week, and they stay with us for a month. And for 2 weeks, they're exposed to a food environment that contains no ultra-processed foods, and then switch to 2 weeks where they're exposed to a food environment that contains 80% or more of calories from ultra-processed foods. And they're randomized to receive these diets in random order. So 2 weeks on each of these diets. And importantly, one of the things that we do is we try to match for the presented calories, fat, carbs, sugar, sodium, fiber, glycemic load, overall energy density of the diets, variety of things that we try to match for to try to get at, is there something about diets high in ultra-processed foods outside of these overall patterns of these nutrients of concern that might be driving some of these effects? And basically, people are given really simple instructions. Just eat as much or as little as you'd like. Don't be trying to change your weight. We're going to measure lots of different things. And what they don't know is that our really talented dietitians and chefs who prepared these different 7-day rotating menus are measuring all of their leftovers. And so they basically have the foods presented to them. They're equally convenient, they're equally cheap, they're free. And so all of the other sort of things that happen in the real world that drive our food choices are sort of taken out of the equation. And in this first study that we published in 2019, people did as they were asked. They ate to the same degree of hunger, fullness, satisfaction, and eating capacity in both the minimally processed and the ultra-processed diet. But to do so, these same folks were eating about 500 calories per day more on the ultra-processed diet as compared to the minimally processed diet. So a huge difference. And this is just a snapshot to think of, how does the food environment, even this very artificial setting, how does it affect the biological control of calorie intake? It seems to have an enormous effect, and we're really interested in understanding the neurobiological aspects of that, but that's a story for another day. Not surprisingly, given those huge differences in calorie intake, people gained weight. The same people gained weight on the ultra-processed diet, lost weight on the minimally processed diet. They're blinded to their weight measurements. They're wearing loose-fitting scrubs. They can't tell if their clothes are getting tighter or looser. And so, but their back is to the scale when we weigh them every morning, and they're changing their weights quite a lot, despite being asked not to be trying to do so. And similarly, when we look at their body composition changes, how much of that weight change is coming from body fat, for example, you can see these parallel changes. They're gaining body fat during the ultra-processed diet, losing body fat during the minimally processed diet. So when we published this paper back in 2019, it was a bit of a Rorschach test for the nutrition science community because everybody seemed to see in the data what their pet theory was about what's driving these effects, right? Was it the fact that people were eating more quickly? Was it that there was slight differences in the amount of protein in some of the meals and maybe that was driving excess calorie intake? So we took a kind of a secondary analysis, post hoc look at the data, and a couple things seemed to pop out. One was, if you consider this definition of hyperpalatable foods, which was created by Tara Fazzino at the University of Kansas, which is defining hyperpalatable foods as crossing certain defined thresholds for pairs of nutrients. So a food that was either high in both fat and sugar, or high in both fat and salt, or high in carbs and salt. If they passed these defined thresholds, it was deemed a hyperpalatable food. And when we did a mediation analysis to say, did the number of calories coming from individual hyperpalatable foods that was presented on the plate, did that influence energy intake, the difference between the two diet conditions? It seemed to mediate about 40% of the effect. And similarly, when we looked at non-beverage energy density, what a fraction of the solid foods on the plate that was that were the overall meals were energy dense versus less energy dense, that seemed to mediate also about 40% of the effect. So these are sort of hypothesis generating ideas of analyzing the data post hoc to see if maybe what's going on is that these ultra processed diets tended to be higher in hyperpalatable foods and tended to be higher in non-beverage energy density. So what we have is now a study ongoing at the clinical center, again, where people are staying with us for 24 hours a day, 7 days a week. And they are we're basically looking at 4 different diets. They're exposed to them for 1 week each in a randomized counterbalance sequence, and all the diets are matched for macronutrients, fiber, sugar, and sodium. Diet number 1 is a minimally processed diet with 0% of calories from ultra-processed foods, and it's low in non-beverage energy density, low in hyperpalatable foods. One diet is quite similar to our original ultra-processed diet. It ends up being high in non-beverage energy density, high in hyperpalatable foods. And then we've sort of re-engineered 2 new ultra-processed diets that are both 80% or more of calories from ultra-processed foods, but one of them is high in non-beverage energy density and low in hyperpalatable foods. And the final one is low in both non-beverage energy density and low in hyperpalatable foods. So just to kind of give you an idea of what this looks like, these are some select snapshots of some of the nutrients and properties of these diets. I'll focus on the things that are varying the most. You can see that in the minimally processed condition, about 53% of the calories are coming from hyperpalatable foods, according to Fazzino's definition. That goes up to 74% with the ultra-processed high, and then back down to the mid-50s with the ultra-processed high-low and low-low. Non-beverage energy density, about 1 in the minimally processed and the ultra-processed low-low condition, and close to 2 in the ultra-processed high and ultra-processed high-low condition. So we recently completed an interim analysis of the first half of the study subjects, and they range in age from 19 to 52. There's a very wide range of BMIs from 21 to 42. It's a pretty wide inclusion set of criteria. People just can't have type 2 diabetes is one of the major exclusion criteria. And so I'll present some of the interim results for you. They did as they were asked. They ate to the same degree of hunger, fullness, satisfaction, and eating capacity in all 4 different diet conditions. Importantly, there was no significant differences in how pleasant people found the meals. Okay, and that was also true in our first study. People did not, it's not just that they liked eating the ultra-processed diet more than the other ones. That wasn't what they reported. They reported liking them equally as much. People, there was actually no differences in meal eating rate in this study, which was interesting. That was slightly different than our previous study, where people ended up eating the ultra-processed meals much more quickly than the minimally processed. In this case, there's no significant difference. Again, all interim analysis at this point. So our primary outcome, like the first study, was the ad libitum energy intake differences. And so when people were on the minimally processed diet, they were eating, around 2,700 calories per day. When we switched them to the ultra-processed high diet, drum roll, there's about 1,000 calories per day increase in this case. Why is it so much bigger than our first study? Well, to simplify things for our kitchen, we basically did not titrate the number of presented calories to every individual subject this time around, like we did in the first study. Instead, everybody got 6,000 calories a day possible to eat. Okay, so for some people that was way more than twice their energy requirements. In some cases, it was less than twice their energy requirements. Maybe that's one of the reasons. Another thing that we did differently was we didn't use snacks in this study. They only had the 3 eating opportunities at breakfast, lunch, and dinner. We didn't find in our first study differences in snack energy intake. And so we removed that from the equation to simplify things as well. Okay, so what about the other 2 diet groups? Well, when we keep the energy density high but lower hyperpalatable foods, that goes down slightly, not statistically significantly yet, but these are interim analyses, 200 calories per day lower. And when we reduce both the number of hyperpalatable foods and the energy density of the diet, that drops quite significantly to about 630 calories a day less than the UPF-HL group, and then 830 calories a day lower than the ultra-processed high group. So at the end of the day, but the difference between the minimally processed and the ultra-processed low group was quite small, relatively nominally at this point, 170 calories, not statistically significant yet, again, only interim analyses with half the data. What I want to point out here is that these changes in the food environment, once again, when people aren't being asked to, control their weight, they're not being asked to do anything, are having enormous consequences in the regulation of food intake. And I think that this is really important to think about when we think about when people are going about their day-to-day life, and they're essentially, for practical reasons, price and taste, being driven to consume diets that are more in the UPF-HH category, right? This is having an enormous effect. What about body weight? Well, basically corresponds to the energy intake differences. And so the kind of encouraging thing here is that you can have a diet that's 80% of calories from ultra-processed food that does not lead to weight gain. Okay, so there's no significant difference. And these are the same people, right? No significant difference between the body weights when these same folks are eating the minimally processed diet that has 0% of calories from ultra-processed food and the one that has 80% of calories from ultra-processed food, as long as it's low in both hyperpalatable foods and non-beverage energy density. So I thought maybe we've nailed this, right? Maybe we finally figured out what it is about ultra-processed foods that typically drive overconsumption of calories. And then a little monkey wrench was thrown in when I said, okay, well, let's look at the body fat data. Only the minimally processed diet led to body fat loss. So the ultra-processed low diet doesn't lead to significant body fat gain, but it also doesn't lead to body fat loss. So I think this is something really interesting to start to unpack. Is there something about the digestibility of ultra-processed foods that's not being accounted for in our nutrition tables that we're looking at to analyze the ad libitum energy intake data? We did not collect all the feces and urine over the course of the entire study to better understand how much was actually being digested and absorbed. I think that's an important question to ask in the future. Is there something going on inside the body with respect to energy partitioning, which is leading to what essentially is fat-free mass loss during the ultra-processed low condition, whereas here it's mostly body fat loss in the minimally processed low condition. So I think there's really interesting physiology, basic physiology to unpack here. And even beyond obesity, there's really interesting biology to try to unpack. We basically did our interim analysis to apply for a bench to bedside award to choose 2 of these diet groups to do a deep dive on the microbiota, the gut function, and immune system phenotyping in these particular subjects. We don't know if we've been awarded that yet. Hopefully we'll know soon. But this is work that's ongoing with Catherine Mackey at the NIH Clinical Center and Michael Sack at NHLBI, all within the intramural research program. And all of this research is being done here at the NIH Clinical Center. We're really fortunate to be able to have the kinds of facilities to house and feed people. This is our metabolic unit that was opened in 2007. And I wish I could say that I have a lot of optimism that we can continue to make a lot of progress on this. Shortly after our first ultra-processed food trial was completed, 30% of the beds were taken from our unit. And Very shortly thereafter, they actually proposed closing the unit permanently. And I wish I could say that this was just specific to us, but this has been an ongoing trend in basic human physiology research. Back in 2016, there was a eulogy written in Diabetes Care for the metabolic clinical investigator. There's just been a dwindling of support for basic human physiology, trying to understand the root cause of what it is about our diet that's affecting metabolic health in America. So what do we do about that? Well, one of the other things that's frustrating about our studies is we're only able to currently feed 1 or 2 people at a time. Why is that? Well, they're on a hospital ward. Do they have to be on a hospital ward? No, they don't have to be on a hospital ward. We could rent facilities in some rural area. We could house them in more comfortable locations as long as we can control their access to food and be able to feed them. We could feed dozens of subjects at a time. Our study that's ongoing now, that's going to take about 2.5 to 3 years to complete, not from now, hopefully we'll be done this coming summer. We've been doing it since 2022, could have been done in a few months if we'd had the ability to house and feed people in a controlled setting. So this is the modest proposal. If you want to do more research like this, we need to have facilities to domicile and feed subjects. This was probably the worstly timed article in Science, came out in March of 2020. Our interests were other things in March of 2020, but I made this case of why we need this kind of facility to really accelerate the research on both ultra-processed foods as well as other things that are causing the chronic diet-related disease epidemic that we currently face. I'd just like to thank the folks who've really done most of the work on these studies, and I'm looking forward to questions.
Thank you to all of our speakers. And can I just say, I am in awe of our speakers' ability to synthesize, to compile and synthesize and cover so much complex information in such a comprehensive way. So please join me again in thanking our speakers. And thank you also to my co-moderator here, Alison Brown, Dr. Alison Brown. So we will go ahead and take some questions from the audience. If you would like to ask a question, please come to one of the microphones at either side of the room here. And we'll also be moderating questions or looking for questions online as well. But maybe I can take first stab, if you like, at asking, clearly there's this very strong signal, as you mentioned, as all of the speakers mentioned, this epidemic of diet-related chronic disease, this urgent problem, and this very strong signal, this linkage between consumption of ultra-processed foods and negative health outcomes. At the same time, there are some very important complexities and nuances to be aware of, and great overlap between ultra-processed foods and those that are high in added sugar, sodium, and saturated fat. But given some of the intriguing results now just presented by Dr. Hall, I'm wondering if I can ask each of the speakers, given all of this, given this strong signal, but also given the nuances, what do you recommend? What is your suggestion on a key next step? What are 1 or 2 key things that you think we should be prioritizing and recommending for action to do to try to tease out even more of an understanding behind these ultra-processed foods? So I don't open it up to anybody who would like to. Go ahead. And please do make sure that the light is on so that our online audience can hear.
Sure, I'll jump in. And since I sort of left it with that, I mean, I think after Dr. Hall's talk, I still feel very much in the same camp of when we have the overlap of nutrients of concern with ultra-processed foods, if we begin there and then use the additional savings and healthcare costs to fund the domiciled studies, we can start to unpack some of those mechanisms around, is it additives, is it certain elements of food processing, to try and understand what's happening, with metabolic parameters more closely.
I want to echo what Maya said in her presentation, is that we have to get it right. And I think that we really need to address the potential for different language that we all agree with. I think that the ultra part might be a little bit inflammatory in some avenues. And so that kind of shuts down conversations right away. But as you can see, there's a lot of overlap. There's a lot of the hyperpalatability, the formulation, the salt, sugar, fat, like that issue is not going away. So there's a lot of agreement. I just think that we're talking over each other because we're using different terminology or we don't agree on the terminology. And so I know there's a lot of movement, like Kevin, you mentioned the WHO trying to redefine the word ultra processed food, but I almost think we should just kind of take a step back and think about using that term going forward, and if that's the most productive thing.
I will say, however, just in defense of the term, in defense of, we have never had this much engagement with consumers or the general public on sodium or sugar or saturated fat as we have within this concept of there's something about our food environment that has changed dramatically over the past half century. What is that, and how has it been related to the increased prevalence of diet-related chronic disease? And yes, we need to understand the mechanisms, and they may be those very things, the saturated fat and the sodium and the sugar and a lot of the things that we already focus on. But we also need to capitalize on this engagement with the public and some political will, as we heard earlier, which is necessary to make advancements in this area. And so I think that has been one really important benefit to this, controversial definition of ultra-processed foods, is that this is not a small problem that we're facing, and we're going to need the political will and the administrative will to actually tackle the problem that we're facing. But I agree with you. We need to know, we can't miss the target, right? We need to better understand the mechanism so that we can actually hit the target and make real change. So in somewhat of a defense of the terminology ultra-processed foods and the attention that it has gotten, I think it's one of the things that we have not seen for a very long time in this space. And I think it's needed.
Yeah, I'll just be very brief and build on it. I think these are three great comments, and I think the engagement piece is key, right? The more we can teach, the more we can discuss, the more we can have a conversation about what the food supply is, and how we're actually delivering on not just what's important, but perhaps missing some steps in that opportunity is going to be important. Just want to be careful here that we don't want to overstep. I don't know if we'll ever get to the definitive answer. I think, Kevin, you're right. I think we need to understand why. The mechanisms are important. I don't think we're going to settle into one single mechanism. That's an opinion. And so I think where we move may be best learned from what we've done well already, with the PHO, with the voluntary reductions in sodium. I think that there are roadmaps for some of those, but I don't think it's as simple as finding a single mechanism. I do think that the complexity in this will make it very difficult for us to continue a very clean line of communication with the consumer, because I think much of this has already kind of run out in a way that's very difficult to kind of wind back. But we should take advantage of that conversation, because I don't think it's been one that we have necessarily had as much dialogue about. So that's a very good point.
Great, thank you. Dr. Shriti Kumnika.
Thank you. What I haven't heard so far is trend data on how food processing has changed. So this is taking the comment, something has changed in the food supply, well, it's the processing all along. But the discovery of de novo labeling suggests that the way things are processed may have changed in ways that change the package, and sugar and fat are being presented. And so I wonder, maybe from the Library of Congress, you can talk a little bit more about that.
So a lot of things I'm not going to live down today. So what happens end of the year with no sleep. So I think I'm going to answer your question in kind of a roundabout way. I think we need to take a step back and realize where Novo was developed, and it was in Brazil. Brazil has a very different food supply than we do. Well, it's coming our way, but when this was developed, a very different food supply. They also have very different home cooking practices. They cook a lot at home. There's more family meals. It's not the same environment that we have in the US. So in Brazil, when the influx of ultra processed foods came to Brazil, those are snack foods that anybody would agree should be consumed in moderation. But it was a subset of the Brazilian food supply that is ultra processed. Whereas in the US, it's what, 70%? And it's not just those sugar sweet snacks in those foods. So I think we just have to kind of come back to what the original intention was in the Brazilian dietary guidelines. And it may not be the most appropriate thing for here in the US or other countries.
I just wanted to add, we were talking about this after the PHO discussion from this morning, the example. I think we also have to understand that components of our food supply have changed over time in ways that consumers may not necessarily overtly see. The oils in this country have changed in fatty acid composition dramatically over the years in response to several factors, one of which is removal of trans fat. The other one is increasing stability of those oils. So we are predominantly dealing with a lot more oleic acid in the food supply today than we were 20, 30 years ago. And certainly that has been a big change. That alters the way we process foods in terms of how we formulate for stability as well. So there are changes that are subtle that we don't see and you may not pick up just from picking up a label unless you have a discerning eye like Lauren and you can read inside the breakdowns. But there's other examples there too. The protein components and others have changed as well. It's not just the little additives that are there. There's been changes in feed streams as well.
Can I just add one other, and also adding to that, if we look at the broad changes in the food supply, to your point before about corn, when we have such a high amount of certain crops that are grown and then we use those as raw ingredients, we have a huge number of different packaged foods, regardless of the additive or formulation, that are priced very low in the food environment and are very, very available and targeted to, communities where there are already a lot of health disparities. So I think those are all food environment problems. And to a point that you made earlier about, we don't get the same momentum with discussing saturated fat and other things. And I think it's a bigger question if we don't get the same momentum with education. We get more momentum when we have changes to the food environment that target a lot of these foods that we know have adverse health consequences.
Great, thank you, Mary. Please.
Mary Story, Duke University. And what hasn't been mentioned is the food industry. Who is making all these ultra-processed foods that many of us would say aren't really even food. And so what role do you think FDA can play in terms of regulation or really changing policies around ultra-processed foods?
I'll open it up to the members of the panel just to see, but one thing that's very clear, Mary, I can say is that in order, as we discussed this morning, the strength of the science underlying and it will depend on the mechanisms as those are teased out in order that would inform then any potential regulatory action. But so it starts with the science that could then determine regulatory approach.
That'll take years. What can we do in the next few years? Because we can't afford to wait. People are dying globally. And so I know that I hear what you're saying, but when you also mentioned the word epidemic. So exactly. We can't just continue and say we need more research.
No,
we have the evidence that sodium, added sugars, and saturated fats, everyone would agree we don't need more evidence.
Yes.
We need more evidence on the additives. But most of the additives that people are concerned about are kind of non-essential additives, not the preservatives. Nobody wants to go back and be in the kitchen 6 hours a day.
Right.
So we're in a real dilemma. because we do need the evidence, but we already have a lot of evidence.
I think you're exactly right, Mary. And we have the evidence is clear on added sugars, sodium, and saturated fat. And I would say there's a lot of action already underway exactly on those. And it was Maya and also several others who mentioned that the evidence is clear and that action is being taken, and that if we're addressing those, we're hitting most of the ultra-processed foods. But there's also an additional gap. And I think particularly some of Kevin's new work is hinting at what may be underlying there. So I feel like there's already action that is in process, that is action is being taken, and more is needed. And in order to inform that, some additional research would definitely be needed. Looks like Mario, you're looking to chime in.
No, I was going to say regulation versus guidance is always going to be the question. I think we can regulate based on what we know and where the science is. That's kind of where you were going. Guidance is a different piece, but it becomes a little difficult to do that in the context of we still get confused over definitions. So I think a big step of where we want to go is going back to a bit what Lauren said and a little bit what Maya said. Let's start with the large percentage of what we know, and that is in the salt and sugar and saturated fat piece. So that's already falling clearly into the regulatory spectrum. Now, once we better understand some of the mechanisms, I think we can provide better guidance before we get to that next step. I think, as I mentioned, the industry would react. You tell them to take PHOs, that ingredient is no longer grass. Okay, they can formulate products and process products. There will still be ultra processed food products, but certain products require saturated fat to be made. Now, you don't have to choose them and you don't have to recommend them, but they require it. And if you're going to deal with salt reduction, you can't take it completely out of the food supply, nor is that a good thing. So the voluntary reduction to 3,000 is far off of where for sodium is far off from where some of the most aggressive public health recommendations might be, but it's also seemingly a level that can be achieved with existing technologies. And when I say that, a lot of folks will stop and say, well, what do you mean existing technology? You just take the sodium out, right? Well, no, you don't. You have to reformulate that product, and that requires ingredients and technologies which may put certain foods back into the ultra processed food category. And so at that point, are we muddying the waters even more? So that's where I fall into a very difficult piece of, do we use that term ultra processed food to regulate on, or do we focus on what we know?
I'll just add also that, I think that 16% gap where the overlap between HFSS foods and ultra processed, I think would be a really good place to start, because then we could stop talking about those foods that we all agree are unhealthy and have more targeted efforts looking at that 16%, because I do think there are still questions about, that low, is ultra processed, regardless of the terminology, whole wheat bread less healthy than less processed forms? I think that's an open question, and we should be studying that to get at your question of, is it all additives, regardless of what they're used for? Is sodium citrate potentially harmful? I don't know. And I think we need to study that, but only when we can get rid of this big elephant in the room that these other products are overconsumed and overavailable and inexpensive and overly marketed.
But thanks for the question, Mary. I mean, certainly hugely concerning, a huge area that is already a substantial amount of action is being taken on this area of ultra processed foods, but there are open questions, and that's what we're looking to answer here today, or at least get an indication of the most critical questions that we should be examining and pursuing more research on. And so, Lauren wanted to add something. Go ahead.
I just want to. I muted myself. I just kind of want to bring it back down for a minute. Ultra processed foods are a component of nutrition. Nutrition is a component of our overall lifestyle. And so why do people go for ultra processed foods? I think we need to think of that. You don't have time, convenience, taste, cost. if you don't have childcare, if you're working, 12-hour days like we do, you're going to be leaning towards these ultra processed foods that are convenient and easy to consume or to feed your child. So I think there's a lot of other things, regulating or labeling ultra processed foods is not going to fix the obesity epidemic. And I think we just have to kind of think of that sometimes. A lot of people compare our food supply to the European food supply and think that that's the reason why Europe has lower rates of disease. They also have free healthcare. So like there's a lot of other things that kind of fall out of the picture when we get so focused on the ultra processed foods conversation, I think. And I think we really need to think about, again, why people are going for these foods, and are there ways in other social networks and social support systems that could help people not have to rely on ultra processed foods. And I think that plays a big piece into this overall picture of the epidemic that we have.
Thank you. Benoit.
This is a perfect
Would you
introduction.
like to-
Benoit Lamache from Canada. Thank you. A perfect introduction to my comments / question. So there are some advocates for banning ultra processed food, and then you could add to the good, the bad, and the ugly slide the unintended effects on who cooks actually, and where is the burden going to land in the family, and that's going to be still women, and we don't want that. Now, I feel there's an opportunity. We talk about ultra processed food or highly processed food or whatever their name on the negative side, but I think we need research to show whether there are such things as healthy, highly processed, or healthy ultra processed foods, because that's the solution. If we can show that there are healthy ultra processed foods or highly processed foods or whatever, then we have a solution. And we don't hear that side of the story very much.
Yeah, I mean, I think that's what we're trying to get at, right? What is it about, how can we incorporate what is within this category of ultra processed foods within a diet that doesn't lead to negative health outcomes, and in fact could potentially lead to positive health outcomes? And, at least as a proof of principle, we showed that you can formulate a diet that is very, very high in ultra processed foods and does not lead to weight gain. And yet people will report that they find the meals equally pleasant as the diet that leads to substantial weight gain. So what kinds of research do we need to do to better understand how to, make individual foods like that, as well as replacement products for the kinds of things that we're going to allegedly start to regulate and have policies around? We have to provide the convenient, tasty alternatives that are not that don't require a lot of time and equipment and skill to prepare, that are going to actually promote health. And many of those products will likely be ultra processed products, but they will have to be informed by the science that tells us how to do that without the negative health outcomes.
So maybe I'll take a question from online, and it might follow on a little bit from this recent discussion. Well, in considering the health effects of different ultra processed foods, have you considered the contribution of enrichment? So I'm thinking fortification, enrichment. I know a couple of folks mentioned that, but I don't know if anybody would like to take that on.
I mean, there's been several studies that have kind of highlighted the role of fortification in terms of closing nutrient gaps, micronutrient gaps in particular. I just don't think that's part of that equation in the conversation today. Absolutely, enrichment and fortification both are key components to what we see in terms of processing, returning what is known to be taken out in the enrichment perspective, or fortification, creating opportunities to meet the nutrient needs that have been identified in dietary guidelines when we know we can't achieve that through a normal dietary pattern in a reasonable way. One of those examples might be omega-3 fatty acids, where if we were to eat all the fatty fish in the ocean, we would probably still have a shortfall. So we have to have opportunities for fortification to meet the needs for the population levels we have. So those are the positives. How we get there and then how it gets defined are different paths, but technology is a big part of how we get there. Especially for things such as omega-3 fatty acids. We're not taking them from fish oil anymore. This is a whole different industry now. So that, and then the role of biofortification in terms of the plant genetics, we're changing of what's happening there.
I just want to clarify. So I put up that table of additives very, very quickly in my presentation. And fortification and enrichment does not make a food ultra processed. So I just want to make that that clear for the audience that may not, be in my head. But so enrichment and fortification does not make a food ultra processed, but they are often guilty by association because there's other additives needed in order to fortify and enrich foods. I imagine that's why. So it's not the reason, but a lot of fortified foods are ultra processed, and they, like Mario said, help us meet nutrient gaps that are critical. So
Yes, I do want to
Would you,
add,
sorry, would you identify yourself?
yes,
So for the
Rafael
online
Perez
audience.
Escamilla from the Yale School of Public Health. I do want to add that another big reason why people go to ultra-processed food is because of the enormous amount of marketing that the food companies do of their products from cradle to grave. And very concerning that in our country, they specifically target low-income people and communities of color. So we have to also remember that 11 food companies control the production and distribution of 70%, 70% of ultra-processed foods in the world. And they have an enormous power of lobbying that leads in our country to receive an enormous amount of subsidies for the ingredients that they use to produce their products at the expense of subsidies that would give access to low-income communities to healthy foods. So I would disagree very, very strongly that this is destiny, that this is the way it is because this is our lifestyle, and that cannot be changed. I also have to disagree very strongly that the NOVA classification is only appropriate for Brazil. I have lived in Brazil, I have worked in Brazil. Brazil started later than us, going into the double burden and triple burdens of malnutrition. But Brazil is not different anymore in terms of the marketing, in terms of the access and the types of ultra-processed foods. This is a global problem. This is not a problem of Brazil or a problem of Mexico. It is a global problem. And with regards to the marketing, this issue is so serious and the evidence is so strong that this is why the laws that have been issued in Mexico and Chile and Colombia and Peru and Brazil and Argentina about front of package warning labels, they are all complemented with the laws that protect children against the marketing of ultra-processed foods. And I want to conclude by saying that it is also extremely worrisome the way the ultra-processed food companies have taken over the Olympics, the World Cup, and almost all the most important sports events in the world. And I always say this was my last comment, but I do have another comment. Yeah, and that is that I do hope that we all understand that the reason the NOVA classification came to be was because us public health nutritionists would use the term junk food. Oh, that is not a scientific term. Junk food doesn't mean anything. We would say unhealthy foods. The American Dietetic Association would state there are no unhealthy foods. All foods can fit in a healthy diet. And we kept on seeing in the dietary guidelines for Americans, when it comes to healthy foods, they would name them. Eat more fresh fruits, eat more fresh vegetables, eat more whole grains, drink more water. When it came to foods of concern, they would not name them. They would say, consume foods that are lower in sugar, lower in saturated fat, lower in sodium, lower in calories. And the big gap that the NOVA classification came to fill was for practically anyone who gets to see the slides that were shown to explain what the vast majority of ultra-processed foods are, to understand what it is that we're talking about. So to me, it is very disconcerting that the 2025, 2030 dietary guidelines are still questioning the validity of NOVA and are still staying away from using the term ultra-processed foods. I served twice in the dietary guidelines for Americans committees, and both times we were not allowed to use the terms ultra-processed foods, and the saga continues. So I would argue, why don't we cross what you suggested, the ultra-processed foods that are also of concern because they are high in saturated fat, sugar, sodium, and call them by name and say, these are the foods going to this website and eat less of these foods. As a public health nutritionist, I would say that from a regulatory perspective, that's what I would recommend that the FDA does when it comes to trying to bring some order into this very, very serious and chaotic situation.
Thank you. Thank you. So we'll need to close here. I think Allison wanted to note.
Yes, I really appreciated your comment, Raphael, bringing in the issue around nutrition equity. So I wanted to take this time just to share a seminar that we are having next year, March, during National Nutrition Month, on ultra-processed foods and nutrition health disparities, and really talking more into the research that we need to help build the evidence base. And we're going to have Lauren O'Connor join us again, as well as Josie Mermate and Gerald Mand to have a discussion, and it will be held on March 12th of next year. So stay tuned for more information.
Great, thanks. Now, before we close, anything that is burning that people from our esteemed panel here, that any final words that you would like to share, or you feel complete? People are feeling complete. I love that. Well, please join me again in thanking our panel for this tremendous session. Thank you so much. So now we are going to have about a 15-minute break. We're due back here in the room at 2:40 to commence our next session. Thank you so much.
Our next portion of session 2. So good afternoon. I am Kelly Casville. I'm Senior Science Advisor for Nutrition in FDA's Human Foods Program, and I'll be moderating our next topic, which is on methodologies for incorporating nutrition science into risk analysis in food policy. So analyzing potential risks associated with exposures to food is really a key area of regulatory science for safeguarding the food supply, right? So foods can be a route of exposures to environmental contaminants like heavy metals and microbial contaminants like salmonella. And one of the most challenging aspects of environmental contaminants like these is that they're coming from the environment where foods grow and live. And if you ask experts what foods in these categories are common culprits, they will list things like produce, fruits and vegetables, other whole food commodities like grains and seafood, among other commodities. And so we just heard some great presentations about the topic of ultra-processed foods. Well, the foods I just listed are foods that are available as whole foods and are foundations in healthy eating patterns. So these issues are really complex. These foods provide essential nutrition and bioactive components that actually help to protect against hazardous exposures. And we know from the evidence supporting the dietary guidelines for Americans that eating patterns higher in nutrient-dense versions of these foods are associated with positive health. So in looking more holistically at food composition and potential associations of the exposures to negative, of these exposures to negative or positive health, it's really an essential area for the future of nutrition regulatory science. And so I wanted to highlight, just as a placeholder, a couple of existing NIH programs that do already provide some opportunities in this space. So the first one is the VICTOR program. It's at the National Institute for Environmental Health Sciences, or NIHS. And VICTOR stands for the Virtual Consortium for Translational Transdisciplinary Environmental Research. And it really focuses on bringing together diverse research teams to foster innovation and to accelerate the translation of scientific research into meaningful improvements in human health. Specific to the session that we're doing right now, Victor has a consortium on the topic of metal nutrient mixtures in epidemiologic and toxicological studies of cardiovascular disease, and one on microbial mechanisms of methylmercury metabolism in humans. And both of those consortia are examples of how NIH-funded research can help us better understand the multitude of mechanistic factors at play that influence how exposures through food ultimately influence health. So then we also need methods that bring together that evidence. And so the NIH Common Fund also has a program called Complement Animal Research and Experimentation. And I've heard that the acronym for that is complementary. And that program focuses on providing a range of ready and standardized biomedical research models for human-based new approach methodologies, or NAMs. And NAMs are lab or computer-based research approaches that are intended to more accurately model human biology. And so I'm really excited today to introduce our speakers to help us think about ways to integrate both the essential nutritive aspects of foods and the known hazards through our scientific methods. So our first speaker today is Dr. Sofia Santillana Farracos. She is the chief of the Exposure Assessment Branch in the Division of Risk Assessment and Prioritization in the Office of Surveillance Strategy and Risk Prioritization in FDA's Human Foods Program. And I know we're going to learn a lot today from her expertise related to scientific methodologies capable of incorporating nutrition science into risk analysis to support food policy. And our second speaker is Dr. Kasia Cordes, Associate Professor in the Department of Epidemiology and Environmental Health at University at Buffalo, where her interdisciplinary research in nutritional sciences, environmental health, and global health is exemplary for the topic that we're discussing today. I also want to acknowledge Dr. Allison Brown, who is our virtual moderator, and online attendees. You can feel free to ask questions through the Zoom, and we'll take questions both virtually and in the room after the presentations. So with that, please join me in welcoming Dr. Santillana Farracos.
Thank you. Thank you. And thank you for the introduction, Kelly, and thank you for the invitation today. As Kelly said, my name is Sofia Santillana Farracos, and I am the branch chief of the Exposure Assessment Branch of the Office of Surveillance Strategy and Risk Prioritization, and we're still getting used to these names, in the Human Foods Program at FDA. And I'm very happy to be here today to talk about methodology that can potentially be used to incorporate nutrition into risk analysis using what we call risk-benefit assessment frameworks. So this afternoon, I will be talking about risk analysis, risk-benefit analysis, how those differ, risk assessment, as well as risk-benefit assessment, and also how those differ. I will also be talking about an FDA-published risk-benefit assessment evidence scan that was published in 2024. And I will also be providing examples of other published risk-benefit assessments that were published in the literature to provide examples of how that those type of frameworks can be used. I will also be discussing other international efforts that have been, vocal and creating more awareness internationally around this topic in the last year, and then touch upon some data gaps for further research to consider. With that, I'll get started on risk analysis and risk-benefit analysis and how they are similar and how they differ. So for those of you who are not familiar with this framework concept, it is a concept that started, at least for food safety, in the early 90s. And it has three main components to it. It's risk assessment, risk management, and risk communication. And it was really developed to help decision makers, make a decision, because as has been alluded to in multiple talks today, there are many components to a decision-making process. And so it's not an easy process, and you have to incorporate various disciplines and various topics, and there's controversy and there's risks and balances and things that need to be considered. And so for food safety per se, they established this framework that's based, I think, in an economics, originally from an economics framework. So risk assessment is more of the science base, is the assessment to inform the decision, and the risk management is more the policy based. What helps inform the decision per se. Those two components of risk analysis are carried out by two different groups of people. So the risk assessors carry the risk assessment and interact with risk managers, which are the people who are making the decision. Once the decision is made, it's usually communicated to the stakeholders, and that's the risk communicators are taking part of that. So it is those three pieces to this puzzle. Now, similar to risk analysis comes risk benefit analysis. And the difference and the similarity between it is that it still considers those three components. So it still has risk assessment, risk management, and risk communication, but it also incorporates the nutritional risks and benefits to food consumption and not just the risks. So with that, I wanted to dive in a little bit into risk assessment and what it takes to actually do a risk assessment. And so risk assessment is composed of 4 different phases. The first phase is the hazard identification, which where you identify the hazard in this case, because we're only looking at risks. So you identify the hazard, you describe the hazard, you describe the host, the food, the outcome associated with the consumption of that contaminated food. And for that, you usually need information on contamination prevalence, you need information on contamination levels, and you need information on bioavailability of that contaminant in the food. The second step is exposure assessment, which takes on from the first step. So they're all interrelated. Once you do your exposure assessment, you estimate the level of the hazard in that food consumed. So for that step, you would need information on the number of consumers, you need information on the amount consumed, and you need information on the body weight of the consumers that you're looking, on to assess exposure on. Once you've completed your exposure assessment, you characterize your hazard. And for that, you need to establish a relationship between that dose that you calculate in exposure assessment phase and what's the frequency of illness or the adverse health outcome you're looking at. So kind of like the dose response as we know it. And so for that phase, you need information on the length of exposure. You need information on a dose response model. And then you can characterize, for example, illnesses in terms of DALYs. And and I think it was alluded to this morning, the concept of a DALY is a disability adjusted life year. So it kind of gives you a single metric for different diseases where you are able to put things in perspective in terms of, what's the DALYs for cancer versus kidney disease. And then you can make a comparison with 2 different health outcomes. For risk characterization would be the last step where everything's incorporating together, and that gives you the probability of occurrence of the adverse effect from exposure, including uncertainties. And for risk calculations, you would need information on all of those data streams that I mentioned in the earlier phases. So when we think about a risk-benefit assessment, we're thinking about the same phases. We're thinking about the identification of the adverse and beneficial health effects. We're thinking about exposure assessment from both the adverse and the beneficial effects. We're thinking about characterizing the adverse and the beneficial effects. And then we are characterizing both the risks and the benefits. And these risk-benefit assessment have 2 independent streams of evidence. So it's not like you're identifying adverse and beneficial effects on a same single metric from the start, but you do have those 2 independent lines of work where your benefit and risk characterization are happening. And then in the final phase, you're integrating both those risks and benefits to get a single outcome. So in risk benefit assessment, you are integrating health evidence from assessment of nutritional benefits and risks. And so that is slightly different from the traditional risk assessment. It is a stepwise approach, similar to risk assessment, and it can be qualitative, it can be semi-quantitative, or it can be quantitative, depending on the level of complexity that you need to answer your question. And so before I dive into examples of those qualitative, semi-quantitative, and quantitative examples of risk-benefit assessments that are published in the literature, I did want to allude to our own work on the risk-benefit assessment seafood evidence scan that we published in 2024. And this was a tremendous amount of work that involved multiple people. There is a big difference between an evidence scan and systematic review, which is more a traditional systematic review of the literature. It follows along the lines of the systematic review in terms of that we are searching the literature, but we're gathering information that are relevant for relevant evidence on the topic, but we don't dive in to analyzing the data. And so for what we did for the evidence scan, we designed and conducted a search on risk-benefit assessment frameworks that were already published in the literature, and we leveraged publications that had already done systematic reviews on risk-benefit assessment for seafood. And so our search date started in 2019. We screened about 1,800 abstracts just from 2019 to 2023. From 2019 to 2023. We had 2 reviewers on the screening and 2 tiebreakers for that. And then we designed a protocol to extract data from the identified relevant studies, which were 83. We published the manuscript and our findings. And as you see on the screen, that's the paper that we published. This information that we collected through the seafood evidence scan was used to inform the National Academy Committee's questions that we had in a seafood child development effort that we were doing in collaboration with the National Academy. So in terms of examples of qualitative, semi-quantitative, and quantitative RBAs, if we want to call it like that, I'll start with a qualitative, which is, from a technical perspective, the simplest. In a qualitative risk-benefit assessment, one would just conduct a systematic review of the literature and look at background studies on hazards, nutrients, and health outcomes. An example of this is the Bernstein 2019 paper that I'm showing on the screen for fish, shellfish, and children's health. Based on their systematic review and background studies on hazards, nutrients, and health outcomes, they concluded that although some potential toxicants may be more likely to come from consumption of certain forms of fish, most fish, with some notable exceptions, have favorable nutritional overall health qualities compared with other forms of animal protein. So this was a conclusion that that group of authors made based on looking at both hazards, nutrients, and health outcomes, without quantifying the risks or the benefits in using their studies. A semi-quantitative RBA by nature is a little more complex from a technical perspective. So it's a more refined assessment and includes exposure estimates. And they typically, what they do is they do the exposure assessment estimates and then compare it to a health-based guidance value. And so an example of this one is the one shown on the screen, which was conducted with data from Central Italy, also in shellfish. And what the authors did was they looked at the levels of cadmium, mercury, and lead, and also looked at the levels of omega-3 fatty acids in the selected seafood species. And then they compared both the risks and the benefits from that shellfish consumption using a benefit risk ratio. To add more complexity to it, the quantitative RBA is integrating both risks and benefits into a single health metric, like, for example, a DALY, as I mentioned earlier. So for this one, we have 2 examples here. One is one published from FDA in June 2014, which is the quantitative assessment of the net effects of fetal neurodevelopment from eating commercial fish. And that was using a single health outcome because they were looking particularly at neurodevelopment in children. And so the health outcome was IQ. And the net effect, as they calculated, was the IQ loss from methylmercury summed to the IQ gain from the nutrient package of seafood. And so that is a single health outcome that they were looking at, and it was a single health metric that they were able to quantify. And I put it the summary here, and it seems like a very simple assessment, but it's a very complicated technical process to do this, including dose response, exposure assessment, et cetera. On the right side of the slide, you can see another example, and that's an example of a substitution. In that case, this was for the Danish population, and what they did was they looked at different scenarios of what would the DALY difference be? And that's again, the disability adjusted life years. If you would change your consumption of fish, of processed meat for fish, in different quantities. And so they have those 4 scenarios there, and you can see the change in the DALYs when they change the diet. And so in this case, though, instead of looking at a single health outcome like I described for the net effects assessment that FDA conducted in 2014, they were looking at multiple health outcomes, because in this case, they were looking at methylmercury, they were looking at dioxins and dioxin-like PCBs, and they were looking at omega-3 fatty acids. So just by the nature of these types of assessments, these are much more complicated than a qualitative risk-benefit assessment that was presented earlier. International efforts that are currently either published or underway are listed here, some. One is the FAO, the Food and Agriculture Organization of the UN, published a background document this year to provide scientific evidence about the risks and the benefits of food consumption, to update their 2010 report on the joint FAO-WHO expert consultation on risks and benefits of fish consumption. So this is, again, if you think about what I talked about earlier in the Bernstein 2019 qualitative RBA, this would serve as a qualitative risk-benefit framework that a decision maker could use to inform their decision. So that is one example background document that was recently published. In addition to FAO, the European Food Safety Authority convened a committee, and they just in 2024 published a revised guidance on risk-benefit assessment of foods. Their update addresses methodological developments and regulatory needs, and it retains that stepwise approach that I presented earlier. And it provides additional methods for complex assessments, such as multiple chemical hazards and all relevant health effects impacting different population subgroups. That was also published in 2024. In addition to that, the WHO had made a call to for experts to develop WHO food-based dietary guidelines on the optimal intake of animal source foods. And in that process, I think for the first time, the guideline development group will be producing a guideline containing recommendations and contextual information to aid policymakers in relevant decision-making, but they will use, as they portray on their website, evidence gathered and reviewed, and models developed by a risk-benefit assessment technical group. And so these are some of examples of things that are going on in other international organizations that also allude to the potential use of these type of frameworks to inform decision-making. And all of this information is publicly available online. So if we think about what I was showing earlier in terms of the data needs for some of these assessments, and we think about the risk-benefit assessment framework that I also presented in the earlier slides, some examples of data gaps that could be further developed to increase our knowledge and be able to run these assessments more steadily is, for example, bioavailability. So we are not yet at a place where we're comfortable, with our knowledge in terms of how much of the, for example, contaminant from a food is available for the human body absorption. So that is one example of data that could be further collected. Another example of areas of uncertainty is dose response models. I mean, we don't have dose response models for all the chemicals that are in the food supply. So it's really hard to make that connection between the dose and the illness. And so furthering those would be really helpful in terms of, moving these frameworks forward. And then in terms of the DALYs, as I mentioned earlier, it's also very difficult to establish those disability weights for these different diseases. And so there's a lot of uncertainty in that area. And thus that would be another area that, would benefit from more information. And with that, I wanted to acknowledge the team that participated in FDA's evidence scan, including Kelly. And so that would be Jacqueline Hillman, Eileen Abt, and Sherry Dennis. And then I would like to thank Kelly and Robin for inviting me today.
Good afternoon, everyone. I also want to thank Kelly for the invitation. It's been a great learning experience listening to everybody's talks so far, and I'm happy to add to the conversation as well. So I'll continue by considering nutrition to understand the health risks from dietary exposure to heavy metals. We already know that there is this duality that comes with eating foods. We eat foods because they provide us with health and life-sustaining nutrients, but they may also contain toxins or toxicants such as heavy metals. So that gets us on the same page. And there's really good evidence of this from the FDA's own studies, such as the Total Diet Study. The latest one was done between the period of 2018 and 2020, and it analyzed heavy metals in 307 foods. And if we look at the heavy metals and metalloids that I list over there on the left, and the percent of foods in which they were detected, that ranges from 7 to 61%. So it's there is not a simple solution where we can say, well, let's eliminate those foods because then we might not be eating anything. So we need to be thinking about how do we think about the risk to the US population from consuming foods that may be containing heavy metals or metalloids. And in particular, we worry about young children for whom these metals are known in a variety of studies to be related to developmental risks. We don't know whether the heavy metals from foods specifically carry developmental risks. We know about developmental risks more broadly from environmental exposures. And so again, an important question here is if young children consume these foods, what kind of a risk does this carry? And then of course we need to think about how does nutrition that comes in these foods also play a role? And nutrition, I say in quotation marks, because obviously nutrition is a wide term. So I will walk us through an example because there's 4 metals or metalloids that are listed there, and there's not enough time to discuss all 4 of them. I will walk through an example of lead. And a couple of things to get us all oriented. Lead is measured in foods in units that are either parts per billion, which would be equivalent to nanogram per gram, or parts per million, which would be equivalent to microgram per gram. ppb is smaller. The FDA also has guidelines that have been released to tell us or guide, how much exposure may be tolerable. And for children, it is 2.2 micrograms over the course of a day, or 2,200 nanograms per day. There's also guidelines for females of childbearing age. And then there are also now released almost 2 years ago, draft action levels for lead in processed baby foods. Such action levels don't exist for foods more broadly. They, for now, focus on baby foods as well as fruit, apple juice or fruit juices and so on. But for the most part, these are what exists. And they exist for single ingredient vegetables or single ingredient baby foods, maybe some mixtures. There's a separate guideline for root vegetables and then dry cereals. Okay, so in addition to walking through the example of lead, I will also start with spinach. Spinach is one of the foods that we probably in dietary guidelines for Americans would consider to be a food that's recommended for us to consume because it is considered a nutrient-rich food. It is a leafy green. It is a vegetable more broadly. It could be selected if we chose to follow a nutrient-dense or a healthy diet pattern. And it could be selected if we chose to follow dietary recommendations for Americans for a healthy diet. So one way of thinking about heavy metals or metalloids in foods is to just take a very straightforward approach. Let's measure what's there and think about whether that may be a concern or not. And this is certainly something that we were already doing. And so when we think about a typical serving of spinach being around 30 to 60 grams, lead through the total diet study has been measured in spinach, and a mean concentration has been found of 6.4 parts per billion. So if we think about a single serving of spinach, that's 30 grams, we would likely consume around 192 parts per billion of lead in that single serving. Now, the maximum concentration of lead that was found in spinach in the total diet study was 18 parts per billion. And so if we again consumed a serving of spinach with that maximum level, we would be consuming around 540 parts per billion. So the mean concentration is below the FDA draft action level for baby foods, but the maximum value is above a draft action level for baby foods. And so again, right there, we have some criteria that we can say, well, if we have a mean value, we're probably okay. If we have a maximum value, then that's in a baby food, then that would not be okay. Both, however, in a single serving of spinach are well below the FDA guideline for daily intake of lead, well below. And so we would probably be okay if no other foods had lead in them. But we can also take a more nuanced approach and actually consider what the nutritional factors are. So let's start with nutrients. Going back to the total diet study, fortunately, we have other information that was also measured in that same spinach. So we have other metals, nutritional metals, minerals, such as calcium, copper, iron, magnesium, potassium, and zinc. There's more, but these fit on my slide. So again, we can look at the mean concentration for these nutritional metals that were reported in that same spinach where lead was measured, as well as maximum values. And we can calculate, taking these mean or maximum values for all of these minerals, what are, per 30 gram serving of spinach, how much of these things are we consuming? And even if we look at, let's just take a look at the mean amount of lead per 30 gram of 30 gram of spinach, it may appear high, but the amount of nutrients that we're getting is like orders of magnitude higher. So is that a good thing or a bad thing? Well, let's hope it's a good thing. And we can also think about the ratio of nutrients to the contaminants that we're receiving. And they're very, very much higher in terms of the amount of nutrients that we're receiving versus the level of contaminant. There are no studies, so I don't know whether this is, again, is it a good thing or a bad thing? I don't know, because one of the major points to take away from this presentation is that there's so little research that has been done on these issues that we're sort of still groping and thinking about, how do we interpret this and what does it mean? And so this is just one way of looking at it, and hopefully that will provide some answers. Since I already mentioned, and I'm sorry, I guess I should go back and say that I chose these specific minerals or metals because they are known to compete with lead for absorption in the intestinal epithelium. And so that's a good segue to start thinking about bioavailability. And Sophia already mentioned that is one critical gap where we don't understand how much these heavy metals or metalloids are available to the gut for absorption when they are part of a food matrix. Lead has no physiological reason to be in our bodies. It has no practical purpose, and therefore it does not have a dedicated transporter. What it does is it's an opportunist, and it uses transporters that have evolved for other metals, such as iron or minerals, such as calcium, to get into the body. And so it will use these transporters to get in, as I said, iron or calcium, but it also uses passive transport. So this is important for trying to understand bioavailability of lead or other heavy metals as they get absorbed in our bodies. So we actually have very little research on bioavailability. Mostly it was done in adult men in around the 1970s and 80s. It was typically done not as part of a food. It was done as how much lead is absorbed in a bolus, and it may be accompanied by food or it might not be accompanied by food. But that's not the same as answering if lead is inside a food, how bioavailable will it be for absorption? So we know some things from that research, but we need to understand more. So we know that fasting increases lead absorption, and because eating is an episodic event, I mean, we think about diets throughout the day, but there are very discrete eating episodes. And so we need to understand, we need to think about foods and diet patterns also within the context of eating episodes. We know that bioavailability is affected by other metals, as I've already mentioned, phosphate, carbonate ligands, fiber, maybe. But again, we need to understand whether the food matrix itself could affect metal bioavailability. So going back to spinach, there has been some research in using Keiko-2 cells. So this is an in vitro cell model that has looked at iron bioavailability from spinach, and this spinach is introduced to a Keiko-2 cell model, and the percent absorbed is calculated, and this was done when spinach was introduced to this model by itself or with ascorbic acid. Ascorbic acid is a really important way to enhance the absorption of iron. And if you look at the column that I've highlighted, iron bioavailability of in spinach or from spinach is really low. And so it gives us perhaps a clue that lead bioavailability from spinach would also be low. Do we know this for sure? No, we don't have any data to show us this. But what it shows is that we have some ways and ready tools to be able to try to address those questions. So back from the segue of bioavailability, I want to go back to spinach as a part of a food group or a diet pattern. We do have some research, epidemiological research or population-based research in using the NHANES, and that goes back to the point that I believe Susan Main made earlier, that NHANES can be a really important tool for us to answer some of these questions that we're grappling with right now. So together with colleagues, we looked at the consumption of different food groups in relation to blood lead levels of young children in the NHANES. And there's multiple results here, but I'm just pointing you to the ones that are circled in red. So when we compare to children who had no intake of vegetables, and that's the blue box, and we look at the children in the highest tertile of vegetable consumption, so that would be that red line, the children who consume more vegetables have slightly lower blood lead levels. So maybe consuming more vegetables, even if they contain potentially lead or other heavy metals, maybe it doesn't impact on children's blood lead concentrations. This is a study of an N of 1. We need more research with young children to be able to answer these questions. In another setting, and with school children, my colleagues and I looked at the intake or the consumption of different food patterns. And here we're looking at food patterns that are characterized by higher intake of dark leafy vegetables, red orange vegetables, and beans and peas, as well as other foods. And we do see on the left-hand plot that higher scores with this pattern are associated with slightly higher blood lead concentrations. So that's a little contrary to what I just showed you in US children who are younger. But it points to a really important gap in research that we need to be looking at these questions in multiple settings, because there are different contamination scenarios in different settings, as well as different food intake patterns or preferences across different settings. So if we want to create guidelines for Americans, and Americans are a very diverse group of people, then we need to be looking at these questions in multiple settings. Another point that I want to make with this slide is that the same dietary pattern is actually associated with lower concentrations in children's urine of inorganic arsenic and higher concentrations of dimethylated arsenic. So the same pattern may look like it's associated with worse exposure to one metal, but actually better exposure or more beneficial for the exposure of another metal. So this is a really complicated picture, and we cannot treat these questions on a metal by metal basis, because it won't work. If we make a recommendation based on one metal, we may have unintended consequences for another. And again, we've already heard about unintended consequences as being an important part of this whole picture that we need to look at. So another way that we've been looking at this is to think about dietary recommendations. So if we look at diet scores on variety, adequacy, moderation, and an overall diet quality index, we also see that there's either very tiny associations or null associations with children's blood lead concentrations. So again, maybe when children eat an overall healthy diet, that contaminant maybe doesn't matter. Do we know this for sure? No, we don't, because we don't have a whole lot of research on this. Another study from another setting, this is in Spain, in preschool children, looked at children's adherence to the Mediterranean diet and the concentrations of metals in their urine. And for the most part, whether you're moving across quartiles of adherence to this diet pattern, there are no associations with the metals that we would be concerned with, such as lead, cadmium. There is a slight association with arsenobetane, but overall there is no association with inorganic arsenic. And we see the same thing for vegetables, since we've been talking about spinach, that higher adherence to a better vegetable intake within this Mediterranean diet study does not seem to be associated with higher exposure to metals. So again, perhaps healthy diets, as long as, as long as children adhere to healthy diets, maybe contamination isn't so important for their exposure. Again, it's two studies. We need more across different settings, and particularly in the United States, to be able to answer these questions. So I want to close with some research needs. Here I'm including this figure from a paper that my colleagues and I published about 2 years ago on research and methodology and data needs. This is really complicated to go from an exposure in the diet all the way to understanding health effects. There's lots of things that we need to understand, including the nutritional status of children, whether these metals or metalloids are actually affecting the internal dose of exposure. So looking at biomarkers, we don't have answers for many of these questions. And so I'll just run through a few really quickly. The total diet study is great. It tells us lots of things, but it's done on 307 foods. And I think that we need to measure heavy metals more often and in a larger set of foods to understand what's going on in the US food supply and how that may be affecting the US population. We're based on this information and, for example, using dietary intake data from the NHANES, we would be able to estimate dietary exposure at the population level and then test their associations with biomarkers. We need to better understand and account for bioavailability of lead from different foods and investigate the extent to which the nutrient intake, diet patterns, as well as eating occasions would affect biomarkers of metals. We also need to think about how these diet patterns, as well as eating occasion, or I'm sorry, food groups or diet patterns could moderate the effects of metals and especially metal mixtures on health outcomes. I already talked about studies across different settings to understand the variability in exposure, and then this exposure biomarker health relationship across sociocultural patterning of dietary intakes and exposure scenarios. We already talked a little bit about, and so I will echo for the need to establish research or collaborative centers on food contaminants and health, and then fund research that addresses questions that regulators actually want answered to support the development of guidelines. Thank you.
Wonderful, thank you. So we're going to our Q&A session for today, and we will start at the podium with Rafael.
This may have already been done, but is there enough evidence out there for the FDA to select a group of environmental contaminants, heavy metals, persistent organic pollutants, metabolic disruptors, and on, that they could mandate all the food companies to measure in all of their products and to report them as part of the food label? For that, I understand we do need to have thresholds. We have to have the research on what is the limit for the consumers to know if they are below or above or not. And I ask this because across the board, consumers are extraordinarily concerned about what on earth is on the foods that they are consuming.
I think I can allude a little bit to what you are mentioning. I don't know, Kasia, if you want to add anything after, that's fine. So we at FDA do have a list of chemicals. For example, if you go on our website, you can find a list of chemicals that we currently are evaluating and have initiatives to address them. That includes heavy metals. So if we have the Closer to Zero initiative for children, we have the PFAS, as you can find all of those on the website. So if that is helpful, you know, for a consumer piece to understand what the risks are with those particular chemicals that we have some information on, it is on our website. I don't know if there was anything particular about Kasia's presentation that you were asking.
Well, is there enough justification or knowledge to mandate the food companies to report the levels of a specific list of environmental contaminants in the food labels?
I think that's a really good question. I believe for metals, for heavy metals, for sure. And there are already things afoot where food companies that produce baby foods will have to, starting in January of 2025, will have to make that reporting. But I do agree with you that the sort of the interpretation of what they are reporting requires a little bit more in terms of sort of threshold or guidelines and what's appropriate or not. So I think they can certainly be evaluated against what already exists with the draft guidance, and I will let Kelly maybe speak a little bit to that. But for sure, for heavy metals and other foods, potentially PFAS and other chemicals as well. Where I think it gets really complicated is that there's increasing evidence of sort of contact materials, so packaging materials, also releasing lots of chemicals into the foods. And so the question is like, where do you stop? Where does this stop? And how would an average consumer interpret those data? But for metals, I think that things are moving in the direction that you're suggesting.
Yeah, I just wanted to add to that, one of the big challenges in this space is that in agricultural commodities, there's a very large amount of variation for metals. Even in one field of sweet potatoes, you may have very different level of a sweet potato that comes from one side of the field to the other side of the field. So, thinking about mean levels and looking at variation, we need a lot more data on variation. And we need a lot more information about the composition of other metals in relationship to the metal of interest and the nutrients as well, because this is Mother Nature, it works in a balance. And to really understand the correlations, in my opinion, between the nutrients and the contaminants in foods is really where we're going to learn the most. And looking just solely at one nutrient, or we always know that's an issue, and it's the same with metals. If you're looking at one contaminant outside the larger scope for a specific food or even diets and dietary patterns, you're going to come up with a very different perspective on what decision you might make, rather than looking more broadly on what food exposures at large look like and a more holistic approach.
Hi, Susan Main, Yale School of Public Health. Just amplifying what Kelly just highlighted. My concern, and I am familiar with what California is doing, but my concern is that consumers don't know how to interpret it, and the unintended consequences could actually cause more public health harm than benefit. And so when baby food companies are making the jars of baby food that I fed my children, things like jarred sweet potatoes and spinach and things like that, those vegetables are some of the highest in some of the minerals that we're concerned about here. And so what we're going to see is those ingredients will not be used by the baby food industry as much. And so you're losing all of those phytochemicals and all those good things in those foods because consumers are fixated on one particular toxin at a time. And that's true with the mixed products, things like that. And so I'm just very concerned with the entire focus in on lead or cadmium or whatever. We're going to lose the benefit side of the equation. And that's why this session, I think, is so critically important, because what I don't want to see is the baby food industry move to some of the fruits and vegetables and grains that maybe aren't as, don't have as much nutrient density, but they may be lower in selected toxic elements. And so that's my big concern by notifying consumers. So how do we work with consumers to help them empower them with information will help them make the best choices? One of the things we saw when I was at FDA is consumers would say, well, look, this baby food product had lead in it. They are adding lead into their baby food, so I'm going to go make it myself. And what we find is that the baby food industry, they're sourcing the products with the lowest levels they can possibly find. So if consumers make it themselves, they may be inadvertently exposing their infants and their babies to higher levels. So this stuff is tricky, and that's why I think the benefit side of it is really, really important to think about in terms of considering how to move forward.
Thank you. You want to take the one-on-one?
Yeah, so we received a couple questions online, and this one related to the risk-benefit assessment. And Sophia, your presentation. So this virtual attendee commented that nutritional benefits need to focus on the complexity of nutrients in food rather than honing in on one single nutrient. So they ask, can this be accomplished by considering a risk-risk analysis? For example, the risk of consuming fish compared to the risk of not consuming fish.
Yeah, and thanks for the question. I think that's a very interesting point, and I don't know if I didn't explain myself correctly, but I don't think a risk-benefit assessment is just one nutrient. You can include 100 nutrients if you want. It's just a framework where you can balance both risks and benefits. But thinking about a risk, and we have a publication on a risk assessment that we did for infant cereal. That's a good way to think about it. You can think of a benefit as a lack of a risk, and as a risk as a lack of a benefit. And so yes, you can do those comparisons, but the reality is that there is some intake in your diet of protein, So for example, if you stop eating fish, you are going to substitute that protein with some other food product. And so if you think about the risk, you can definitely do those analysis. But if you're not going to eat fish, you're going to eat something else other than fish. Mhm.
Maybe I'll just add a couple of points. So just going back to what we saw with spinach or other foods that were analyzed by in the Total Diet study, specifically for baby foods, I agree with your point that it's really important to understand the benefits of nutrients, because some of the foods that, for example, may have been higher in arsenic were also some of the highest in providing iron. And iron, especially for younger children who are going through, who are starting complementary feeding, who are going through those first couple of years of life, is one of the most important nutrients that is required to support children's, sort of proper cognitive development. And so, eliminating a certain food is like throwing the baby out with the bathwater. And so maybe thinking about that ratio of nutrients to contaminants is one way of thinking about it. Another way to think about it is that just because something is detectable in a food doesn't mean that it's harmful. And I think that we haven't been yet very strongly thinking about sort of communicating what it means. Like how do we explain what it means that, a heavy metal or metalloid was detected in food, at what level, and is it necessarily at a level that would be harmful? So yeah, I think that there's, I basically agree with what you said, and I think we need to be thinking about the communication piece as well as the sort of the more fundamental research to give us answers to our questions.
All right, we'll take another question from the room.
Lauren O'Connor, Texas A&M Evidence Center. So you both mentioned that there's a limited amount of research on bioavailability, and this is something that my research group has found as well with human milk and infant formula. And we've been struggling on how do you close that gap, because bioavailability studies in general are expensive. They're just huge studies. But then when you put in the contaminant component, you can't knowingly feed someone contaminated foods. So how do you suggest attempting to close that evidence gap on bioavailability of contaminants?
Yeah, the ethical issue is an important one here in this area for sure. So, aside from doing human feeding studies, I think that probably animal studies or in vitro types of methods would be the next thing that we could try. Will it give us the exact number that we can translate to humans? Perhaps not, but it gives us something that we don't even have currently. And so maybe that's the next step that we should start with. But I don't know what your thoughts are.
Yeah, no, I think it's a great point. It is a very complicated subject. But yes, I agree with what Kasia said in terms of the types of assessments that can be done. We can move from animal, but then comes the uncertainty of how do you move from an animal to a human, right? So there are always uncertainties, but you just have to, at least from the risk assessment and risk benefit assessment perspective, you can include them quantitatively if you have some sort of information, even if it's from an animal or from an in vitro study.
All right, I am going to take the opportunity to ask a question. Moderator's privilege, right? And I have a bunch of questions. I've made a whole page here, but I'm not going to get to ask all of them. Not today anyways. I can pick your brains at other times as well. But, in both of the presentations you gave and in the Q&As that we've heard as well, it seems like the importance of interdisciplinary research teams is just so central. And I wanted to see if each of you could just say a little bit about what those interdisciplinary teams might look like in the experience that each of you have had in your respective research experiences, and which aspects of those interdisciplinary teams that you might feel were missing or could be included in the future, or which ones are most definitely the most essential.
Yeah, I can get started. I think the field of risk assessment, and I'm not talking about risk benefit, but risk assessment per se is already interdisciplinary, right? Because, you know, you have the contamination levels, you have the prevalence, you have the modeling, you have the risk, and you have multiple disciplines included in that. And then you add the benefits, and then comes nutrition, right? And so you would need to add the nutritional expertise into that multidisciplinary team. And so but at the same time, though, I mean, it also depends on what the question is. So, because I think I heard in other sessions where people were talking about health equity and people were talking about economic issues. And so in that case, then it depends on what your question is and what you want to base your decision on, what type of expertise you would need.
Yeah, I mean, I would echo that. And some of the teams that I've worked on have consisted of nutritionists, toxicologists, exposure assessment experts, certainly child development. So it's a very large group of people who contributes to answering some of the questions or to the data that I've presented so far.
And we'll take one more last question from the room, and then we are going to move on to the next part of our
Thank you
meeting.
so much. My name is Jacqueline Fetzal Fryer. I'm a cancer prevention postdoctoral fellow at NCI. I was wondering if I could ask a two-part question. The first is sort of your distinction between a heavy metal contaminant that's naturally part of a food, such as you were talking about with spinach, how it's in the soil becomes part of the vegetable, versus, let's say, the water supply and old lead pipes, for example. Would we be just as concerned with consumption from contaminants that are naturally part of a food versus being leached into the food supply or the water supply, for example? What do we know about the bioavailability of the lead when it's consumed that way? And the second part is just related to mitigation strategies. So it's one thing to have companies try to put the levels on there, but if they're naturally in the food, we can't really minimize that. So is the main goal to try to fix the soil or try to control the soil so that it doesn't naturally end up in the spinach? Any thoughts you have on those would be helpful.
How many hours do you have? So your first question was, should we be as worried about food or let's say lead in food as lead in water? I think that's a really good question. I mean, obviously, if there's lead in water, and you're drinking the water by itself, not in addition to food, then there's nothing. Well, it doesn't all have to get absorbed because there's also issues of particle sizes that I don't think we have the time to get into. But yes, lead in water would be a major concern, and perhaps more so than lead in food, depending on how much you consume of each, depending on how much lead is in, what the concentration is. I mean, there's so many factors to sort of factor into that. But certainly we cook foods in water that may be contaminated. We prepare baby formula with water that may be contaminated. So if we think about water as part of a broader diet that we consume, then it's certainly something that we should be thinking about. In terms of mitigation strategies, oh my gosh, there's several that people are thinking about. I'm not an expert in this area, so I cannot talk about merits of one versus the other. But some of the things that people talk about are, sourcing foods from fields that have been testing fields for their levels of contaminants, doing different food processing. So, and there I just mean peeling, peeling, or maybe sort of for arsenic in rice, for example, it's soaking the rice in water, cooking it in large amounts of water. I mean, there's like a whole gamut of things that people have been thinking about. So, so it's both mitigating what already exists as well as preventing through field selection or testing the metals getting into foods. And again, I'm not an expert there, so there's other people who could discuss sort of merits of one versus the other.
All right, thank you. With that, help me in thanking Sophia and Kasia once more for the presentation. And now I'm going to turn the floor over to my colleague, Dr. Claudine Kavanaugh, for our next part of this session. Thank you.
Good afternoon, everyone. I'm Claudine Kavanaugh, and I'm work at FDA in nutrition and food labeling. So we're on our final panel session today, looking at the impact analysis of an implementation science related to nutrition regulatory action. So just to give you a really quick background, part of FDA's regulatory process is to draft regulations. And one of the requirements for that is actually to do a regulatory impact analysis that evaluates the cost and benefit analysis. Of course, with any of our regulatory actions, we also want to be informed by our policies and see what impact they have, because that can help inform our future policies and any changes that we may make. So measuring the impact is really important, but as we'll see, it can be challenging by a multitude of factors. So today we have two really distinguished speakers today. The first is Dr. William Hallman. He's a distinguished professor from the Department of Ecology at Rutgers University, and he's going to be outlining systems approach to evaluating the impacts of labels. And our second speaker is Dr. Jason Lusk. He's the Vice President and Dean of Agriculture from Oklahoma State University, and he's going to cover some of the credible and trustworthy estimates of consumer-related policy impacts. So I'm going to turn it over to Dr. Hallman, and I do want to note that we're going to start off our questions at the end online to make sure the participants online really get an opportunity to ask some questions. Thanks.
So while he's doing that, I'll introduce myself. I'm Dr. Bill Hallman. I am a distinguished professor at Rutgers, and the Board of Governors would like me to know that that is the State University of New Jersey. My dean would also like you to know that part of my line is Rutgers Cooperative Extension. So ladies and gentlemen, these are your tax dollars at work.
All right, thanks very much. And start my timer. Start. Okay, so I am a psychologist and an expert in risk perception and risk communication to follow the risk analysis group. And one of my specialties is looking at how people think about foods and the risks and benefits associated with foods. And so today my talk is going to probably confirm some things you already know, provide some evidence for some things that you suspect, and perhaps introduce some new things that we should probably talk about. So for those of you who are watching this in the recorded view, you may want to go back to speed 1, because I've got a lot of things to say. All right, so let's begin with, we need to understand the context of food labels and remind ourselves about the problem of invisibility and why we label in the first place. Frankly, most of the foodborne risks we want to avoid are invisible, including things like bacteria, viruses, parasites, toxins, and allergens. But so too are most of the things that we want out of our foods, including micro and macronutrients, taste, calories, and lots of other things, including production methods. So we rely on trusted labeling information to make the invisible visible. That's what labels do. And consumers need to trust us to give them the information that they need. And so they rely on things like ingredients lists and nutrition facts and content claims and lots of other things that we regulate and need to talk about. The second thing we need to come to grips with is, what is the label meant to achieve? And you go, well, obviously it's to inform people, but frankly, there are more implicit goals beyond simply informing people. We inform people, but we expect people to take action based on that information. So what do we want to do? Do we want to simply inform them or warn them, for example, that an allergen exists, and simply let people make up their own minds about what it is that they should do with that information? Or are we trying to educate people? Are we trying to do structure function claims and say, products that are high in calcium may in fact prevent osteoporosis? Or are we trying to persuade people to do something new, to purchase something new, or for God's sake, to stop doing something, which is to try to get people to stop eating so much sugar and sodium and so forth? Or are we trying to motivate people to do what they already know what to do? They already know that they should be doing, for example. We need to be clear about what our implicit goals are in providing the information that we have on a label. I will note, for example, that it's called the nutrition facts label, not the nutrition advice label. Getting back to compelled speech and First Amendment rights. So it's truthful and not misleading, but it's not necessarily telling you what to do. Now, so in understanding, well, what is our goal? Frankly, we're giving people nutrient information so that they can alter their diets appropriately. And in order to do that, we need to understand people's starting points. How do they interpret this information? And as a psychologist, I start with, how do we figure out what the intuitive meaning is of this information? What do people think it means? Not what does it really mean, what do they think it means? And frankly, what prior information or prior knowledge do they have to appropriately interpret that information? What's the distribution of that knowledge? How many people have it? And what's the likelihood of misinterpretation of the information without that particular knowledge? And frankly, what additional explanation do we need to provide people so that they can put this new information into context? Now, I do mixed method research. I will argue that qualitative research is needed. It's typically where I begin my work. So I do something called mental modeling interviews, key informant interviews, for example, where I will sit down with a single person and make them nuts by asking them repeated questions. So tell me more about that. Tell me more about that. So this is an example of going through the nutrition facts label with someone who is not necessarily really knowledgeable about nutrition. And I'd like to point out at this particular juncture that those of you in this room and those of you watching online are not normal human beings, right? You know what this means. Most normal human beings have no idea what the nutrition facts label information really implies. So here's an example very quickly. So if you ask people, well, which of these things should they increase or decrease? Going through an interview like this, well, I know that fat is a bad thing, so I probably should reduce fat. And I guess that saturated fat is one of the bad ones because it's under fat. But there's no entry here under trans fats for percent daily value. So what does that mean? Does that mean I shouldn't have any? Okay, well, that's what I'm going to interpret. I know that carbs are a bad thing because there are low carb diets and I want to lose fat. But dietary fiber is under carbohydrates. Does that make dietary fiber a bad thing? You laugh, but these are normal human beings, right? I know that sugar is definitely a bad thing. Then we get to protein. I know that protein is healthy, but there is no entry, again, here, 4% DV. What does that mean? And so on. And, when you get to vitamins, is going over 100% a good thing or a bad thing? We don't separate the nutrition facts into the things we want people to have more of and the things we want to have them, have less of. And we don't really provide any context for any of the vitamins, some of which we would like people to maximize, and some of them we don't want them to go overboard on. So your mantra for today is mental models matter. Mental models matter. Mental models matter. The deal is we can't know everything. We often have incomplete mental models of many of the things that we care about. And so in order to navigate the world, we fill in our mental models with available information, and I would argue misinformation, that simply is plausible and connects in a simple way with what we think we already know. And the question then we have is, well, how do people integrate labeling information into their existing mental models? And how can we facilitate that? How can we make it easier for them to take correct information and insert it and help them complete what it is that they think they know? So here's an example. Some of the work I did with my colleague Amanda Bearhaupt, who's now at FDA actually, on qualified health claims. So one of the things that we did is we asked a representative sample of 1,335 participants, 55 and older, so these are older adults, their degree of familiarity with several diet-disease relationships. And here's a list. Some of these are real, some of these not so much. Carrots and eyesight, for example, is a popular one that many people believe is true and perhaps not so much. So you can see that 91% of Americans are familiar, for example, with the relationship between cranberry juice and urinary tract infections. I've highlighted in green nuts, omega-3s, and walnuts. About 80% of Americans, about 8 in 10, are familiar with this relationship between eating nuts and omega-3s and heart health. This is the FDA sample statement, approved sample statement for the qualified health claim for walnuts. Supportive but not conclusive research shows that eating 1.5 ounces per day of walnuts as part of a low saturated fat and low cholesterol diet, not resulting in increased caloric intake, may reduce the risk of coronary heart disease. See nutrition information for fat and calorie content. Well, that's helpful. So helpful that in reality, the California Walnut Commission does not use the qualified health claim. They simply advertise based on their nutrient content claim. Because people already know the relationship between omega-3s and walnuts and heart health, they don't have to say this complicated stuff. And so you don't get any of the disclaimer information. So we have to look at how things are actually used in reality. We know that labeling information exists within a broader context. I'm so sick of hearing this. Do your own research. Lots of consumers do their own research. And unfortunately, a lot of the advice that's given is inconsistent with the best available science. Look at the nutrition information that's available on TikTok. Better yet, wait till cocktail hour, because you may need a drink after you see some of it. So in addition to doing their own research online, people are also increasingly turning to technology. And one of my students, Telema Briggs, published his dissertation looking at commercially available diet and nutrition apps and looking at the results of what you enter based on recommended adult guidelines for adult weight management. And he generously concluded that these diet and nutrition apps offer limited adherence to expert guidelines. Many of them are really pretty bad because they are designed by programmers, not designed by nutrition experts. So we need to be aware of what additional information people are going to in order to interpret labeling information. We need to see the broader context if we're going to take a systematic approach. Labeling information obviously also needs to be relevant to real-life practices, which includes evaluating the interpretation of graphics, not just the labeling information. So here, I just picked this at random. This is the Simply granola box. Want you to focus on is the cereal bowl. And frankly, my granola does not float. So how much of a serving is this? Actually, there is clearly milk there. The serving size is on the nutrition facts label, 2/3 of a cup, which is about 5 1/3 ounces. This is one of the most popular cereal bowls sold on Amazon. It is a 30-ounce cereal bowl that holds 28 ounces. I'm familiar with this because this is the cereal bowl I have at home. And so when I Googled most popular cereal bowl, it went, you bought this in November of 2022. And what I want to point out is that Consumer Reports did a study of this in 2014, where they asked people to just pour the normal amount of cereal that they eat into various size bowls. And not surprisingly, the larger bowls resulted in people pouring in 2 and 3 times the recommended serving size. So we need to be realistic about kind of how people are judging the amount of cereal should go in their bowl. And on the right-hand side, in the upper right-hand corner, is the advertisement for this bowl showing cereal up to about the 28-ounce serving size. So we need to have a holistic view of what people are actually doing, real people, not us. We also need to evaluate before and after implementation. So we looked at the National Bioengineered Food Disclosure Standard. For those of you not familiar with this, it was intended to provide greater transparency about which products are or contain bioengineered food ingredients. And this is overseen by the Agricultural Marketing Service at USDA. To remind you, bioengineered foods, genetically modified foods, have been sold in the United States since 1994. There are ingredients from bioengineered crops in many, many food products. 99% of the sugar beets, for example, which provides an enormous amount of sugar in the US, are GMO and so forth. The new disclosure standard went into effect on July 1 of 2022, the first time that a federal government required the disclosure of bioengineered foods to consumers. We had done some work prior to the implementation of this, giving some advice about some of the symbols. We then decided to look at it a year after implementation. So we looked at 1,840 participants recruited from a YouGov panel in February to March of 2023. So again, a year after. We looked at the various kinds of disclosures, the symbols, the text-based disclosure contains a bioengineered food ingredient, QR codes, phone and text instructions, a URL, and so forth. It's a very complicated thing. Each of these examples is its own presentation. I've got 3 slides each. Here's the results, selected results. Only 20%, only 1 in 5, had heard that there was in fact a new law which required the labeling of bioengineered foods. Only half believe that there are any foods with bioengineered ingredients in US supermarkets. Only about 1/4 believe they have ever eaten a food containing bioengineered ingredients. This was supposed to be about transparency. Which of these disclosures have you seen on a package in the last 12 months? A third said that they had seen a QR code. I think that that's an exaggeration a bit because they're confusing regular QR codes that are on it for lots of other things. The color bioengineered symbol by about 1 in 5 and so forth. So lack of awareness of the law, lack of awareness, lack of seeing these disclosures on a food package. 43% reported having seen none of the disclosures. So people, it's not having the intended effect of transparency. But had they seen, they had seen the USDA organic symbol and the non-GMO verified symbol, both of course, which indicate that the product contains no GMOs, no bioengineered food ingredients. So we should think about that. We also need to anticipate and evaluate unintended meanings and their consequences. When we showed people, oh yeah, this is what it looks like, 62% thought it was a health warning. So perhaps an unintended consequence. We also know that people are using label information as heuristics, so as shortcuts to know whether they should buy something and whether it's healthy. Non-GMO is one that lots of people use, and that's one of the reasons that we see non-GMO labels on products that couldn't possibly be GMO. But USDA organic and all natural and local are other kinds of things that people are using as a heuristic to think that this is a product that they should buy. Similarly, vegetarian and vegan, plant-based, people are using as a heuristic. And one of the questions I ask as a psychologist, when people, for example, buy a plant-based burger at their favorite drive-through, are they misinterpreting that? Yes, and it is in fact plant-based. It's based on pea protein, but many people think that plant-based means that they're actually getting all the nutrients of a salad, and they're not. And do they then engage in compensatory behaviors and think, oh, I've got a healthy plant-based burger, I'm going to have a large fries to go along with it. You laugh, but people do this. Real ingredients as a heuristic, clean label as a heuristic. I'm concerned that ultra-processed will ultimately be used as a heuristic, and we've talked about that. Free from is used as a heuristic, pesticide-free, antibiotic-free, all of these things. The metaphysically impossible chemical-free are all used as heuristics to think, well, I know this one thing, so it means that it's healthier based on that one thing. We also know that single nutrients are used as a signal that food product is healthy. My student, Gina Pope McCain, did her dissertation on protein health halos, comparing the real product packages of Special K protein and Special K original. Most participants perceived Special K protein as healthier, more nutritious, less tasty, but providing greater health benefits, including being more likely to help them build muscle, stay healthy, and live longer. But many perceive no differences in the amounts of sugar, sodium, or calories between the two products. And of course, Special K protein has more sugar and sodium to make it taste better to cover up for the addition of the protein. When you actually equalize to 1 cup servings, Special K protein has more of all of those things. Most reported viewing the facts up front label, but only 1 in 5 correctly chose Special K as having the larger serving size. So we need to be also clear about evaluation criteria in advance of our work. We need to use a systems approach. So this is one of the things that we are working on now, evaluating the appropriate nomenclature for cell-cultured proteins. We published 3 papers. Thank you. 3 minutes. I got 3 minutes left? Oh, okay. So we've done 3 papers on this. If you're interested, I suspect the slides will be available later. You can always click on the QR codes, transparency, to get these particular papers. One of the things that we did going into this was to specify our criteria for evaluation before we actually did the studies, knowing that common or usual names have to communicate to consumers without prior or additional explanation. Well, why am I dealing with common or usual names? Well, what you call something is really, really important because it creates a frame, it creates a lens through which the product is actually seen by consumers. And marketers spend a lot of money trying to figure out what to name their products. I also did some of the first work on public perceptions of GMOs, and I would prefer that the industry not make the same mistakes. So let's look at this particular group of products. So we can't rely on consumers to already know what the product is. Most consumers in our research show that they're unfamiliar even with the idea of cell-cultured proteins. There are no products on the market. There's one that's been approved. There's a chicken product, but it's really not being sold. And there's been relatively limited media coverage. I want to emphasize that this is not simply a marketing exercise. Unfortunately, most of the research that's been done has been focused on what name is most appealing to consumers, taking sort of a single-handed approach. So we established key regulatory criteria. We decided that an appropriate name should enable consumers, for example, to distinguish cell-based proteins from their conventional counterparts. Following it must be truthful and not misleading. You shouldn't mistake it, for example, for cell-cultured, for farm-raised or for wild-caught, or frankly, with plant-based. Secondly, they should recognize that if you're allergic to the conventional product, you shouldn't eat this product because it's got the same allergens. And for companies to be willing to accept this name, it should not disparage either conventional products or these new products. And it should not evoke thoughts or images or emotions that are inconsistent with the idea that the products are safe and healthful and nutritious. And finally, it should be seen by consumers as an appropriate term to identify the products. We laid these things out. We did the research. We made comments in the request by the FDA. And what I want to underscore here is that the industry, the Center for the Science and the Public Interest, the Environmental Defense Fund, the National Fisheries Institute, all cited our work, and particularly the criteria by which we did the work. And it's the first time I'm aware that they've ever agreed on anything, because we did it in advance and we went to some of the stakeholders and talked with them about this. Finally, to reinforce what you already know, resources are necessary to conduct rigorous evaluations. You need the expertise of social scientists. I'm a psychologist. How many other psychologists are in the room? Yeah, this is what happens. I'm typically the only psychologist here. And that's a problem, because it's not about information, it's about changing people's behaviors. And finally, I want to highlight this for the academics in the room for whom this is not a reality. There is this evil thing called the Paperwork Reduction Act, which requires federal agencies to obtain OMB approval prior to collecting information from 10 or more people outside the federal government. You have to file an information request describing the information you're going to collect, why you need it, the amount of time and money it's going to cost, and you have to have approval before you actually begin the research. If you've ever been through this process, it takes 6 months to a year if you know somebody at OMB. And unfortunately, a lot of the information we really desire needs to be on a sped up timeline. We really can't wait for that. So approval takes time. Finally, if this is where you can find me if you would like to complain or like more information. And I'd also like to note that these statements have not been evaluated by the Food and Drug Administration, and this product is not intended to diagnose, treat, cure, or prevent any disease. I look forward to the questions. Thank you. There was a bet as to whether I could do it in 20 minutes or not.
All right, well, good afternoon, everybody. It's a little daunting task when you're one of the last people on the program. I've been looking all day at the positioning of this podium, and it's, give me a little bit of concern that there's a door right behind here. Nobody's run out yet. No, you didn't run Bill out for being a psychologist, and I hope you don't run me out for being an economist, but we'll see how it ends up. I was asked to talk a little bit about, how you evaluate policies and do sort of impact analysis. And from an economic standpoint, really our standard go-to kit is cost-benefit analysis. But I will say we do bring a particular bent to our way of thinking, and often I find myself at odds sometimes with nutritionists and epidemiologists, because I like to draw a distinction between what science can say, which is there are increased health risks from consuming certain kinds of food, which I think are different than what you should do, which involves some mix of values and preferences. But nonetheless, we do take, estimates from nutritionists, epidemiologists on certain eating practices. If it saves lives or reduces rates of diabetes, often we can come up with benefits by applying value statistical life, those sorts of things. But I think one, and it's going to dovetail very well with what Bill talked about that I've spent a lot of time thinking about, is information. And when is information valuable? And that really comes about, I think, because of a lot of situations where consumers don't have full information. We can never really be fully informed, so we're making decisions in some sort of ignorance. But most of my talk is really going to focus on research methods that economists use. And you might think, why would we want a research methods talk here? But I think we can all appreciate that we're living in a world now where there's probably been as little trust as has existed, at least in my professional life, for experts and expert decision making. So I think whatever we can do to make sure that the information that we create and put out is credible and trustworthy is only to our benefit. I'm not going to talk here much about firm behavior. I'm going to talk about a lot of consumer related studies, but I did want to at least put one bullet point, which is if you really want to do a good impact analysis, you need to also calculate and study how firms are going to respond to the policy. Firms are not idle. They're not idle participants that just take whatever the policy is given. They change formulation, they change prices, they change advertising, and so forth. Okay, so information, as I said, is really one thing that I think is a big variety of policies that we might want to study. One thing that I think is important to point out is when you think about information, the value of information, at least economically, is not derived from asking people if they've seen the information. It's not derived from asking people if they use the information. It's not derived by asking people if they're willing to pay more for a product with a label versus another. Really, it's related to whether that information changes behavior. That's where the value comes from. The conceptual thought experiment is imagine that you've made a set of consumption choices. I inform you, you have better information now, but I don't let you change your behavior. How much would you be willing to pay me to allow you to change your behavior now that you're more informed? That's the value of information. You can think about it several ways, but that's one of them. And so we've done a variety of experiments in trying to think about how we value information. One is what you might call an indirect approach, which is we give people information and see how their behavior changes, and we do some math to calculate what that value is. That can be a within subject design, so people make choices. We give them some information, we re-observe those choices. I tend to like my studies. I do a lot of surveys like Bill does, but I also do a lot of non-hypothetical experiments. I think if you could make people put their money where their mouth is, we can get better and more accurate information. Or we could do between subject studies. You know, one group of people makes choices without some information, another group of people does the other. But the important point here is what we want to do is observe what behavior changes. The last thing we can do is we could just elicit how much people are willing to pay for information directly. I'm going to show you a couple examples of those. So I know we had a discussion very early this morning about tobacco and tobacco labeling. So this is a study with one of my colleagues, Matt Rosu, where we recruited smokers as they came into a smoke shop, had them bid in a non-hypothetical auction on different packages of cigarettes that had different labels. And so we looked at several different labels on the front, on the side, with graphics on them. The top little graph on your top right, which you probably have a hard time seeing, is really how did people's value for that, their willingness to pay for that pack of cigarettes change when they had a front of text versus when the text was on the side. And you say for some people, their value was lower. For some people, it was exactly the same. And for some people, it was a little bit higher. But down below it is our math of, well, what is the value of that information? And you can see for a big chunk of people, if it didn't change what you wanted, what you chose, it really has no value in an economic sense. But you can see on both the left and right tells there was indeed value of that information because it led to behavioral change. Let me give you an example of a more direct approach. This is not under FDA, but USDA was tasked with implementing a policy, which is no longer on the books, for mandatory country of origin labeling of meat. And big discussion about the value of that policy. And so one of the ways we tried to determine this value of information was to recruit people in a grocery store, offer them a free steak. It's a way to make a lot of friends and recruit participants. The free steak I gave them, I said, you can pick that free steak out from this red cooler. It's free, but in that cooler, there's a bunch of packages of steaks from different origins, and you don't know which origin any particular steak came from, but they're roughly distributed according to the origins of steaks that we have in the United States. Or you can pay me a little something and pick out a steak from the blue cooler that has steaks with labels from where their origin is. The important thing is I never even observe what origin steak they chose, but rather what I did observe is were they willing to pay me a little something for the privilege of having that information. So on average in this study, we found that people are willing to pay about $1.37 per choice just to have the information regardless of what steak they chose. One thing that you may find interesting though is what was the single most determining factor of that value of information? It was something we call ethnocentrism, essentially sort of a patriotism applied to meat. So it's a big scale of items. You ask people like, how proud are you to be an American? And my citizenship is on good, well displayed when I'm buying American-made products. Like it's that kind of thing. That's not normally what we think about when people are valuing information. We might think thought they were getting something safer, or tastier, or whatever. But you might find that interesting. The other interesting thing is in the study that value of information was much lower among people that bought a lot of meat. So it speaks to aggregation, right? If you just took that $1.37, multiplied it by the population, that really wouldn't give you the correct value of information because people buying the most meat cared less about the value of information. We did a bunch of studies back when calorie labeling was being mandated on restaurant menus. So one of my former students, Brenna Ellison, who's now on faculty at Purdue, she did her dissertation work by working with a local restaurant. And when people walked in the door, they randomly got one of three menus: the standard menu, a menu with calories in parentheses, which you can barely see up there, or a menu that had the calories plus a traffic light system. Essentially, the highest calories had a red, middle had a yellow, and the lowest calorie items had a green. And we did several things. Again, people over the course of about half a year randomly got one of those menus. We also raised the prices of the highest calorie items. Other people got menus where it lowered the prices of the lowest calorie items. And so you can sort of see on the figure there, you know, what happens in terms of the average calorie intake from that menu. So in the status quo, no information, prices at the status quo, about 640 calories were ordered. Making the low calorie items less expensive brought calories down a bit, but you can see the most effective, if all we cared about was calories, was the traffic light system. So 584. Although interestingly, we could translate by changing prices, we could get people's sensitivity to prices, and then we can, again, do some math and make a calculation about the value of that information. And you can see it's kind of small in a way. Just the calorie information alone was worth about 3 cents a meal. That's how much it changed people's behavior on average. The traffic light system was created a value of about 13 cents a meal. So a little bit more, but it goes to show, like, if you're trying to just do a cost benefit analysis, looking at calorie change may not give you a good sense of what that translates into in terms of dollars. Speaking of price policies, and I want to talk about, sometimes it's hard to even do cost benefit analysis because you get these equity issues that you also want to talk about. So along with some colleagues in France, Laurent Muller, Bernard Raffieux, they did some studies with high and low income consumers in France. This is a non-hypothetical study. We recruited people into a lab setting. They made a day's worth of meal choices, both under a status quo and then when we changed prices. In the one example I'm going to show you here, we made fruits and vegetables about 30% less expensive, and we made the unhealthy items, we had a particular way of measuring that, about 30% more expensive. But I want to show you something here about the sort of how it affects those different groups of consumers, high income versus low income. The average subsidy received from subsidizing fruits and vegetables, higher income consumers received more of the subsidy than the low income consumers. And then in terms of those taxes, think about the fat tax, the high income consumers paid less tax on average than did the low income consumers. In fact, the overall cost of the meal that people chose, or their daily bundle of foods, was much lower for the high income consumers. Why did that happen? Well, two things happened. One, the high income consumers were already eating a healthier diet. So they started off in a world where they were already consuming more of the healthier things and fewer of the unhealthy things. But this one kind of surprised me, particularly as an economist. You would think low-income consumers ought to be more price sensitive, but at least in the context of our experiment, they weren't. They were more habit prone. The low-income consumers tended to stick with their original diet more, so they didn't change as much. And if I had more time, I'd explain how this graph shows that, but the most important thing is there. Price policies, I think, are interesting, although I do want to make a point here that in a conventional economic analysis, if you assume consumers are completely rational, you can never make a consumer better off by increasing prices. So when you talk about fat taxes and you talk to an economist, you need some other model of how you make people better off. You can say I'm paternalistic. I think they don't know enough about their future self. Most people don't like to admit that. Or you got to conjure up some behavioral model. And so I conjured up in this example a simple behavioral model, which is you have two functions. One function decides how much you eat at any given time. And then another one is another function that decides is a future self that decides how much you just ate, how much pleasure that gives you later. So for example, when you're making consumption choices now, you may choose to ignore, maybe you're not choosing to ignore, maybe you accidentally ignore the future consequences on your weight and health. And so if you have a model like that where one function is governing your choices and another function is governing your well-being, you can derive situations where a fat tax can make you better off, but it isn't a very limited set of situations. In this little simple example, it's when prices are really high and people are only consuming at those high prices because they're ignoring their future health benefits. So when you think about implementing policies, say FDA or what have you, I think, experiments are really important. And I think it's sort of the gold standard of causal identification. And so I think one way, and this might be hard to do, but by sort of random rollout of where policies hit, either in geography or perhaps even in time, is a way to create some random variation that might be able to be useful in identifying the effects of a policy. The other thing, and I'm thinking in here particularly of that traffic light implementation. Sometimes it's not so much whether a policy can pass a cost-benefit analysis, but what kind of policy implementation. So Bill just gave us several examples of ways bioengineered foods could be labeled. Some of those are probably more valuable than others. So it's not whether a policy could pass a cost-benefit analysis, but which one. And you can only really learn that, I think, by doing some of this kind of socioeconomic analysis. Another approach that's really taken off, I think, in the economics literature, again, we'd all like to do experiments, randomized controlled trials if we can. It's just not always possible. But there are some things that I think are particularly relevant to nutritional policy, which might be useful. And there's something that economists have used a lot recently called regression discontinuity designs. And essentially, what you're doing is exploiting some arbitrary threshold in the data. So this is a paper that looks at what happens to driving accidents just before and just after turning 21. What happens at 21 years of age? You can drink legally. Okay, so essentially what you're doing is the people that are just below that 21 cutoff, they're 20 years old in 11 months. You can compare them to somebody that's 21 years old in just a month. Shouldn't really be any difference between those two people of any meaningful difference between them. But what you can see is there is this jump there that happens. There is a sharp discontinuity there in traffic accidents that occur right on that birthday, which tells us that there probably is some relationship between allowing access to alcohol and in driving accidents. So, one way to think about that is when policies are implemented or that exist, are there these arbitrary thresholds that exist? So age was one that I just showed you, but income could be another one. Nutrient content, maybe there's some policy that kicks in right when you pass some nutrient threshold. Sometimes firm size, we're going to impose this. If you have a certain number of sales, this policy you're required to implement versus you're not. Time is another one that you just think about in terms of an event kind of study analysis. I'll give you an example of another one of these there's lots of examples of these kind of studies, but sometimes you implement thresholds like firm size because they're related to cost of implementation, but there would also be a benefit occasionally to doing that because it helps you identify this effect. Now, what you want to make sure is that firms aren't responding to that and like reducing size to get just under a threshold. So you got to make sure that threshold is something that is not manipulatable. Another approach I think that's really become in vogue in the economics literature is difference in difference designs. And again, the idea here is to try to create some counterfactual. Is there some control group that you can benchmark against the intervention group prior to the policy being implemented, that after the policy is in place, then you can see this jump that happens. So this is just an example of implementation of animal welfare law in California that increased cage spaces. So prior to that regulation going into place, eggs in California were about 62 cents more expensive than they were in the rest of the country. Afterwards, that gap, the difference increased even higher. This animal welfare law led to higher prices for consumers. Another approach I think that's common is something called instrumental variables. And again, the idea is to try to create some quasi experiment. Think about an instrumental variable, the way I think about it is just like a coin flip. You need some variable, like a coin, that is strongly determines whether somebody's assigned to a treatment or control, but is not directly related to the outcome itself. So one example of this in the nutritional literature is location of fast food restaurants and health outcomes. So there is a correlation, an association between fast food restaurant locations and obesity. But is it causal? Because you could say, well, maybe fast food locations are going to where obese people are because they can have higher sales there. So several papers have identified a good instrument for that, which is the introduction of exits on interstates. So that's directly related to whether a fast food chain locates, but not probably directly related to obesity. And there's several papers on that topic that use exits as an instrument and find some causal relationship between fast food locations and obesity, at least in some populations. Last point I want to make on here is really just about something kind of Bill also touched on, which is policy popularity is not always related to cost benefit analysis. There are lots of things that drive, people have meta preferences for policies. Sometimes policies act as signals. So like a fat tax, sometimes if let's say a soda tax, there's an example in Berkeley where sales actually started falling for sodas before the soda tax was implemented. Why? Because people, first of all, they're exposed to all the campaign discussion around it, but then they were also thought, well, if we're going to implement this policy, maybe drinking sodas isn't as healthy as I thought it was. People can also go the opposite way. How dare you tell me not to drink soda? If you're going to tax us, I'm going to drink even more. Don't tell me what to do. and people suffer from this form of reactants. I want to give you one example of this, and I'll stop. And just to tell you that policy preferences are complicated. Here's a map of California. I've got counties shaded in according to how they voted on a particular policy issue. So you can see kind of the coastal areas shaded. They voted in one way. Central Valley, where all the agriculture is, voted in another way. My question for you is, what policy do you think this is? Any guesses? Well, it's actually same-sex marriage. But what I want to do is show you that map of California next to mandatory GMO labeling vote. By the way, I could also put up the vote on the animal welfare ballot I had before. It's not a perfect one, but you can see it's pretty darn close. And in fact, if I take the vote share in each county and just plot it against each other, I could, with more than 70% accuracy, take how a county voted on same-sex marriage in 2008 and predict how it would vote on GMO labeling in 2012. You must be asking yourself, what does same-sex marriage have to do with GMO labeling? And the answer is nothing, except that it tells you that our views on political issues are complicated and they're tied up in a whole set of cultural norms and cultural issues that often have very little to do with each other, which tells you what hope did people think giving people scientific information about GMOs was going to have when this is how our voting preferences are determined. I'll show you one last slide and then I'll stop here. On a little bit unrelated to this, it's not about political preferences, but it's very much on the point that Bill mentioned about how people perceive words and information. So on this topic of healthy, what does healthy mean? I asked over 1,000 respondents in the US 2 questions. One was, which of these following best matches your view? One was, if I know the nutrient content, I can know whether a food is healthy or not. Or I need to know more than the nutrient content to know whether a food is healthy. You can see it's roughly split, about 50-50. Some people say, no, you tell me how much fat, protein, I can know. The other was no. All right, very similar question, but now I said, is it better to think about healthy on a food by food basis? Like some foods are healthy, some aren't. Or do I need to know your overall dietary pattern to know whether something is healthy or not? Again, you can see sort of 50-50. Now, if I take those 2 questions and overlay them with each other, it's like a scatter shot. You got about 25% in each category. In other words, if you use the word healthy, there's only about a 1 in 4 chance that you're using the same thinking as the person on the other end of that communication is having with you. So it's just, again, kind of makes a challenge. So I'll stop here. Just a call to us all to do what we can to create credible and trustworthy estimates, causal estimates, I would say, of the impacts of policies. And again, we should do cost-benefit analysis, but know that it's not sufficient to know whether a policy is popular or not. Like Bill, you can find my email and tell me hateful things if you really want to later.
Thanks so much. And I know actually we were going to give people online the opportunity, but we don't have any questions yet. So if people in the room have some questions, you can come up. I'll kind of start off. Given you guys both talked about labeling, and that our regulatory agency obviously does a lot with labeling, and it has to be accessible to all US consumers with very diverse backgrounds. And I think you guys were kind of in both of your examples showed how different backgrounds can have different impacts. Given that we have very limited resources, what analyses and evaluations do you think we could tackle to see if our policies are being helpful?
Well, I'll also add, in addition to this being complicated, all the labels are primarily in English. And not everybody speaks English and not everybody can read English. So you're already missing a significant portion of the population. So what do we need to do? Well, given that you've got limited resources, one of the things that we need to do is to work together. NIH, the FDA partnership here is great. There are a lot of people like Jason who have really great ideas that should be funded by the NIH, should be funded by FDA.
Yeah, that's a great idea.
The question is, how much are you willing to pay for Jason's expertise? No, but there are lots of academics, there are lots of folks who don't have a dog in the fight, who can do a lot of this research. And I think the trick is to coordinate it, to coordinate the research that is objective, and to work with NIH, to work with FDA, to work with USDA, as we did, for example, talking to stakeholders about, well, what are the criteria? What would make sense for us to measure? What would be useful to you to know in order to hone the policy? Many of us are willing to do this. Many of us are paid to do this. And I think, that sort of cooperative understanding is really, really helpful, can be really, really helpful. I also think it's possible to reach out to particular groups of folks. So in New Jersey, there are groups of people, at least 60 languages are spoken in New Jersey. It's possible to work with some of those groups to figure out how this translates to particular cultures, for example. How this translates in a particular language. I'm a big fan of, for example, individual ID, either QR codes or enhanced barcodes for products. Why can't we have those? Why can't we have my smartphone scan that and have the information translated into Spanish, or the guidance in Tagalog or Hmong or whatever? There's really no reason that we couldn't do that. And that's how we could reach a wider group of people with the same information. So let's have a drink. I can talk more about this. So.
Do you want to go?
Yeah, Shariki Kumanyika, UPenn. You haven't talked about why manufacturers like to put certain things on their products. So there's the factual information, and then there are the claims, some of which are actually misleading because it couldn't have been in there anyway, just something like that. Is there another line of research that looks at what manufacturers perceive as, are they closer to what the consumer is thinking than we are, so to speak?
Do you want to start with that one?
I mean, I think we know why they do it. It's because they think they can make more money doing it. And you're right, they spend a lot of time hiring people like us to look and see what will move the needle for consumers. And I think in part, if I could piggyback on what Bill said, it's not to level the playing field. I'm just saying, if you want information to be effective, a business is not going to randomly pick some label that meets some legal guideline. They're going to do a lot of A/B tests over and over and over again with a lot of focus groups and a lot of big studies to decide what to put on that package and label. Doesn't mean they get it right all the time, but they're motivated by a profit margin, and it's not always short term, but long term. I think they don't want to necessarily risk short term gains over their reputation, but I think they spend a lot of time and effort trying to understand how their claims, brands, and everything will cause people to choose their product more often. I should say, by the way, I think companies are definitely in it for the money, but I should also say the food business is very low margin business. That there's not a lot of exorbitant excess profits being made in this industry, and so. I know there is this view that the companies are causing all these outcomes for consumers, and there's probably an element of truth to that. But at the same time, these companies are in very fierce competition with each other, and if they can't get a little market share, there's a good chance they may not be in business tomorrow too. So it's even for the companies, they're doing a lot of this work because of even to eke out the 2 to 3% margin they can. They've got to sort of stay on that treadmill of trying to stay on top of whatever it is will get consumers to buy their product.
So I'll follow up on that. Most of those companies have way more resources than FDA and USDA. I think that's a given. And a lot more expertise, a lot more experience, I think, than most federal agencies. I know many people in the food industry, as I do in FDA and USDA, and there are people in the food industry who really want to do the right thing, who want to create products that are healthy, that consumers will choose. The problem is that those are often incompatible. That when you make products that are better than, consumers don't always choose them. And so the question really is, how do you create products and market them in a way that helps consumers make better choices? It's not a given. It's really not a given. We talked about ultra processed and hyper palatability. How do you make that happen? How do you get people to choose the better product? It's really, really difficult. So I don't want to, totally demonize the industry. There are really good people trying to do really good things. It's just really, really hard when you're up against freedom of choice.
Yeah. So let me just ask a quick question to clarify. I mean, these answers have been helpful. From a regulatory perspective, and this may be for Claudine, can the FDA put anything on the package other than a nutrition facts label or something like the healthy that's going to be, I mean, so is it like one side can say anything it can get away with and the other side can give you information that we now know makes no sense at all, right?
Kind of just speak a little bit, we still have several speakers have alluded to the First Amendment. So we definitely have to do that. So there are certain restrictions or levels that we have to meet to mandate that speech. So what they always told me when we were mandating new things like added sugars, we have to show that it's actually they have to have it. It's not just a nice to know. So the bar of evidence that you need to mandate that speech is very high. So not to say that we can't mandate things, but we have to have a lot of evidence to show that it's not just a nice to have, that it's a has to have. That's kind of putting it a little bit in layman's terms.
I think we need to remember that the front of a package is expensive real estate. And as many things as we would like to be on the front of the package or the back of the package, there are realities here. So we need to have some priorities.
We'll make this the last question because I want to make
great.
sure everybody gets home on time.
Shuan Ong, University of North Carolina in Chapel Hill. So thinking about the presentation that Kevin made earlier today that showed the energy density actually was a key factor as well in terms of, affecting weight change and fat mass. And Dr. Hallman, your presentation about the cereals and like the bowl of cereal that probably was like 28 ounces. And the other work with your student that showed that compared to 2 Special Ks and the different serving sizes that they put on the front of pack on the GDAs. I guess I was sort of wondering, why doesn't the FDA require the nutrient on the nutrient facts to be reported in terms of per 100 grams or some standard measure across products, because it will make comparison a lot easier, much more straightforward. It still requires some math and some degree of numeracy, but at least it makes comparing the 2 types of Special K, for example, much easier, right? Like it's the same denominator. So that's one question I have. I'm curious about the FDA sticking to
I can
racks.
probably have that. So we do have that, and the racks are similar for similar products. For cereals, there's different types of cereals, like Bill alluded to. So there's our different kind of serving sizes based on our reference amounts customarily consumed. That's what racks are. For like heavy cereals like granola versus like light cereals like cornflakes. But we do have that, so it's very comparable. And for like the serving sizes, that's actually in the statute. So that's not something we can actually change. If you want to change something like that, that would
it's just
actually
interesting because
be
many other
Congress.
countries are using units of per 100 in Europe and Latin America. In Canada, it's all per 100 grams, 100 ml. So the US is a real anomaly.
Well, I will say it's more of what people are eating. For some things like eating 100 grams of butter, there's probably not a lot of people who are going to eat 100 grams of butter and like a thing. So
You
it
haven't
does
met my
work for some
brother-in-law.
things, but for other things, maybe not.
Okay, thanks.
All right, so thanks everybody for coming today. I'm going to do a really quick wrap up because we only have a few minutes left, but I think it was really good to kind of go over with the discussion. So just to kind of give a little bit of a summary, some of the goals of our workshop really were to discuss the strategies for conducting nutrition science and really how those could be informed for our food-related policy and regulatory decision-making. It was also to identify critical research gaps and resources related to nutrition regulatory science and kind of articulate how NIH can support FDA in conducting that kind of science. And also to provide some examples for how NIH and FDA can work together to support the research and address the regulatory process. I think with looking at today's meeting and our sessions, I think we really accomplished a lot of that. I know we started off today just really identifying what NIH and what FDA do, and I think some of our goals are a little bit different. In the first session, people were really saying, especially about the research, we look a lot at cost benefit analysis and, you know, what's going to show have the most benefit. That's not always what NIH looks at. They sometimes look more at mechanistic. So I think it's kind of making sure that NIH can kind of help leverage some of their research that can help inform our policy. I think a lot came out in a lot of our sessions around kind of some of our regulatory requirements and the First Amendment and kind of mandating speech. We work under restrictions. We can't just slap everything on the label. So I think that was really important. And I think we also heard a lot about some of good models about the research with the tobacco as well as the trans fats. We had a really robust discussion, I think, on ultra-processed foods as well. What are they? A lot of debate over formulation and processing and kind of teasing those out. And I think something else that really came up is there's a lot of other considerations when people are making choices about foods and kind of their nutrition. Related to ultra-processed foods, about the longevity, cost, accessibility. It isn't just about like the nutrition. And then I think really following up in our last session, looking at consumer understanding of that. And I know in the ultra-processed food section, people were talking about what people were understanding. And I know there's lots of fads in nutrition. Like the carbohydrates are bad. You should have the low carbohydrate diet. I almost think the ultra processed foods, that's kind of, it may be sticking. A lot of these things do stick, but sometimes it's kind of the in vogue thing. And ultra processed foods is definitely the in vogue thing to look at now. And how do consumers understand that? I think Dr. Hallman pointed out that kind of understanding what it is that and where the peak consumers are starting at really can have an impact. And again, I think as kind of nutritionists and scientists, we're starting at a different place than a lot of the consumers. And then really looking kind of, FDA always is looking at kind of that cost benefits, because every regulation we do, we have to look at that. So we have to show that there's some type of benefit. So tomorrow we have a final session that's going to be looking a little bit more forward and looking at research opportunities, resources, technologies, and key priorities. So thanks everyone for coming today, and we look forward to seeing everybody tomorrow.